Okay, very good morning, everyone. Welcome to the day 3 of AgriVision 2024. We will now begin with the plenary sessions, but for the first session, we are talking on the topic plant and animal genomics and ethnobotany. Chairman Dr. A.K. Mukherjee, sir, Principal Scientist, Crop Protection Division, ICAR-NRRI, Cuttack, and co-chaired by Dr. Sarada Prasanna Sahu, Director, CCBF, Chiplema, Government of India. Rapporteur is Dr. N.B. Patil, Senior Scientist, ICR and RRI. The first lecture of the session is done by Dr. Prashan Kumar Mukherjee. Before the lecture starts, I would really want to let you know a little bit about the gentleman. Dr. Prashan Kumar Mukherjee worked as the head environmental biotechnology section and group leader, agriculture, microbiology, nuclear agriculture and biotechnology division at Bhabha Atomic Research Center, Mumbai. He obtained PhD in plant pathology in 1991 from GB Plant University of Agriculture and Technology. He was the university topper of BSc, MSc, and PhD, and he has been a visiting scientist and scholar at the Israel Institute of Technology, Haifa, and Texas, and A&M University and College Station. Dr. Mukherjee has been working on the biofungicides Trichoderma spp for more than 30 years and has made seminal contributions in genetics and genomics of these fungi, especially in the area of signal transduction and secondary metabolism. He has also developed novel mass multiplication method for which a patient has been filed and a patent had been filed and the technology transferred to private entrepreneur. He has published more than 100 research papers and review articles in the highly reputed journal like Nature Microbiology Review, Genome Biology, Microbiology, BBRC, etc. with 8,621 citation. He has won several awards for his research and contribution. He is an elected fellow of the National Academy of Science, India, National Academy of Agricultural Science as well. He topped ARS examination in plant pathology, joined BARC. He reviewed Dr. K.S. Krishnan DAE Research Fellowship. And yes, the discussion of his is on a video mode. So he is not present over here, but let's just go through this now. Yeah, he's present online though. At least we can clap for the gentleman and then I can start. Yes, thank you so much. So here we go.
A very good morning to everybody. My apologies for not being able to come personally. So I'll be presenting my experience on Trichoderma, the most commonly used biofungicide. The title is Basic Biology Meets Applications. Basically, I'll be covering both the basic research which we have done and also some commercialization aspect. As you know, like pests and diseases, they cause like huge damage to crops. According to this report, like it is 10 to 40%, but in certain cases, like it could be 100% losses. Traditionally, chemical pesticides and fungicides have been in use for management of these diseases and pests, but due to their toxic effects and environmental, side effects, many pesticides are getting banned, as you can see from this news article. Also here, many of them are fungicides. So basically, our options are getting limited to control these diseases. So we have been targeting pathogens like Sclerotinia routine, Rhizoctonia solani, which are very broad host range. Because they have very broad host range, it is very difficult or almost impossible to get disease-resistant plants. So either chemical or biological, these are two options, but they are like soil-borne in nature. So chemical control is also not very effective. These fungi produce resting structures which are called sclerotia. As you can see here, a close-up of a sclerosium. So these sclerotia, they survive in soil, and whenever they come in contact with the seeds or the root, they start infecting it. For these type of pathogen, especially biological control is a very good option. And as I mentioned earlier, like Trichoderma species are the most popular biofungicides. Trichoderma species have certain advantages, like they're omnipresent. There are more than 300 species recorded, but out of these 300, about 20 are frequently isolated from soil and rhizosphere. Most of the new species are found in forest, on deadwood, tree bark, and mushrooms. And some species are true endophytes, like they live within the plant. They are used widely as plant disease biocontrol agents. In India alone, more than 300 products are available. They also promote plant growth. They reported to control soil-borne foliar pathogens, fungal and bacterial pathogens, also nematodes, viruses, and in some cases insects also, because they can induce systemic resistance. Among the mechanisms of biocontrol, there are three very well-defined mechanisms. One is mycoparasitism, one is antibiosis, and one is induced plant defense. By mycoparasitism, we mean that when they come together, trichoderma can parasitize other fungi, as you can see from this picture. The thick one is the pathogen, and the thin one is trichoderma. So trichoderma can coil around and then lies the mycelium of the pathogen. They can also parasitize the resting structures, which basically survive in soil. So this is an additional, advantage with these biocontrol agents. In this assay, you can see the sclerotia are getting colonized by trichoderma, green growth on the brown sclerotia. And when they are plated on a selective medium, the sclerotia cannot grow as compared to control. So that means the sclerotia have been killed by trichoderma. Antibiotics, like they're basically secondary metabolites which are produced by microbes. And trichoderma also produce a lot of secondary metabolites, and some of them are inhibited to the pathogen, as you can see from this slide. And trichoderma virens, especially like which I work on, they produce viridin, heptadenic acid, cleovirin, and volatile sesquiterpenes. We have been able to, identify the genes for all these metabolites. I'll come to that. Trichoderma, volatile metabolites, they are very good, promoters of plant growth, especially root growth, as you see in this picture from our own laboratory. Arabidopsis plants are exposed to the volatiles of Trichoderma virens, and as compared to control, you can see the profuse growth of the root. There's no contact with Trichoderma here. Only the volatiles, influence the roots to grow to such an extent. Trichoderma can colonize the roots, as you can see here in the left side, without trichoderma and with trichoderma, extensive growth on the root. They also can go inside the root. And when trichoderma is inside the root, they can induce resistance in the plant, like in the leaf also. So if you inoculate the leaf with a foliar pathogen, it's very likely that if the root is colonized by trichoderma, the disease reaction will be less compared to the lesion size will be less compared to the non-inoculated control. So we have been working on all these three aspects, identifying the genes using knockout, knockdown, and overexpression, the genes which are involved in mycoparasitism, secondary metabolism, and induced resistance. I'll give some examples from our studies. First, the mycoparasitism. So we have identified two genes. We have identified many genes, actually. I'm showing data for two. One is called PGOI gene, PGOI1 we called, and one is ECM33 gene. So you can see that wild type, it is overgrowing sclerosis ulcerae. But if you remove this gene PGOI1 or ECM33, actually the pathogen is overgrowing trichoderma. So it has lost the mycoparasitic ability on sclerosis ulcerae. Similarly, on Rhizotonia solanae also, if you can see here, like wild type has completely overgrown Rhizotonia solanae in 6 days, whether PGOI knockout mutant and ECM knockout mutant, they are yet to overgrow. They cannot overgrow basically, very slow on Rhizotonia solanae also. So so this is this is an example where we identify the genes which are basically involved in mycoparasitism. Induced defense also we have been working, especially the identification of the elicitor proteins or effector proteins. There was a study in Israel where they have done a secretome analysis of trichoderma when it is interacting with maize roots. So they have found one small secreted 16-rich protein, which is upregulated. Actually, 13 are downregulated and 1 was upregulated. So we targeted this upregulated 16-rich protein. This is the sequence of the protein. It has no conserved domain, no structural ortholog when we studied. So what we did is we expressed this in E. coli, this protein, and then in solution, it was characterized and it exists as a dimer. So we elucidated the structure of this effector protein, and surprisingly, even though there is no homology with any protein, known protein, the structurally it was very similar to ALT-A1 and PEVD1. So these proteins are ALT-A1, you know, like this is an effector protein produced by Alternaria, and PEVD1 by Verticillium. So both are involved in pathogenesis. But so surprising, it was like 8 to 10% only sequence identity, but the structure, it was very similar to these effector proteins known in plant pathogens. When we inoculated the root of maize with this protein, what we found is, and inoculated the leaf with Cochleobolus heterostropus, a necrotroph, what we found is it produces a bigger lesion size, which is not surprising because it's a necrotroph, and it induces salicylic acid pathway. So this particular protein induces salicylic acid pathway, and that's how the plants becoming more susceptible to necrotroph. We are testing if it is becoming resistant to biotrop and hemibiotrop. In terms of secondary metabolism, we have done again extensive studies. Objective was to identify the genes involved in secondary metabolism. So we started with mutation of trichoderma, and we were lucky to get 2 mutants of trichoderma. One is called G2 and one is M7. So G2 is an overproducing mutant for secondary metabolites, and M7, it does not produce or produces below detection limit of secondary metabolites. So M7 we have used as a genetic tool for unearthing the secondary metabolism related genes, and G2 we have used as a developed into a commercial product. So I'll come to that later. Most secondary metabolites are produced by gene clusters. What do you mean by gene clusters is like there are several genes which are together and they produce the code for the same metabolite. Different genes do different functions, and then, as an example here, it is gliotoxin gene cluster, which is produced by Trichoderma virens, Q strains, and also by Aspergillus fumigator. So, these gene clusters are very often horizontally transferred from one to the other. We have isolated or identified the gene clusters for gliovirin, which is a non-ribosomal peptide. It's a very strong antibiotic against oomycetes. So using gene knockout, whole genome sequencing, followed by gene knockout, we have identified these metabolites, these gene clusters. And also we have identified the same way viridin gene cluster. So 21 genes, they are responsible for production of one compound, that is viridin. Heptuluric acid, again, it's a very important secondary metabolite. And interesting thing here is that we have found out that 2 gene clusters, independent gene clusters, they are responsible for production of heptuluric acid. So one cluster, it takes the terpene cyclase, it's a terpene, and other cluster, it takes the 3 cytochrome P450 monooxygenases to produce the compound. Siderophores are very important for competition for iron in the rhizosphere. So we have also been able to identify the siderophore gene clusters and also proved that they are producing siderophores by gene knockout. The Vir cluster is for volatiles. I told, like Trichoderma produces both the volatile and non-volatile metabolites. So this is the cluster which is responsible for production of 22 volatile sesquiterpenes. We knocked out the Vir4 gene, which is a terpene cyclase. And as you can see in this picture, the red one is the wild type and the blue one is the mutant, knockout mutant. So almost all the volatile metabolites are gone in the mutant just because we knocked out one gene. So this gene cluster was identified as a volatile metabolite producing gene cluster in Trichoderma variants. Now we have done the basic research and now time to discuss about the commercialization. As I told you earlier, we have a mutant which is called G2, which is producing more secondary metabolites. As you can see here, wild type filtrate color and G2 filtrate color. G2 filtrate color is darker. If we add the filtrate to the medium, the growth of the pathogen is inhibited more by the mutant than wild type. Also in HPLC, we can see it's not a neat picture, but you can see it is producing more secondary metabolites. We did a transcriptome analysis also, wild type and G2 mutant. And again, we find 20 genes per secondary metabolites production are upregulated in the mutant. And also there are several other genes which are also upregulated in the mutant. G2 is as good as a wild type in high parasitism. So we did the microparasitism study. You can see here, against Clostridium rhabdosis, wild type and G2 mutant both are overgrowing and lysing the mycelium of Sclerosia. For parasitism of Sclerosia also, we have did the assay. So wild type and G2 both are equally effective. After knowing that G2 is producing more secondary metabolites and is as effective as the wild type in the mycoparasitism, so we tried to formulate it. We looked for different seed, different grains. Traditionally, jowar is used, but jowar is expensive. So we used tamarind seeds as a mass multiplication medium. Tamarind seed has an added advantage because they are very sticky. So you don't have to add any sticker in the formulation. So this was formulated as a talcum powder formulation, and it produces about 20 to 10^8 CFU per gram. Very easy to coat the seeds with this formulation. You take the seed, little bit of water, and then put the formulation required amount. We recommend 5 to 10 gram per kg seeds. And here it is 5 gram per kg seeds, and still you see the seeds are beautifully coated. We did a lot of studies in pot and small plots, wild type and mutant comparison, seed treatment. So here in one example where Clostridium rhabdosis was added in a huge amount quantity so that we get almost 100% mortality. In that disease pressure also, you can see the G2 is performing better than the wild type in controlling the pathogen Clostridium rhabdosis. We took up a trial to begin with in 2015-16 in Raipur. My friend, Dr. Cuttack from IGKV Raipur, he was interested to study this test in the field. And then we put in a small plot and then see the effect, seed treatment with Tricho, BRC, we gave the name, and control. Huge difference. This encouraged us to take up this further and then take it to the commercialization scale level. Then again, 2018-19, we took up a trial. This plot was not infested much with the collar dot pathogen, but the growth promotion effect was very much visible here. In farmers field, we took up trials in many farmers field over years. This is one example, where the yield gain was 4 quintal per hectare, which translates to about 20,000 rupees benefit per hectare. Seed treatment cost very little, like 100 rupees or so. ICAR got interested to study this in AICRP, chickpea in multi-location trials. So 17, 18, 18, 19, we have taken the trial. And here also you can see on an average, Trichobark formulation gave better control than other isolates. So that I will have not included that data because that can be seen in the next slides. But you see the huge difference between control and the treated. So ICR continued this trial for another 2 years. 2021, they have taken the trial in 7 locations, and along with our formulation, 3 other formulations are included. And you can see here, like, if you see the average, total average, like overall performance, our formulation was the best. And the average yield gain was 63% over control. Similarly, again, 21, 22, it was extended further to 8 locations. And then many other trichoderma strains were included. Again, here also it was showing the superior overall performance. And here yield gain over control was about 73. So encouraged by this, a frontline trial was also taken up in MP, FLD. So here again, you see like 24% increase in the yield. So that is based on these data, ICAR recommended seed treatment with trichobark for chickpea cultivation in 2022 meeting. We continue to take the trial in farmers field. Like 24 farmers were covered in this trial. Again, the average you see like control and treated, 24% increase. So it was 2.9 quintal per hectare, which again translates to about 15,000 rupees per hectare net profit. This was covered in a local newspaper in Chhattisgarh. And another trial in the farmer. We tried in soybean also in KVK. And again, here we found that in soybean also it is giving a very good yield, as you can see here. So here about 3 quintal per hectare enhancement was there, and it translates to about 11,000 rupees benefit per hectare. In 2020, again, we have taken up the trial. Again, we found the similar result. 4.2 quintal per hectare was the additional gain in yield, which translates to more than 16,000 rupees per hectare profit. In lentil, we have taken up extensively the trials in farmers field in West Bengal. In this picture, you can see like 3 years, 3 seasons. So average of 30 farmers field in 2 years, and then third year 10 farmers field. Again, taking all the averages, it is about 17,000 rupees per hectare profit. You can see a control and treated plot. The growth was robust, which we extended to the next year also, 21, 22, again 10 farmers. So here it is very like high big difference we got, like 674 kg per hectare, which translates to 37,000 rupees per hectare, and this is average of 10 farmers field trial. You can see the plot here, like treated, and T4 is control. The right side is treated, seed treatment. Very apparent, like the robust growth due to the seed treatment with trichobark. We took up the trial in paddy also. Again, Dr. Kotasthana was interested. He basically works on paddy. I know a lot of work is going on in NRRI, Cuttack, like very excellent work on trichoderma application in paddy for growth promotion and disease control. For our formulation also, we tried in transplanted paddy, where root deep method was used. And you can see here, like, again, we have like about 21% improvement in the yield, which was attributed to the, increase in number of tillage. We tried in cumin in Jodhpur. It's a cash crop there. So we thought it is very interesting to know if it is working in Rajasthan, which is a very different type of soil and environment. So it was found to be very good in Jodhpur also, in cumin. Other crops also like guar and chickpea, I'm not showing those. This formulation has very good growth promotion effect. You can see here in maize, again in Chhattisgarh, control versus treated, very clear. In spinach, Dr. Kotasthane wanted to take up a trial in spinach and then see seed treatment improve the seedling stand, because the soil having damping up issue. So you can see here the stand between treated and control, a huge difference. In Navsari, Gujarat, again a damping of infested brinjal nursery. We took up a trial. So you can see the control versus treated. Both the growth promotion and plant stand was excellent in the seed treatment. under conditions, field condition, in other vegetables also it was tried, and then everywhere we are getting like a bigger biomass, a greater biomass in vegetables. Again, Dr. Kotas, thanks to him. So after all these data, we had like confidence that yes, this is working in the field. So next challenge was to register this with the CIB-NRRI, which is very challenging. And most people stop there. But fortunately, we had a good support of our institute and then other places and of course ICAR. So we could proceed with register. As you know, it's a very complicated process. We need a lot of data, including like formulation, nature of formulation, then fingerprinting data, and then biological characteristics and chemistry of the product. Different tests we have to perform, whether there is a contamination or not. Then bioefficacy data, of course, field data, laboratory data, toxicity data for both mother culture and formulation. It's very expensive, but we could do it. And then mammalian toxicity, environmental safety testing like on mammals, bird, fish, invertebrates, or like earthworm. So all these tests, packaging and labeling, all these generated. And then finally, we could get it registered as a biopesticide. It was the registration was done by Mrs. MBio Limited. This is the first mutant microbe to be registered for field application. And then, the product has been launched already. It is called Viren. This technology has been transferred to 5 companies so far, including Patanjali and UPL. UPL, is a very big company. So is Patanjali. So we are hoping that many more products based on this technology will come out in the market. Those who are interested to read some of the publications can visit my Google Scholar page or write an email to me. I'll be very happy to. I acknowledge my collaborators, especially Professor Kotasthane. I told you many times, like he is the person who drove this technology to the field. Professor Rajiv Nath from BCKB, Dr. Saxena, Dr. Nilima Karmakar, Dr. Manish Kumar from Jodhpur. She's from Gujarat, Navsari, and my students and colleagues, Ravindra Bansal, Sharkane, and Sikha Pachori, of course, BRC and Department of Atomic Grant. Thank you so much.
So that was from Dr. Prashant Mukherjee. And now we'll move towards the next by Professor. Is he there online, sir? Okay,
Yes,
any
hello.
question? Any questions for sir? Okay, sir, seems like that was very clear. So probably no questions for now. Thank you so much, sir, for joining online as well.
Thank you.
Thank you, sir. Okay, sir, can we now proceed? Yes. Okay. The next lecture is by Professor Robert Henry, University of Queensland, Brisbane, Australia. He'll be talking on the topic genetic improvement of crops to balance the needs of food security and climate change. We'll connect him on Zoom call.
Good morning. Today I'm going to talk about genetic improvement of crops to balance the needs of food security and climate change. The roles that agriculture can play in addressing climate change are many. One of the things we often do is try to adapt agriculture to climate change, and that's been an immediate response that's been underway for a long time. More recently, people are looking to use climate change or use agriculture to mitigate climate change by reducing emissions of carbon and greenhouse gases in agricultural production. I wanted to talk about the balance of those with a third strategy or third important area, which is using agriculture to reduce carbon emissions in industries outside of agriculture. So in this, we're talking about using agriculture to produce renewable sources of carbon that can be used to replace oil for the production of not only biofuels, but bioplastics and chemicals, all of the things that we currently produce from oil. And this is particularly important for biomaterials, where we can't use things like solar and wind. We can do that for energy uses of oil, but not for the non-energy uses where we actually need carbon compounds. So agriculture is the crucial industry for addressing climate change in this aspect. And back in 2010, I wrote a book that really stressed the way in which food production and food security from agriculture needs to be balanced with the conservation of the biodiversity on which agriculture depends, and with the production of non-food products from agriculture. All three of these things are essential. We can't just conserve biodiversity or just produce food, or in fact just produce non-food products, but equally they all are required. So what we need is to be efficient, particularly in food, but in the non-food production from agriculture, so that it doesn't compete significantly or to any extent with food production. So the way that we prefer to do this is to produce particularly those biomaterials that represent the main products that are made today from oil. Energy is actually a minor use, although we have for some time been producing ethanol and biodiesel. Into the future, the things that will remain that need the use of agriculture will be long-distance flight, so we need sustainable aviation fuel from agriculture, and we need those platform chemicals that we use to produce plastics, carpets, fabrics, all of the things that we currently produce from petrochemical sources. And the favoured path to achieving this is what we call the biochemical route. In this path, we take the non-edible parts of plants, the residues and biomass that doesn't have immediate human food use. Using enzymes, we produce sugars, which we then, using microbial approaches, convert to the chemicals that we need. And this provides a path that replaces the refining of oil. One of the critical applications of this technology is the conversion of biomass to fuels, particularly jet fuel. And here we're depicting a path to dimethyl cyclooctane. This is a superior molecule, better than Jet A. Planes can fly further using this product. So we're talking about superior products derived directly from agriculture. And the need for these products will remain with us for some time. We're going to see aeroplanes become electric just as cars are, and but this will allow regional flights to be electrified in the relatively near future, as you can see on this timeline here. We'll start to see that already by 2025, and this will be widespread. By 2040, even short-haul flights and medium-haul flights may eventually become electrified. But the long-haul flights of several hours are going to continue to require a more concentrated energy source, a liquid fuel, into the future, even beyond 2050, with all of the technologies we're aware of at the moment. And so this needs to be sourced from agriculture. If we're going to reach net zero carbon emissions by 2050, we need to produce jet fuel for long haul from agriculture. The two preferred crops that we work with in this regard are sugarcane and eucalypts. One is an example of a grassy biomass, and there are many other grasses that are also candidates for this role. And the eucalypts are an example of woody biomass. Woody biomass has an advantage in that it can be stored longer than the grass-based biomass. And the reason that we focus on species like this is that they're highly productive. If we're going to avoid competition with food production, we need to only focus on the production of very fast-growing, very highly productive biomass. These are both species where 100 tonnes of biomatter per hectare per annum is achievable. In fact, 200 tonnes is now routinely achieved. Species that have significantly lower biomass potential really do risk the need to plant such large areas that they will actually compete with food production. And the technologies that we have that really drive the innovation here are the advances in genome sequencing, genome technology, and the advances in gene editing, allowing us to modify these plants to make them suitable for this use. Gene editing, we now have become routine in doing in the grasses, and so this is a way that we can modify the composition of the biomass to make these processes more efficient. We're also using high-throughput mutagenesis. We've developed a platform in sorghum. We have 150,000 mutants of sorghum developed in collaboration with the Carlsberg research laboratories. And this population allows us to very quickly, in a matter of days, find a mutation in any gene and phenotype it. And this is a way in which we can test very large numbers of genes to find the ones that will give us the attributes that we want. The advances in DNA sequencing that we've made allow exact gene modification, because now we have an almost perfect gene sequence that we can work with. We can avoid non-target changes. We can make quite deliberately the changes in the plant that we want. We've applied these advances, and here's some work headed by Adam Healy's efforts, where we've achieved a full sequence of the polyploid sugarcane. This is the last of the 20 major crops in the world to have a genome sequence. It's very complex, but it's now possible with the advances in technology that we have. And this means through the advances documented in the papers listed here, we've compared all the long-read sequencing techniques. We've looked at improvements in the chemistry and improvements in the software available for the assembly and analysis of these genomes. And in the last 2 years particularly, we've seen dramatic advances in those areas, which mean that this can become much cheaper and much more routine. We've got a number of reviews that have followed this advance. What we've done for sugarcane is come up with high-quality reference transcriptomes using long-read sequencing and various other techniques that mean we now know of all the genes that are expressed in the plant, and this allows us to better target efforts to manipulate them. We've also got a greater understanding about how the composition of the biomass influences the efficiency with which we can process it to produce these desired end products. We've also, with IoT Delhi, we've got a major program understanding the chemical engineering options for more efficient conversion of sugarcane bagasse, particularly to high-value chemicals. To underpin this, we've focused a lot on the sugarcane metabolome and proteome and understanding the major differences that we have in the current gene pool, the diversity in which we can currently select, as well as the things we might change to go even further. We've got great reference transcriptomes that have been repeatedly updated, and we've made a number of discoveries in how you should do that. We've discovered that the very long genes in plants are not represented in most of the analyses that have been done in plant science to date, and we've now corrected that, so we're getting a much more complete picture of all the genes in a plant and being able to target all of them. And in sugarcane, we're getting better understanding of where in the plant genes are expressed. We're going to extend that soon to the individual cell level and individual tissue level to understand the exact pattern of expression of the very complex and large numbers of genes in a plant like this. A lot of the work's been done on the leaf, but the real focus is on the stem itself, the culm, the part of the plant that we use industrially, and that's where we need to understand the role of each of the cells in determining the composition of the biomass and how we can improve that. So by looking at all these different stages of development, we've looked at more than 1,000 transcript libraries from sugarcane to build up a picture of how the sugarcane metabolism changes throughout the season in different parts of the plant. And this ultimately will tell us how we can modify the composition so that a higher proportion of the carbon in the plant can be converted to these desirable high-value end molecules. So genomics of the plant has a large role to play in making these processes more efficient. The goal is to reduce the cost of these products so that they're fully competitive with similar products produced currently from fossil carbon. And the sort of thing we're doing is relating the transcriptome to the lignin composition and seeing how we can modify lignin composition without in any way altering plant performance. One of the really exciting things that we've been able to do is look at the allele-specific expression. Sugarcane is a 12-ploid plant. There's 12 copies of every gene, and the levels of expression of each of those are in fact important. And we've studied this by looking at the relationship between the level of expression of a gene and the number of copies, because you can have from 1 to 12 copies in the 12 chromosomes of a particular allele. And what we find is that there's a remarkably linear relationship and that most of the genes in sugarcane are expressed in a completely unbiased way. All the alleles contribute. So if there's 5 copies out of 12, 5/12 of the genes will be expressed. We'll have that sequence. And there are a small number of genes depicted along the top of this graph that are expressed in a completely biased way. Despite there being 2 alleles, only one of them is expressed. And these turn out to be the genes involved in pest and disease resistance. So we're getting a better understanding about the allele-specific expression in this complex polyploid system. And sugarcane has a number of subgenomes. And so in the very large number of chromosomes, and we're dealing with more than 100 chromosomes in a typical sugarcane hybrid, we've really now revealed the contribution of the different subgenomes by using long-read transcriptome sequencing, and this is work with Prathama in India, and we've looked at the different contribution of the 2 subgenomes and really now understand the role of the genes from the different progenitors in the important traits of a hybrid sugarcane and how we might change that by changing the composition of the hybrid in novel ways. So this is a really important new insight. So we've identified many candidates in the transcriptome, many thousand, and now we're in the process of systematically evaluating. So we're proposing to edit or mutate every one of these thousands of genes and establish its function. And from those, identify the genetic options we have for improving plants to replace oil. So we're going to be able to edit. We're already able to edit very large numbers in rice. We're transferring that ability to sorghum and sugarcane and other grasses so that we can test the phenotype of all of these mutants, where we have this very large mutant population of sorghum where we can also find a knockout in any gene in the genome and establish what its phenotype is. We've got a large number of investigators that are now joining us in this new initiative. A large number of industry collaborators have also joined this effort, and quite a large number of partner organisations, many more beyond those depicted here, are now joining this effort. So to return to my initial proposition, there are three approaches we can take in agriculture to climate change. The first is to accept that it's happening and just adapt, develop drought-tolerant, heat-tolerant plants. And here we have a centre of excellence that's focused on doing that over the next few years. And really, this is the approach that we've been taking for many years in developing plants that perform better in the environment. The second option is accept that agriculture has a role in reducing emissions and that there are carbon emissions during agricultural production. And it's just been announced here that the federal government has funded almost $300 million initiative to, over the next 10 years, reduce the emissions from agriculture itself. But what I've talked about today is a third option involving agriculture, and that's using agriculture to remove and reduce emissions in other areas. For example, in the production of plastics or in fuels for long-distance flight or for other sorts of biomaterials. And this is another essential role because we don't have any other options than to use renewable plants for this if we're going to avoid the use of fossil carbon. So hopefully that provides a useful perspective on the different strategies that we're using and the critical role of agriculture at many levels in addressing the challenge of climate change. Thank you.
Any questions for the gentleman? No. Thank you so much, Professor Robert Henry, for joining, and thank you for the wonderful talk. Okay. Yeah, I guess so. So now we have a break for 10 minutes. And before that, I'll request all of you to please be on time for the next session. Also, there's a group picture that I'll inform. We'll do it as early as possible. So for the group picture, can we invite everyone? Mukherjee, sir. Please join us on stage, everyone, for the group picture. Okay. Okay, we'll now disperse for the coffee break and we'll click the group picture outside of this hall after the coffee break. Thank you.
Ayo Dio.
Do we have Dr. D.S. Gurjar over here? Sir, can we please have your talk? Is there anyone in the audience who has to submit their talk? Check. Okay, we are moving ahead with Dr. Javed Farsi from BT Petrokima Gresik, Indonesia. I guess he has joined online. Can we connect with him? Javed Farsi. Can we connect him over Zoom call, please?
Yes, hello. Is my audio is clear?
Yes, sir.
Thank you.
Yes, sir. You can start your talk now, sir.
Okay. Okay. Thank you for the session. I would like to share the screen.
Check. Dr. Iqbal, please share
Can
your.
I share a screen?
Yes.
Excuse me. Okay. Okay, good afternoon, ladies and gentlemen. My name is Iqbal Ramadan. I'm from Petrochemical Company, and in this team, there is Mr. Mahendra from Petrochemical too. We would like to share you all about our improvement and our innovation in Indonesia. Actually, for the first compound fertilizer, organic compound fertilizer, compound salam, as an effort of PT Petrochemical increasing into Indonesian organic agriculture. First of all, this is our company profile. We established in July 10, 1972, and in 2012 we are under the Pupuk Indonesia Holding Company. We are the complete fertilizer manufacturer in Indonesia, which produce various kind of fertilizer and chemical for agro-industry solution. At least we have total current production reach 8.9 million tons per year, consists 5 million tons of fertilizer product and 3.9 million tons per year of non-fertilizer product. This is our product. As we said before that we have two basic products, fertilizer and non-fertilizer. For the non-fertilizer, we have ammonium fluoride and also ammonia, carbon dioxide liquid, chloric acid, and etc. product. We also have a product in farm and fisheries such as petrochick and petrobiofeed. For our first introduction in our improvement in Indonesia, actually in 2019, there is 3,350 of organic agricultural landfill, and there is the increasing value on 2020 and 21. At least in 2022, it's 4,766, which in Indonesia, the interest of people to make their land to be organic agriculture is increasing. It because the health and environment issues and also the effect of chemical fertilizer in Indonesia give unbalanced nutrient in soil. So the highest peak in 2022, there is 4,766. And also the increasing of the farmer in Indonesia, the farmer move to the using of unorganic agriculture to organic agriculture, and the amount of organic farmer in Indonesia increased in 2022. It shown that the interest of Indonesian people increasing than 3 years before. We have the value on 12,752 the organic farmer in Indonesia. After that, because of the increasing of the organic agriculture in Indonesia, but in our research in Indonesia, there are no balance about the fertilizer of organic and also the productivity of their land, because of organic fertilizer is need time to take a big quantity, and also the dose of fertilizer need a large quantity too. So in this case, we would like to improve the using of organic fertilizer to another fertilizer that support the organic agriculture in Indonesia. And then we found that every research, the NPK compound fertilizer is a good fertilizer if we combine the compound fertilizer with the compost or organic fertilizer. But in accordance to standard in Indonesia about organic agriculture, there are no allowance of using the non-organic materials. So we need the organic materials, fertilizer to support our system in Indonesia. Based on this case, we create with our technology to make the NPK compound fertilizer that has been certified with organic materials. It will support the organic agriculture in Indonesia. We have found that compound fertilizer and compost organic fertilizer give real impact on growth of the plant and also the good result of the plant and increasing the productivity of the plant. And we call our product as name Ponska Alam, which is a complex macro fertilizer. And then we launched the product in 2021. The data on 2022 show that the increasement after the 2021 that our product has been increased the organic agriculture in Indonesia. And this is our product. As we said before that we use the NPK compound fertilizer, and this is specification of our product. This product has been certified for organic materials, which is the specification such as nitrogen minimum 5%, phosphate minimum 10%, potassium minimum 10%, and the total of the N, P, and K is minimum 25%, and for the water content is maximum 3%. And we noticed that the environment issues in Indonesia, we are laws about the metal contamination for our product to give allowance for the contamination in environment issues such as mercury maximum in 10 ppm or 10 milligram per kilogram, and then cadmium 90 milligram per kilogram, and then plumbum and for 500 milligram per kilogram, and also the arsen in maximum is 100 milligram per kilogram. As we said before that our product has certified for an organic product, so the farmer of organic in Indonesia isn't worried about the regulation in Indonesia because it can use in organic agriculture in Indonesia. For the raw materials in our product, we use the natural mineral such as nitrate sodium, phosphate rock, finish, and clay. For all these materials has been certified as organic materials. So in our factory, there are no chemical or other chemical that is forbidden as our regulation in Indonesia. So all these materials has been certified as an organic materials. It gives the certificate of organic product. For the nitrate sodium is the source of nitrogen, and also the phosphate rock is the source of phosphate. Finish as a side product of ethanol factory as the source of potassium, and also the clay as the filler. This is our process of Ponska Alam. We use a common NPK factory for the design capacity in 10,000 ton per year. We use common NPK factory such like mix in pen granulation. So our product is granule, has good quality in granular. The process such as feeding material and then mixing, and then we put in pen granulation. We add water and no chemical in the reaction just water and all the raw materials to granulate all the materials. And then after we produce the granular, we dry to decrease the water content, and after that we put in the cooler. And also in this process, we screen all the products so we have the same size of the product. And then we packaging. Before the packaging product, we also coating the product by the dolomite to decrease the caking problem. And after that, we packaging the product. For the effect by using Ponska Alam in Indonesia, we have a trial in every village in Indonesia, actually for the paddy farmer and also other commodity plant. We can see that the uses of Ponska Alam, it can increase the result value or the productivity on land in 7%. And also, if we combine the compost fertilizer and also Ponska Alam, the highest peak percent of the increment result value is in 33%. It also give a big impact on other commodities such as onion and also on other vegetable and fruit. The using of NPK compound fertilizer is can use combined by compost fertilizer to give a good impact in productivity. And this is our socialization of Ponska Alam to organic farmer in Indonesia. They give a good impact to us about our product, and also they have feel the all the increasing of their field in every farmer in Indonesia. Okay, maybe that's all that we can share about our product and our improvement about Ponska Alam as an organic fertilizer in organic agriculture in Indonesia. Thank you very much.
Professor Iqbal, thank you for
Yes.
your but I got a little bit confused, that as you told it is organic composition and you are using sodium nitrate directly. So I'm a little bit confused. And another thing that you are telling it is a complete replacement of NPK. What is the recommended dose of NPK for your country for rice?
I beg your pardon, sir. My network is not unstable. I'm so sorry.
Any questions from the audiences?
Okay, thank you so much, sir, for joining and giving a wonderful talk. We now move ahead with Dr. Narottam De, Vishwa Bharati University, Shantiniketan. He has already joined online and we'll now hear his talk. Please connect through Dr. Narottam De. Yeah. Sir, can you please try speaking? He has joined already. Sir has already shared his screen. Sir, can you hear us? Sir, please unmute. Dr. Ray, can you please try speaking? Can you please unmute yourself? He is already on unmute though. Okay, by the time he rejoins, there's two information for all of you. First, to all the participants, you are requested to collect your participation certificate from the registration desk post lunch. That is one important thing that you all need to do. Also, in case if you people are clicking any pictures, selfies, whatever you people are clicking, while you are posting it, please do not forget to tag @SARM Odisha. ODISHA, SARM Odisha. You have been given this leaflet. There's this already written at the back, SARM Odisha. That's very, very, very important. So we are trying to reconnect Dr. Narottam Dey. And the other speakers, if you are here, please give me your pen drives. Dr. Sirigi Reddy, Dr. Surya Kumar Saikia, Dr. Christle Ledroit. All of you are requested to please give me your pen drives. Given already? Okay. Dr. Day, are you online? Yes, okay. So please try reconnecting. By the time we will have our next speaker first. Dr. Sirigi Reddy, Sivajyoti. Dr. Sirigi Reddy is from College of Veterinary Science, Kodatur, India.
Good afternoon. So my presentation is on cerebrospinal fluid analysis, hematobiochemical profiling, and therapeutic studies in buffaloes with babesiosis. So in India, buffalo rearing is an important component of rural economy. So apart from milk production, these buffaloes, they can also use for draught purpose and meat purpose also. So these buffaloes, they're suffering with a number of diseases. So in that, the babesiosis, so it is an important hemoprotozoan disease. That means it is a vector-borne disease. So here the vectors are the ticks. So it is present in the RBC of the buffaloes. So if the disease is not treated, it is causing high mortality and morbidity. So usually the buffalo cows, so they are resistant to this disease because of inverse ACE resistance, and also strong innate immunity due to the maternal antibodies. But because of some lack of transfer of cholesterol and lack of maternal antibodies, sometimes there is a reversal of inverse ACE resistance. So then we are seeing the disease in cows. So little documentation is available in cows, especially with babesiosis, with nervous signs. So in India. So the present study is conducted during a period of 1 year from January 2023 to December 2023. So during this, the objectives of the present study is to record the instance of cerebral form of babesiosis, to record the clinical signs, especially the nervous signs, to record the hematology, and also serum biochemical profiling. So, and again, on one of the objectives to record the ECG and echocardiography of the nervous form of babesiosis. And finally, the confirmation was done based on cerebrospinal fluid analysis. So the infection of, that means the cause infected with babesiosis was confirmed based on the identification of the pyroplasms within the RBC of the buffalo calves. So during this 1 year period of study, so out of 120 buffaloes which are suffering with babesiosis, so from that only 8 calves are showing the nervous form, that is a cerebral form of babesiosis. So the incidence rate is 6.25%. So the animals that are affected with babesiosis, they are showing the clinical signs like absence of rumination, anorexia, wobbling gait, hyperthermia, scleral congestion, tachycardia, hyperexcitability, delirium, esthesia, and grinding of teeth. So in this video is showing the nervous signs, cough which is showing the nervous signs, that is wobbling gait, torticollis, that is turning of the head, so unable to stand. So these are the clinical signs because of the cerebral form of babesiosis. And hematobiochemical examination, the parameters which were examined were hemoglobin, packed cell volume, total erythrocyte count, total leukocyte count, neutrophils, eosinophils, total protein, albumin, globulins, and blood urea nitrogen, creatine, potassium, sodium, chloride, and glucose. So there is in the blood picture, yes, it is showing the anemia because of hemolysis. It is a hemolytic anemia, that is of microcytic hyperchronic anemia, because the parasite present within the RBC, it is causing the lysis, and they are increasing in number. That's why you're seeing the anemia, and there is a leukopenia. So it is mediated by the tumor necrosing factor alpha. Then there is elevated eosinophilia because of it is in a response to the allergy. That means the parasitic response. So that's why you're seeing eosinophilia. And elevated globulins levels, that means this is because of the infection, there is rise in antibody levels. So this is like hypersensitivity reaction. Then elevated burn, because here the parasite complex is deposited at the kidney level. So because of glomerulonephritis, we are seeing the elevated burn. Then ECG was carried out while animal in standing position based on base lead effect system. Here lead 1 is considered as per trace reading. So it is given finally recorded as the mean values. So here in this table, the P wave, QRS complex, T wave. So all values were recorded from apparently healthy animals and also babesiosis infected, especially the cerebral form of babesiosis. So from these, the interpretations were wide P wave. So this is because of atrial default. Then there is a spike T wave and tachycardia, and that means the increased heart rate and ventricular defect. There is a cardiac arrhythmia, then Y T wave. Then echocardiography was also conducted based on the animal is in standing position on right lateral axis. Echocardiography. Here, left ventricular increased systole, diastolic volume was increased. Systole volume was also increased. Then left ventricular systole volume is also increased. Then there is a cardiac output is also increased. Decreased fraction ejection. So this cerebrospinal fluid was collected from lumbosacral junction. So the video is showing the same. You can see here the fluid is coming from lumbosacral junction, that is the cerebrospinal fluid. So here you can see the pictures of cerebrospinal fluid. The turbidity was increased because of the influx of the RBC into the cerebrospinal fluid. So in apparently healthy animal, it is very clear in the top of the picture. Then the components of cerebrospinal fluid were estimated by the specific gravity, pH, total protein, and glucose. Usually the total leukocyte count and total erythrocyte count was 0 in case of apparently healthy animals. But in case of cerebral form of babesiosis, we are seeing these cells. So because of the presence of these cells, you are seeing the turbidity, there is increase in the specific gravity. So in the stain picture also, you can observe these erythrocytes and also WBC, leukocytes. Then the disease is also confirmed by molecular, that means by using the PCR. So the picture is showing the 56 base pairs, so which is confirmed the animal is suffering with babesiosis. So the animal was treated by using the injection diamycin acetate and planixion maximin, dextrose normal saline, and mannitol. So mannitol to counteract the glomerulonephritis, then oral glycerol, and injection B complex, which contains the methylcobalamin, pyridoxine, and nicotinamide, and supportive therapy by using injection containing vitamin A, D3. So after treatment, there is improvement. That means the animal is responding to the treatment from the first day. So immediately after the second day, there is a nervous signs were less. Then on third day of presentation, we can see the walking of the animal clearly. So in this video, we can observe. So there you can see in first day it is showing the nervous forms wobbling gait. Second is somewhat stability, and third day straight walking of the animal. That means the animal is responding to the treatment. So this in this picture, that means after treatment the animal is responding. That means the erection. That means the appearance of the clinical signs. There is increase in the rumination. So the animal is start taking the feed. Wobbling gait was reduced. There is a fever, means hyperthermia is also recovered. Delirium, aesthesia. The animal is start passing of the dung sample. Then even we can also see the hematological changes after treatment. So there is increased hemoglobin, increased packed cell volume, total erythrocyte count, total leukocyte count, and there is a reduction in eosinophil count. So some of the cows, they were died. So after that, we conduct the histopathological examination. So in the brain sample, where it is showing the vascular congestion and blockage of microvasculature in the central nervous system and microglia. That means because of the parasite invasion, the parasite is present within the RBC. So it is causing multiplication and destruction. So and there is a change in the charge of the RBC on the surface. So because of that, the RBC, they are secreted into the cerebral vasculature. So because of that, there is obstruction. So vascular obstruction means sequelae is the vascular edema. So and there is a destruction of the tight tensions in the blood-brain barrier. So when there is a disruption, means there is a gap. Then there is influx of erythrocytes. So cerebral form of babesiosis can be reversible. So if if there is no endothelial vascular damage and there is only stellate erythrocytic membrane protrusion, so this can be reversible by using fluid administration. And there is membrane glycoprotein alterations in erythrocytes because of the presence of babesia within the RBC. So these can be treated by using the parasiticidal drugs and also anti-inflammatory drugs. And here the differential diagnosis is important because the neurological signs may also seen in incoes because of malaria also, as in case of humans. So these babesia infections can be used as a role model for treatment, as in case of cerebral malaria. So the same mechanism is also happening here because of the role of COX. Here we are seeing the deposition of and that means parasite and globulin complexes at the kidney level. So in this picture, you can see there is a damage to the blood-brain barrier. There is influx of RBC. So once there is influx, there is obstruction. Whenever there is obstruction to the, then there you can see the edema and nervous signs, and there is increased intracranial pressure. And finally, I'm concluding that the nervous form of babesiosis, so it's very rare in case, of course, once it can be diagnosed by using the cerebrospinal fluid analysis, and it can be treated successfully by using the fluid therapy. Thank you.
Thank you, madam, for the excellent presentation. Actually, this in our house, one of the calf was having this type of symptoms. So I did not know that that is due to babesiosis. So now, if any questions from the audience is there any question? Or we'll go to the next speaker, Madam Dr. De.
Thank you.
Thank you, ma'am. So now we'll connect with Dr. Narottam De again. Can we please connect him on Zoom call? Can we please connect with the Zoom call, on the Zoom call with Dr. Narottam De? So please talk. Yes, the same issue. Yeah, he is unmuted already. His connection, not us. Yeah, yeah. So that won't be clear. Uh-huh. A lot of connections are already there. If you connect with the mobile, it won't sound good actually. Hmm. Okay, there's some issue with his connection, probably. So we'll move towards the next now. Dr. Surya Kumar Saikia. Yes. From Vishwa Bharati University, Shantiniketan, he is. He's already here. He is talking on sustainability in fisheries, resources, intensification, and species selection.
মাইক্রোফোনটা এরে হতো নাকি?
Check.
Now it is okay?
Okay. So yes, sir, you are audible now. You can share your screen.
Hello?
Yes, sir, you are audible.
This is Narottam. Yes? Now it is okay?
Yes, sir, it's okay. You are audible. Please share your complete screen.
Okay. So, okay. So, a good afternoon to all of you. This is Narottam from Shantiniketan Vishwa Bharati. And this is my... Okay. So, the... Okay. Just a minute. এটা বন্ধ করে দিবো, এটা বন্ধ করো, এটা জাস্ট এই স্টপ করো, এখানে পিন করবো নাকি? এগুলো যাচ্ছে না তো। এটা বন্ধ যাবে না?
Sir,
Okay, yes. Now it is okay. Now it is okay. So good afternoon to all of you. The topic on which I will discuss is that genetic regulatory switch between SAB1A and SK, the key adaptive mechanism behind shifting from quiescence to elongation in submerged rice. Now, regarding the so first of all, I have to tell you that there is the background of the origin of my topic on which I am doing research here. So you all we know that is submergence tolerance is one of the major problem in lowland rice fields of the eastern India, particularly Assam, Odisha, and West Bengal. Now there are two oil mechanism, the two oil described oil developed mechanism, that is we know that is one is quiescence and another is deep water flooding. Now due to changes of the environmental condition, because you know there is a huge changes in the environmental condition, for which though these two mechanism which we known as quiescent as well as the deep water flooding, there are two separate mechanism is working, but theoretically those that is available in theory, but in practical what is happening, due to this geopartition of this particular environment, a particular field have to face initially with this type of first class, then it is under the one type of water stagnation. So a single plant have to face both the things, that is both the first flood as well as this type of deep water flood. Sometimes it is one type of water logging. So it is very much difficult for a plant to become survive if a particular rice line have a single loci, either that is sub1 or that is the snorkel loci. Because you know sub1 in this particular picture it is showing there is two particular loci. One is that is the sub1 loci, another is sk loci. Two are interplaying into different condition. In the left hand panel it is showing that is under the submerged condition what is the how a particular plant face, and whenever the water level is up to 5 to 50 centimeter, then in generally that is the sub-1 loci is interplaying. But whenever the water level goes more than 50, then this particular scale locus is interplaying. But the condition is that now the situation, so just one. So this is the one diagrammatic view of the genome of rice, particularly in chromosome number 9 and 12, two loci are located. Particularly two QTLs are located. One QTL is sub-1 cuticle, yet it has sub-1 A, B, C, 3 different components. And in chromosome 12, there is the snorkel loci, and it is showing that is the both of them are ethylene responsive factor. Whenever a particular plant undergo under the this type of oxygen deprivation, anoxic condition, then they are interplaying through the ethylene responsive factor. So this concept is well developed, and you know from the Odisha, that is there is one of the rice land races which is commonly known as the FR39. The sub-1 have been integrated into a number of other high-yielding background to develop a number of high-yielding rice lines like Swarno sub-1, Sobita sub-1. So up to this, everything is okay. So there is sub-1, a good number of Swarno sub-1 or this type of rice lines is available to the farmers. They are growing in this way for the lowland, particularly on the mid-lowland rice ecosystem. But problem is that whenever a particular field is suffering from a water stagnation, that is initially a particular field is suffering from this type of fast flood, then the water remain around say 70 to 80 centimeter for a more than 1 month. Then plant have to face, that is then during that time sub-1 loci is not working. Then the situation will be that plant have to be die or producing very little amount of yield. So plant is not too much productive. So that is the reason these are the different type of fact what I have mentioned here. And the last paragraph I have mentioned, but the genomic diversity study on a number of lowland rice lines like this a number of rice lines I have mentioned, Gongasali, Kalirai, Sadajavda. Interesting, this type of rice lines contain both the sub-1 loci and SK loci. But most of the literature still now reported, they are mentioning that a particular rice genome may contain either sub-1 or SK. Both of them cannot be present in the same times. If they are present, generally SK remain in non-functional case. But then the problem is that what will be the condition in case of those rice lines which I mentioned here, a very few limited lines which contains both the loci sub-1 and SK. So what will be their particularly expression profile as well as regulation, that is not known. That is the main aim of my talk today. So in these cases, I have collected, that is our group has collected 3 different sets, one from West Bengal, another from Odisha, another from Assam. So today I will discuss only for the Assam groups. So these are the different type of Assamese rice lines, particularly they face to face the fast flood as well as deep water flood. So these are the different type of rice lines: Kalapatia, Gongasali, Samba Masuri, Khada, Kusuma, Purnendu, Ranjit. So all these lines are not typical land races. There are some rice lines which are also improved rice lines. Say Purnendu is one type of such rice lines. Ranjit is one type of such rice lines. But most of them are actually the different type of land races. We have collected around 25 such rice land races, of which 10 or 20 or 10 or 12 shows the good results. So I am showing only the 10 these things, only the 10 rice lines I am showing here. You see, this is the key problem was, what is the most effective genetic combination of sub-1 and SK? Because sub-1 is a QTL. It has a several type of components. Sub-1 ABC, again sub-1 ABC, sub-1 1. Similarly SK1, SK2. So what is the actual, or what is the good combination which is giving the best submergence tolerance, not only for the one type of flood, that is both quiescent as well as elongation strategy, under both quiescent elongation strategy, which will give the maximum amount of this type of submergence tolerance. You see, this is the different type of land races or some improved line also. And here the conventionally two different rise lines, one is used as a negative, another is positive check. That is IR42 is a negative check and FR13 is a positive check. Then all the different type of normal, what is the commonly used as physiological screening, anatomical screening, and the different type of that is the different type of biochemical screening and expression profile, all the things we have performed. So some of the results I am showing here. Our group have recently this. So you see, the basic thing is that for this selection of those rise lines, we have thoroughly studied the availability of sub-1 SK locus and sub-1 locus and SK locus in all those rise lines. The right hand side is the panel showing it is the fingerprint profile with the sub-1 loci upper one, and the middle one is showing the SK locus. You see, out of these rise lines, all are showing the sub-1 locus, and SK locus, for SK locus it is showing the VB69, which is FR39. There is no report that is FR13A having the SK locus, because you know FR13A is famous for quiescent strategy, but it is showing it is also the SK locus. Then the problem arises, so what will be the mechanism of regulation? Because a particular genotype, if contains both sub-1 and SK locus, what will be their regulation? But interestingly, according to the literature, though in FR13A SK locus is present, it is not expressing. In our study also it is showing, it is totally showing one quiescent strategy still, but it has some SK locus. Maybe SK locus is in dormant form or something. That is the main theme of our study. That is what is the regulation of this one, how the gene expression profile regulates this particular two locus according to the different type of depth and different type of duration and depth of the water level. Now it is showing that is in VB604 and 605. These are basically one. VB604 is one type of land races. It is known as Gangasali. We are all the different type of rice, then different type of gene expression profile, mRNA profile, transcriptome profile, all we have used on this Gangasali variety, because Gangasali is so, because first one is Gangasali 604 and another is 605. There is the Kalaputia. Both of them shows SK locus as well as sub-1 locus, but their physiological performance shows there is Gangasali is performing much better than the Kalaputia in terms of combination of both the two type of that is growth, that is quiescent strategy as well as elongation strategy. So in the lower panel, in the left hand side lower panel, it is showing that is the how the sub-1 and SK locus present in this Gangasali along with the that is the FR13 and IR42. In IR42, though, that is the sub-1 locus is present, SK locus is not present, as literature is supporting this view. Then it is the, you see different type of allele, different type of the right hand side, it is showing the different type of Odisha and as well as West Bengal rice lands, for which for those lines we have studied details about the location, details about the different allelic form of sub-1 and SK. That is, we have already published these things, we have not presented here. Now regarding the physiological performance, in physiological performance it is showing there is both the IR42, FR13 and Gangasali, they shows a contrasting different particular physiological performance. And what we have done, we have seen, that is the shifting from the sub-1 and SK. There is how a plant grows quiescence from the quiescence when it shifted, that is from the quiescence to elongation. It has been shown that it is somehow done, it is somehow play within the 8 to 13 days. That is from the 8th day to 13th day. In between this period, this particular shifting happening. So between 8 to, because initially we have studied the 7 days, 14 days, 21 days post-submergence period. So we have studied this one. Then then we have noticed that is this shifting is basically taken in between 8th and 13 days, 14 days actually. So from 8 to 13 days, every day we have studied the physiological performance, the biochemical performance, the gene expression profile. Every day we have studied, and interestingly we have noticed that is, yes, in case of Gangasali, that is which is showing both the elongation as it's both the quiescence as well as elongation, and this shifting is happening in the 12th day. So 12th day is our main priority. If we, because in we are our hypothesis is that there is the shifting from the sub 1 activity to SK activity, it is happening on the 12th day. So we have to study all the different type of test, biochemical, physiological and expression profile during this 12, that is the throughout this day. So in this it is showing Gangasali is showing a contrasting and very good results, and then it is then similarly all the different type of respiratory, because we know that is that there are a good number of enzymes, particularly the which are involved in this type of anaerobic respiration. Alcohol dehydrogenase, sucrose synthase is responsible for sugar locating, then pyruvate decarboxylase, all the different type of enzymes which are commonly reported to be linked with this type of submerged tolerance. For all the this type of enzyme, we have performed the real-time PCR as well as the amount of this particular enzyme activity. All that we have studied, and we got a good results. There is a contrasting results. There is maybe in the 12th as well as 13th, in between the 12th and 13th. In this period, the shifting is happening. Though in all the cases they are not showing the same pattern of result, there are some deviation. But for as a whole, it has been shown that I am not going into details about this one, but it is showing that is the graphically it is showing that is the they basically whenever we compare with the control plan with the test plan, it is showing that particular time period, particular point, they are shifting from one phase to another phase. Then then we this is the profile you see, that is the particularly expression profile of the sub 1 and SK locus, and there is the sub 1 and there is the all the sub 1 and SK locus throughout all the days. It has been studied, and it has been shown that yes, in the physiological, biochemical, which in both the study what it was being displayed, that is the particularly shifting from the quiescence to elongation on the 12 days. Somehow the plant is shifting from quiescence to elongation periods. Then this is then after that one, because you know regarding the switching as well as regulation of gene, there is another candidate loci, candidate genetic elements, that is microRNA, because microRNA are also responsible for different this type of, that is the fine tuning of different loci, because microRNA can target a particular loci, then accordingly they have control their expression. Sometime it is over, sometime it is just block a particular loci so that it will not be expressed. So, and till now there is no such miRNA available for this particular sub 1 and SK. So we have initially we started one bioinformatics analysis to find out what are the different type of microRNA loci available in the RISE database which are targeting the sub 1 and SK locus. Then those microRNA, we have studied those microRNA. So here it is the what I told you, that is the same thing it is mentioned here. Then it is the actually the pipeline. That is initially we have to isolate the particularly those miRNA. Then those miRNA we have to find out whether those miRNA binding in which location. Then out of those location which is thermodynamically constant, the thermodynamically favorable, that means which is the best candidate microRNA for that particular sub 1 and SK locus. Here I am showing only the SK locus because this data has been already published, but sub 1 loci is not yet published, so I am not presenting here. So we have identified there is the Osamir 319b and Osamir 439. These are the two candidate miRNA which are supposed to be targeting the SK locus. Then in the next page, the same thing we have studied, that is their sequence diversity, how much they are conserved, and their interaction with the particularly the SK, that is the SK locus. And here are the molecular network, that is what are the different type of miRNA supposed to be interact with the particularly SK locus, because you know SK locus has two form, one is SK1 and SK2. All the different type of microRNA, their network analysis, this one, and the lower one is that is the bioinformatics analysis. It is showing, because you know whenever a particular microRNA targeting a particular target, then where the binding is most bioinformatically suitable or more economic. So that will be the best mRNA, and which is targeting the particular loci, target loci. Then finally we have to validate our concept, our hypothesis. Then what we have seen, whenever we study at the stress material, that is the SK locus, its function, its expression, along with the mRNA. Here mRNA 1319B, this is one mRNA we have studied. Another mRNA did not show very good result during the validation study. So here MIR 131B, it is showing the good result. So it is showing that is whenever the mRNA expression is increasing, mRNA level is decreasing. So that is what we want to what we want to study, what we want to prove. That is already proved for the one mRNA. From here we can say that this is the mRNA which may be the responsible for this sub-1 loci fine regulating. In the second job, what we will do, that is what are the total mRNA which are responsible for both sub-1 and SK at a time, and what are the different type of that is the same thing we will do, that is the from 8 to 13 days on particular which day, that is the one this all the microRNA is regulating these things, as a result of which this type of regulation, this type of switching from sub-1 to SK1 SK locus will be done. So, and here are some of the relevant publication we have done for the last 5 or 6 years. SK, that is the for SK locus, this this one study of physiological responses with aerial diversity of sub-1 SK, where it is showing the total diversity, and the lower one in the right hand sides. This paper is yet we have that is communicated, is under review. That is the how the sub-1 and SK is interplaying. There is the shifting. Okay, and in the left hand side is in the below, you see mRNA mediated regulation of SK locus in rice under submersion. So these are the different type of publication we have done in the last 5 or 6 years. So I am finished. Thank you for providing me this platform to present my talk in the online. Thank you all. I will be happy if I able to accept some questions.
Thank you, Dr. De, for the nice presentation about the differential role of SK gene and sub-1 gene. So I ask if any question is there from the audiences. So if there is no question, then we can go for the next presentation. Dr. Soikia. Just one question is there. Dr. De,
Yes.
you are there?
Yes. Yeah. Yes, I'm here.
Dr. De, this is a very good presentation, and actually that I have one doubt, that whether you are telling that sub-1 and SK, one
Yes.
is, that SK is shifted from sub-1 to SK shifting.
Yes.
It is done maybe from 8 to 12 days.
Yes.
But whether any that proof is that both are acting together?
Yeah, no, actually, they are actually up to this time there is no literature where they are studying both the sub-1 and SK. Only one Japanese group has done this one. They have not studied the combinatorial effect. They have recorded that is a single variety contains both the loci and both are performing. But we are doing, that is what you are asking. That is, we are trying to make their connection through the mRNA based pathway. Otherwise, no, and that is actually shifting is not from the sub-1 or SK. At one time, sub-1 stop, SK start. That is, there is a switching. Two loci are working in separate way. But at one time, rice play decide that is sub-1 is not good for me. Now I have to shift to SK. This type of things happening, what we hypothesized. Then we try to prove this one, but how this shifting is happening, because basically both of them are ethylene responsive factor. But because there is no such difference between the they are activating. Only the thing is that is the depth and duration of the submergence. So how depth and duration of the submergence, the water level, trigger this shifting, that we have to establish. That is our main thing. So that is not yet there is no record regarding this one.
Okay, thank you.
Thank you, sir. Okay.
Dr. Soikia, please, 15 minutes, sir.
good afternoon to all of you. And thank you to the SRM and NRRI for inviting me for this talk. I think it's come okay. So my topic is sustainability in fisheries, resource intensification and species selection. And I will start from the one of the conference that was held in China, that is in sustainability in aquaculture. There it is said that fish is actually a lifestyle food. And I have got a chance to access the presentations. And also they have very pinpointedly highlighted that in 2020, the captured fisheries inland, it was 11.9 metric ton. And most of them, the fin fish, can see 49,120 million tons. And also after 1 year in Nature, one review was published, that is 2021, that it is increased manifold. The captured fishery from inland fin fish increased manifold. So it means that there is an increasing pressure on the inland fisheries, and then population is increasing, land area is decreasing. So obviously the pressure is there on the fisheries. And then it comes that intensification of aquatic resources is a new approach. And if you see the sequence that how to intensify, the word is very lucrative word, intensification, but how to intensify. And you know that Zé Aubin, that is from France, INRA, I have got a chance to interact with him. He's working for a long time to develop a model for intensification of fish farming. And there are a few publications came up, but still the point is not highly focused. And also I think in India, this is the first conference on sustainable aquaculture from SARM and then Rice Research Institute. And good, a lot of time. Indonesia, I have been there. Also, I think 2-3 conferences already held there on this intensification. And this is the first time in India I have seen this. If I miss someone, I don't know, but this is the first time I've seen that Cuttack is organizing this seminar. So I got the opportunity to present some of the concepts. And these are mainly from my 3 PhD researches and some of my work. And the first work I want to highlight here that intensification, how it is to be done. We have a good model of intensification that is in Arunachal Pradesh, which is called Asani in their local language, and it is called rice fish culture. And this is a traditional fish culture, and they are in a highland. It is called around 1800 meters above mean sea level, and there they used to stock common carp without any supplementary feed. And there is no record of what is the stock they have to give, what is the how to manage, what could be the maximum capacity, optimum capacity. But people they normally practice keeping fish in the rice fields, common carp, and after 6 months they get very good result, 400 gram per season, per 6 months, or 500 gram per season per fish. So we did some research in there, and we found that they are actually using some of the submerged resources that is called periphyton, which is colonized on the submerged rice stem. And the common carp being a grazer, it grazes on the substrate. So otherwise, if you just put in a water column without any substrate, it explores the water column. Once the substrate is there, this common carp, it started grazing on the substrate. So it shifts from the planktonic feeder to the periphytic feeder. So it is a kind of a resource intensification. The resource there we are providing by just adding the substrate. So that is one of the example that we have studied. And this is not only in Arunachal Pradesh. Southeast Asia, you can see the names here. Malaysia, Indonesia, China, Cambodia, Laos, Thailand, Myanmar, Bangladesh, India, Nepal also. Rice fish culture is very popular. And in this whole area, the rice fish culture is a kind of but without any extensive study, people are doing it traditionally. Although some of the concerns now growing up, but still they are doing extensively. Now, one question is that what kind of rice we need to grow there so that our products, the resources can be explored. And some resources are there. Although I would like to inform, there are very few research in this area. that what is the compatibility of fish and rice to be grown in one place. And there are few rice, that in terms of rice, I have not very extensive knowledge. This is Aman, Radha, like some rice varieties are there in different parts of this area. And you can see the figures that stocking density very highly variable. And again, you can see the fish production highly variable. There is no a kind of a standard figure that if you put 3,000 fishes in one rice field per hectare, you will get this much of product. All are not predictable. So in this case, what is lacking, it is still lacking in our rice fish research, is that when we grow two crops, rice and fish, what would be the threshold level to keep both the crops in one place? Because once we keep only rice, it may have different productivity, but once we keep only fish, it may have different productivity. But when you keep both together, the rice productivity may reduce, fish productivity may increase. So what could be the threshold level to what extent we can keep these two crops in one place so that we get the maximum product? So this sustainable part is not even addressed till now. As you know that in rice field we can get zooplankton, periphyton, and phytoplankton. Three resources are very clear. So how these three resources can be explored at the same time? And one of my research scholar, Sandeep Mazumdar, he did PhD because the concept we got the rice fish culture, that periphyton based aquaculture. And at the same time in 2002, Wazini University, they have come up with some concept, I know P. Kesavnath from Mangalore. So they have done Wahab from Bangladesh. They've done some work on periphyton based pond culture. So but they did some other carps, and we wanted to know how the Lebirohita, they're stocked by P. Kesavnath and team, they are getting the periphyton, because Lebirohita is not a grazer, it's a filter feeder. So that my student, he worked on this system, and he found that, that when you go monoculture, only Lebirohita is stocked, and we stock with other Sapinus carpio and Calvasu, still Lebirohita only gets the weight. It is 1.9 times than the periphyton free system, the weight gain. So how it is there? So then we tried with different ecological parameters, indices were there, videographs we have done. We found that the fish actually, whenever the plankton, bamboo substrate is implanted in the pond, it comes from the free area where there is no bamboo substrate near to the bamboo substrate. It never grazes on a bamboo substrate. It nibbles. It starts nibbling on the bamboo substrate. What does it mean? It is not a grazer, but the vicinity of the bamboo substrate, the plankton density grows. So it tries to explore the plankton density. So what we have given, we have given a name as subperiphytic zone. Means the Lebirohita can also be used as a part of the intensification once bamboo substrate is implanted in a system. So this is what my student has got. And the same system is prevalent in many parts of in the northern India, maybe Odisha, I don't know, but they used to implant the bamboo branches in the pond where they get enhanced production of fish. It is traditional, but the main thing is that the subperiphetic zone is explored by the Lebirohita. And this concept, if we add, means we can make the planktonic area free for other fishes. If we can shift the Lebirohita to bamboo substrate area, the other part is free. Means we can add one planktonic feeder in that area. So we are intensifying that one trophic level. And this is whatever you do with the carps. Here I want to highlight another point. You can see that this area, this part is of the figure we can use for this intensification. But whatever we do, what is a product? We get product, protein is a product. Lebirohita is a protein, Calvus is a protein, miracle is a protein. But can we intensify? Can we intensify at the same time the product? Product means not the fish, product means the nutrient. It is a micronutrients. Can you go for this one? Because if you see that micronutrient, yeah, this is what, sorry. So this you can see that we have added, and there one more fish we can add there as a component. Now this area is totally free. Now this is this fish is now merged to the subperiphetic zone. Now this area is free. The surface area is free. So where we can we add some of the small fishes there, which are planktonic. So small fishes, you can see these are highly demanding fishes. And very unfortunately, these fishes have no record in the global statistics. Even national statistics, you don't have record of it. We always talk about the carps, big fishes, but small fishes never know. We never put any statistics in our national data or in a state data. And even in the red data book also, you can not evaluated, not insufficient. But these fishes of late growing into attention because they are highly rich in micronutrients. And adding these fishes, we can intensify the output as a new protein plus other micronutrients. One of my research scholar, Sudarshan Anandi, she did on amplified mola, and she found she worked on two sizes of mola. She found that small size of mola, they usually the mola have a higher amount of calcium, iron, zinc compared to the bigger size of mola. Means the relationship we cannot establish that size increases then micronutrient reduces. We don't know, but small fishes, they have higher micronutrient than the large fishes. So even another study say that EP and DHA, which are very important for milking mother as well as the small kids for their growth, these are also very high in the small fishes. So then if you see the Southeast Asia, this whole area, if on record, the malnutrition rate is very high. But this is highly rich in rice fish culture, where you can put all these fishes if we want to maximize the threshold level. So here, rice fish culture, because once we add the periphyton grazer, column is free. The column, we cannot keep large fishes there. We cannot add Labirohita there. So we can only add small fishes. Can we add small fishes there? So these efforts are not available. And even in many places, what they are designing that we can keep some areas living for the carps, Labirohita as well as common carp. So these are very complex concepts because we don't know what would be the productivity effect of the crop, what would be the productivity on the individual fishes, the products. So in these cases, we need to have at least to know what threshold level we have to reach for a particular product. And now another part is that even though we experiment, we propose the products, we present all these things in a graph, but if the farmers are not accepting it, that is not sustainable. So we have to go with an approach the farmers should at least, sorry, what's coming? Oh, farmers should accept this social fishery. Social fishery is these small fishes, what threat is going on, we don't know. Once we pick up them from small ditches and canals, put it in the local ponds, and yes, finishing one slide, okay. And finish, we are there, we're putting them in the system and a small aquatic cultivable system. They are growing, and at the same time, we are getting the benefits from the small fishes, micronutrient intensification getting. So this is an approach can be, social fishery can be done. And these are the questions we need to know actually, what is the appropriate, because system to system, location to location, this and social fishery, the people's choice may also vary. So we need to know what appropriate species for a particular selection. And growth modeling and impact on ecological system, we need to know. Then again, stress tolerance, mitochondrial behavior, because growth, we know mitochondrial behavior, how it is there, and growth performance of fish species individually. Then finally, that the threshold for sustainable resource intensification, which is very, very important until unless we cannot go for further implementation. Thank you very much.
Thank you, Dr. Soikia. If any question is there, any question for him, then we call the next speaker, madam.
Thank you.
Dr. Christelle Ledroit. Clean screen, please.
Hello, everyone. So today my presentation is not on rice, but it's on cotton, but it's on sustainability, actually. So I'm going to present my PhD study, which is on the socio-ecological sustainability of cotton farming system in central India. So first, I'm going to give you a small background why we choose this topic. Here. Okay. So India is the second biggest producer of cotton in the world, with an estimated production of 1/4 of the world production. However, when we look at the production per hectare, it's relatively low in comparison to other countries. And plant protection actually has been the weakest link in the production process. And the main devastated pest has been obviously the American bollworm, which is called the Helicoverpa armigera. And to fight this attack in India, the BT cotton technology was introduced in 2022. So GMO technology has been protecting plants against the American bollworm, but it's not been protecting the plant from other pests such as the sucking pest. And also in recent years, another damaging pest has also surged, which is the pink bollworm, which is called the Pectinophora gossypelea. So due to all these pest attacks, conventional cotton use around 44% of the total pesticide and insecticide consumed in India. And so focusing on improving Indian cotton productivity while reducing the pesticide pollution will bring India also closer to realizing the sustainable development goals. And to fulfill the SDGs, there is a need to evaluate the sustainability of current agricultural system. And this is why I've done my study on the ecological implication and sustainability of different farming system in cotton crop. So I've done my study in with Coventry University in UK in the Center for Agricultural Water and Resilience, and in collaboration with FIBEL, which is a research institute for organic agriculture. And the study has been done, actually it's a case study, which has been done in Nimar region in Madhya Pradesh in India. And it has been done also in collaboration with Bayore Association. On one part of the study was done on the long-term farming system comparison program in the tropic. So just a small slide about Bayore Association and FIBEL. So Bayore Association is an organization which is working in livelihood, in education, in research, in seed development, and they are working with around 2,500 farmers to give them technical support and access to market as well. And where I have been doing the first part of my study was done on the long-term trial, which it's called SISCOM. So FIBEL has 3 projects, one in Bolivia, in Kenya, and in India, which was started in 2007. So the main aim of my study was to assess the socio-ecological sustainability of cotton farming system, looking at 4 different farming systems on the long term, but also looking at the 2 farm-scale farming systems and focusing on the functional biodiversity with a focus on crop protection as well as soil health. And the last part of the study was to try to integrate the measurement of functional biodiversity into the whole sustainability assessment to compare the two major farming systems currently practiced in India. So indicator-based study have used actually invertebrate to evaluate the impact of agricultural practices for decades now. And they have been frequently tried to compare different type of farming systems, such as BT conventional and non-BT conventional, as well as organic system and the conventional system. They have also been used to evaluate soil quality and to assess the sustainability of agroecosystem above soil. Yes, so this is just a summary of my methodology. So I have used a framework, which is a guideline which has been done by the FAM. No, you cannot see, I think, but it's okay. It's just to tell you that basically first, here it's written, oops, sorry. Here I first done the ecological sustainability on the long-term trial. And then I have gone to the farmer's field. So we have used 6 pair of farmer, organic and BT conventional. And I have developed indicator using the FAO guidance framework. And with this indicator, they have been converted into interview with farmers, and also I have integrated ecological indicator. This is just a quick summary. So this is a framework which has the guidance which has been used. And as you know, for sustainability has 3 pillars of sustainability. There is the environmental factor, the economic factor, and the social factor. So I have focused on the pest and pesticide management, soil management, and the biodiversity and land use. And for this indicator, I have done ecological surveys. And then for the economic and the social aspect, I have done interview with farmers. So for the methodology, I am first going to speak about the ecological methodology, which is ecological sustainability. So I have done the survey on the above ground biodiversity. So in 2 parts. The first part was a canopy dwelling arthropod, which is on top of the plant. And then the other part, which is on the ground dwelling arthropod, which is all the arthropods that you find at the ground level. And for the below ground, looking at the below ground biodiversity, I have looked at 2 indicators. One is the earthworm and one is a fungi. So these are the 2 site survey. In 2016 and 2017, I first done the ecological survey on the plot scale survey, which was done on the feeble long-term trial, as I say, which was started in 2007. And you can see it is a random designs with 4 blocks, so 4 replicate, and on each stripe, here you can see stripe 1 and stripe 2, there is a rotation crop where there is cotton, wheat, chickpea, soy, and wheat, which is being planted alternative here. So in 2016, I had done the second stripe because I have focused on cotton only, and in 2017, I have focused on the first stripe. And then in 2018, we went to the farmer's field where we were comparing 6 pairs of organic and BT conventional farmer. And all our farmers were small-scale farmers, smallholder farmers. That means we selected farmers which were having 1 acre, which were growing 1 acre of cotton. And there were also other factors such as type of soil. So we make sure that to be able to compare these two, this is on vertisoil. So all our farmers also were growing their cotton on the vertisoil, and all the seed were planted around maximum 10 days difference. So this is when we have done the survey. So this is in 2016. So the first year, it was more of a pilot project, I will say, because we first did in May, in July, in October, and in December. And the second year, we focus in the where the rain was happening. This is when we are generally finding the most number of biodiversity. So it was done in July, August, September, and October. This is our long-term trial. As you can see, there's plots, and the plots I forgot to mention are 16 by 16 meter, and the net plot is 12 by 12. So our survey was done in the net plot. And this is for the farmer's field. Here we can see one farmer's field. It's quite clear. And the survey was done in 2018, and it was done in August, September, and October. Now for the social economical sustainability we have done the interview with these 6 pairs of BT conventional farmer and organic farm. So for this, I have first all the indicators that I had selected from the framework has been converted into interview. So we went and interviewed each and every farmer. So that was a total of 12 farmers. And also I had integrated in the environmental pillar, I also integrated some indicator which was not present in the framework, such as the soil health and the ecosystem services. And in the social aspect, I've also added one more, which was the business resilience. So now I'm going to speak about the result. So I'm not going to speak about all the results, but I have just selected the result which are the most interesting. And here you can see that in 2016, on the canopy during arthropod community, we could see, so in yellow, these are the sucking pests, it's Jacid, Cicadelidae, and we can see that here we are looking at the percentage, not the number, and we can see that the percentage in the BT conventional cotton in comparison to all other biodiversity was higher in the BT conventional crop. And this can be due to the fact, first of all, there's a weather pattern in 2016 because this was not replicated in 2017. In 2017, the the percentage of Jacid was similar in all crop. But the hypothesis is that in 2016, because of the weather pattern and BT, this is the only GMO seed which was used. The plant was busher and there were much more biomass which attract much the sucking pest. And an interesting point, these arachnidae are the spiders. Spiders are known for predators, general predators, so they are beneficial arthropod. And we can say that basically we can cluster the organic system, which is organic and biodynamic, and the conventional and BT conventional. And we found that there were significant difference between both organic system and both conventional system. And if you look at the number, we can see that in average, the number of spider was higher in the organic system, almost double than the BT conventional system. So this was an interesting result. Then in 2017, here we are looking at data from the ground drilling arthropod community. There were not much difference actually, apart that maybe here in grid we have the Optoptera, which include grasshopper and cricket, and it was a little bit higher in the conventional and BT conventional, as you can see here between the organic and the BT conventional, it was significant. Here I have looked actually at the functional biodiversity. So here I have put the data again for the 2017 ground drilling arthropod. And when we look specifically at the functional biodiversity, we can see that the percentage of pests was lower in the biodynamic and organic in comparison to the BT conventional and conventional. So for BT, it was almost 50%. Conventional was more than 50%. When in the biodynamic and organic, the data shows that it was less than 40% of the total biodiversity was pest. And here on the side, these are all pictures that I have taken myself. And these are on the left, you have the beneficial arthropod from the canopy drilling community. So we all recognize the ladybug, the syrphid, the long-legged fly, which is a parasitoid, and the spiders. And on the other side, you can see the jacid, the redbug, and the gray weevil. Okay, I'm going to go a little bit faster. Here, there's the data from the below ground ecosystem now. So here it was very clear that when we look at the earthworm, the density and the biomass of the earthworm in 2017 and 2018 was much significantly higher actually in both organic systems. Unfortunately, we could not look at the species because 80% of the earthworms that we had collected were juvenile, and we cannot identify a juvenile earthworm. It's not possible. Another point that we also looked, it's at the fungi community, and yes, here there's a mistake. It's not the farm scale here. It's still the splot scale in 2017. So here we could see that actually the biodiversity of fungi was much higher in both organic system, and we can also find some symbiotroph, endophyte and epiphyte in biodynamic and organic, which was not present in both conventional system. And previously also we had a talk on the trichoderma. So here we use trichoderma as an indicator of soil health. And we could clearly see that the number of trichoderma species was higher one more time in the organic and biodynamic. And here we have isolated one trichoderma species. So I think this one was a Trichoderma viride, if I'm correct. Now we are moving to the farm scale, and here again the same pattern of data is showing that the organic system has a higher number of spiders, which spiders are also used as an indicator to indicate the health of the ecosystem. What was very interesting is that here we can see that in the field, even in comparison to our plot scale, is that almost 50% of the biodiversity found in the organic on the ground floor was a spider, which is a very good sign.
Madam, 2 minutes more. Huh?
Okay. So earthworm same. And now all this data, I converted all this data to be able to integrate them, add them to our sustainable framework. And we could see, so 6 farmers, the B is for the BT conventional farmer and the O is for the organics farmer. And when we just look at the average of the ecological indicator, it was slightly higher in the organic, with the organic farmer, so organic farm. And this is, again, this is a summary of after a combination of the interview, research, plus ecological survey. So when we just look at the overall sustainability, it was higher in, again, in the organic system, significantly higher. And the same data that we saw in the graph previously, I have also put it on the spider radar so we can visualize the data properly. I think also what it's very important to notice here to mention is that all these organic farmers, they were all assisted, I mean, supported by an organization, which is Biore Association, which are giving them training on organic farming and which are giving them also access to market. So to conclude, sorry, I have 1 minute left or it's finished. Can I just finish? So to conclude, on the ecological survey, the ladybird and the spider are good. We know that they are good indicator to evaluate ecosystem services for pest control. And for the below ground, it shows that the earthworm and the trichoderma fungi are also a good indicator for soil health. So our recommendation is to encourage conventional farmers to spray pesticide when the population is above the economical threshold, and to reduce the amount of pesticide applied. And we also recommend to the organic farmers that they should practice integrated pest management. The challenge is to actually encourage cotton farmers to use ecological solution to increase the productivity and conserve the functional biodiversity. And finally, something which was not mentioned in this guidance, it's the importance of the ecological knowledge for the farmers. And it's very important to educate the farmers for them to understand that the health of the ecosystem is very important. And for that, it's important to have farmers organization which can support them. Thank you for listening and giving me this opportunity.
Thank you, madam, for excellent presentation. And if any question is there. So there is no question. May I ask you one question?
Yes, please.
In a crop like cotton,
Yes.
how much it is relevant to go for organic?
Sorry?
In a crop like cotton, how much it is relevant to go for organic? Because you know, organic is little bit costlier also and it consumes lots of time.
So here, if you have the support of organizations such as Biore, they are giving this technical support. They are also helping them with organic input, and then they are giving them access to the market with a 30% bonus. So then it's relevant to go with organic farming. But a single individual farmer to go for organic cotton, it's very challenging. Definitely. Thank you.
Thank you so much, ma'am. So we now disperse for lunch. We will resume back the session at 1:45. The speakers who are giving their talk post lunch are requested to give their presentation, submit their presentation over here. Also, please collect your certificate post lunch from the registration desk.
Good afternoon, one and all. I am Sonali Ghike. I am working as agriculture expert with Vadhwani Institute for Artificial Intelligence, and we are based at New Delhi. So I will be presenting here our few of our solutions and what work we did since last 4-5 years. So I begin with, we are working on few solutions. We are working on Android-based solutions for farmers. So I will introduce my institute first, and the problem we have taken up, many of the speakers have spoken already about the pest issues and the losses caused by them in India. So cotton is one of the commercial crops, as you all know, and it has been infested by more than 250 pests. But out of them, a few are of major concern, and they are causing a serious damage to cotton crop. But still, the farmers would like to go ahead for cotton because it is a promising crop in India. And as we all know that pests are causing havoc in cotton, specifically the bollworms. They are causing major issues and major losses in cotton in India. So what we have done, we have developed an Android-based solution technology which helps farmer to identify the pest and take timely measures and to reduce the pesticide usage. So need-based pesticide uses we are recommending. So how we are going to deal with that I will explain in upcoming slides. So we have developed an AI-based pest detection model which works on Android mobile, and that is handy tool to a farmer who are not able to identify the pest, because many of the farmers, they are still unaware about the cotton bollworms. They are confused with the identification part. So we have developed an AI algorithm which helps the farmer to identify the pest and to help them to take proper measures at proper time so that they can reduce their input cost. Also, I would like to introduce my institute. So we have been established in 2018. We have been inaugurated by our Prime Minister Modi ji. And the two people you can see at extreme right and left, they are our co-founders, Vadhwani brothers. So we are a non-profit independent organization. We are building AI-based solutions in agriculture, health, and education. So we are building these solutions free of cost for the users, end users like farmers or the beneficiaries. And we are not asking or taking a single penny for the users. So our base is like we are working for social impact. So we have our own donors. So they donate money for developing low-cost technology-based solutions for end users like farmers. As I told, we are also working in health and education. So we are using the money for building AI-based solutions and to for the underprivileged people in India. So what we do exactly? So we are developing AI Center of Excellences with governments and civil bodies, and we do AI-based trainings, AI awareness campaigns with them. And also we are currently developing 30 plus solutions in health, agriculture, and education. So our approach is multidisciplinary, collaborative, and impact-based. These are our partners. So we are working with Ministry of Agriculture, Ministry of Health, Ministry of Education, as well as Niti Aayog, and many of the private partners. We are working in PPP model, if I explain it in public-private partnership model. And these are our two leaders who are guiding us throughout all the solutions. They are having a very good expertise in AI and machine learning. Shekhar is our Chief Executive Officer, and Alpan is our Chief ML Scientist. So these are some of the solutions which we have developed so far, and there are particular solutions which are directly related to farmers, like Cotton Ace, the National Pest Surveillance System. Agri-AI Collect is a digital diary for farmers, which farmers can use. It is an audio and text-based solution and image-based also. The farmer has to just record whatever he has done. So automatically it will save what the farmer has done. So this solution will be soon launched. And this is Krishi 24/7 and Kisan Call Center knowledge management system. These two solutions are particularly built for Ministry of Agriculture, and they have been launched recently during September. And ministry is using it by all the departments of ministry are mostly using this solution for purposes like we are providing a dashboard to them. I will explain these solutions later on. And few solutions are still in pipeline. So let us go to the first solution, which I mentioned. It is related to cotton. And this is an early pest warning solution for cotton, which helps the farmer to identify the pest and take timely control measures. So it is AI-based early pest warning system, which works for cotton bollworms. We started this solution in 2018, and slowly we are incorporating and improving this solution in terms of accuracy. Because in AI, it is not like that other softwares, like once you develop the software and put it on the ground and that's done. It is not like that. It needs continuous improvement and analysis to improve on the solutions. So we received 2 awards for this as well, Google Impact Challenge Award and also H&M Foundation's Global Change Award for this. Recently, we have received $3.3 billion grant from Google to expand this solution to other crops. So as we all know, India is the largest cotton producer, but still we know that India grows 25% of world's cotton. 36% of global area under cotton is in India under cultivation. And India has around 6 million cotton farmers. But still, if we see, the average production is very less as compared to the world production. And the losses estimated crop losses are 30% due to pest attacks, and 70% estimated proportion of pest damage is caused by these bollworms. So that is why we took up these bollworms first. And another thing is that what I would like to mention here is that 50% of pesticides are consumed by cotton only. So this solution works at 3 levels: the farmer, the extension worker, and the program partner or the administrator, we can say. So the farmer has to install the traps. This is a trap-based solution, and farmer has to install the pheromone traps in the field. Say if it is BT cotton, then 2 traps for pink bollworm. But if it is organic cotton, then 3 traps, 2 for pink and 1 for American bollworm. And another level is, and he has to download the app as well. Second level is of extension worker. To the extension worker, we will map all the farmers in the particular village. So we all know that pink bollworm is a major issue in India, and we cannot control the pink bollworm by concentrating or focusing on a single farm. It should be done at a village level or a cluster level. So that is why we are engaging the farmers, all the farmers in the village level. The next is administrative level. And the administrative level, we are providing a separate dashboard to each of the partners so that they can manage their own data, they can formulate their own advisories, and they can monitor where the attack is more, and they can allocate the resources accordingly. So this is this slide denotes the 3 layers, like at program. Other than this, we are also providing some additional features to the farmers. Like we are providing the mandi rates to the farmers. We are providing daily weather and also the advisories related to whether they should go ahead for spray or not. So for this purpose, our app has been used extensively this Kharif season. So this is the dashboard, the third larger picture, which you can see here. So on dashboard, the administrators can look at what is exactly going on the field, how many alerts have been generated, and how many farmers are using the app, how many extension workers and farmers are monitoring the field. So this is how the app works. So you can see a farmer emptying the trap on a plain white paper. He will take the picture, he will upload it on the application and instantly he will get the advisory. That is based on ETL level of the pest. Each pest has its own ETL level. So based on ETL, he will immediately receive the advisory. So this is our deployment status. We deployed with 1,16,000 farmers this year. Also, we had generated around 15,000 alerts. So 3 types of alerts are generated. If it is above ETL, then red alert is generated. If it is below ETL, then yellow alert is generated. And if it is safe, if the pest has not crossed any ETL, then it is green alert. So we have covered around 4,573 villages across 11 states, which are major cotton growing states in India. And also we had 31 partners who took our solution to the ground through their extension workers, which also included 3 state governments, Karnataka, Punjab, and Telangana state government, and Madhya Pradesh government. So the impact observed is that we do third-party evaluations. So E&Y and University of Agricultural Sciences, the Harvard, they had done the impact assessment, and they found that the farmers have reduced their expenditure on pesticide, and they have got increase in yield, price, and net income. So as I told earlier, it works at village level. So the farmers are attached to the extension worker. So we have a structure as lead and cascade farmer. So the farmer who has the application and trap installed, he will be called as a lead farmer. And the farmer who does not have an Android mobile, so he will be also tagged with that lead farmer so that he will receive the information whether the pest is introduced into that village or not. And we will just give that other farmer who does not have Android mobile, we will just ask him to monitor his farm and to get connected with his extension worker so that he can get proper guidance at proper time. So we are not advising him to spray directly. And this application that farmer provides early pest warning, these are some of the features. So that we have already seen. So this is the real-time dashboard, how it works, that I will show you. You can see here on the left side that there are some drop-downs, partners, supervisor, advisory management, cotton app users. So our partners have access to this dashboard and they can manage their own farmers. They can see the continuous status, how the monitoring is going on, how farmers are involved in this activity, how many farmers are getting red alerts, how many farmers are having above ETL population. So like this, we have provided access to the farmers. These are some solution implementation requirements that our partners should have on-ground presence, because we are a small team all over India, and we are just 4 or 5 people who are looking after deployment in multiple states. So we require partners who are having on-ground presence of their presence. Then they should have good quality data, like basic information of farmer we are taking with his location, his mobile number, because it requires Android mobile should be there with the lead farmer at least, who is leading from that village. Oh, sorry. So these are some key features. So once at first you need to install the app, and once it is installed, it works offline as well. And if we talk about the accuracy of this solution is around 90%. And like it will give 90% accurate results of the images that they have been uploaded on the application. And this application is available currently in 9 languages in India. And also it gives real-time advisories. Like it will not give generic advisories. That is what government platforms are doing right now. So it will give advisory for the particular farmer who has monitored the farm. So these are the findings of the third-party evaluation. So they have found that estimated reduction in expenditure for pesticides is around almost 25%, those farmers who are using this application. And estimated increase is there in 20% yield because they have taken timely decisions. So this is our project outreach. We started with some pilots during 2019, 2020, 2021. And this year we outreached around 1 lakh farmers, 1,16,000 farmers. So these are our learnings so far. Regarding cotton-S application, I have already told that users have higher family income, higher number of pickings, higher yield of cotton was observed. But like AI interventions in agriculture have major challenges to scale due to lack of data, lack of technology backbone, and other program challenges. And weather also plays its crucial part everywhere in agriculture in India specifically. So this is our another solution which I talked about. Like we are expanding based on our CottonX experience. Like what we do, we develop some solutions, we run them for some time, then we open source the solution. So CottonX is already available on Play Store. It is totally free of cost. Anybody can download it and anybody can use it. So based on that, now CottonX has been adopted by Ministry of Agriculture and Farmer Welfare under this National Pest Surveillance System, Disease and Pest Surveillance System. So this is also AI-powered pest and disease surveillance system. What new thing we have approached here is image-based analysis. Based on symptom, is the farmer can take the picture of the symptom or the disease symptom or the pest damage, and immediately he will get the result, what is the infestation is of, and he will get the advisory based on the identification. So this solution is being run currently by Ministry of Agriculture and Farmer Welfare. So this is very simple and easy to understand. Advisory in farmers' preferred language. These are some of the features of NPSS. Real-time monitoring, then verified and crop protection advisories given by experts of Government of India, DPPQ, and Plant Protection Department. And it reduces the crop loss risk as well. And instant and personalized advisories are given to the farmers. So how it works, how does AI works? Suppose a farmer clicks the image, the AI model checks it, whether the image is proper or not. And then if the image is not proper, it will again ask to reload or upload the image again. It will analyze the image, it will detect the pest or disease, and it will give the output and it will suggest the remedies. So these are features, as I explained, same as of CottonX. This solution is available in Indic languages, and expert curated farm protection advisories are already there. Accuracy is almost 96% with AI models, and continuous and real-time pest monitoring is happening through this. Currently, this application is being used. It is under testing by CIPMC officers throughout India. So these are roles and responsibilities at ministry level, at divisional or nodal officers, at state level, and agriculture experts and supervisors and extension workers, what roles they are doing in this solution that is given here. So this is the interface. This solution is also free of cost. This is available on the Play Store. Anybody can download it and use it. So this is how actually the pest disease detection takes place. The flow has been shown here. The first image is the first image you can see here. This is the original image which the farmer has uploaded. The AI algorithm checks it and it draws a bounding box around the target, and immediately it gives the result like this. It will analyze based on ETL or the grade of the disease. So this is how the solution works. So these are some of the features. We can integrate this solution with any of your applications which are previously available with you. And we can provide the APIs for this integration. And we are open for this because our aim is to reach out to the underprivileged farmers. So this is another solution. This is a web crawler which we have developed specifically for Ministry of Agriculture. This is a web crawler. What it does, so it gathers all the e-published news in any of the languages in India. And what it does, it compiles the information and it is provided on a dashboard in ministry. Various departments in ministry are using this application for their own purpose, that where to allocate the resources, where is the pest infestation, any news related to agriculture will be catched up. How that will happen, that I will show you. It eliminates the language barriers. These are some of the features of this application. Increased efficiency, instant dissemination of information, then human in the loop. Whatever solutions we are developing currently in AI, they are based on human in loop. Because AI has its own limitations, so we prefer to build the solutions which has humans in the loop. So this is how AI, our Krishi 24/7 works. So common crawler will be there. Whatever news are published online, they will be, these will be digital news. The Google alerts are also taken care of. The AI model accesses all this information. It compiles the information and puts it on the dashboard for the ministry users. So this is how it works. This is the process flow, how actually the users once logs in, the AI search for news, the news event is shortlisted, news alert is created, and that news alert will go to the nodal officer. Nodal officer verifies it and assigns it to the particular division in the ministry. And then it goes to the divisional coordinator for the investigation purpose. So it is verified both ways. And then the division coordinator disseminates the information or submits the investigation summary and along with the proof that this news has been verified. And after this, the nodal officer approves the investigation and an alert is generated. So this is related to any type of news which is published digitally. So these are, currently we are analyzing or crawling 12 languages, news published in 12 languages, and summaries are generated with breaking news alerts like we get on the news channels for the ministry people. And they are using it into 150 subcategories in agriculture domain. And also live feed for the latest agricultural news personalized only for particular division of ministry. They can summarize or they can filter the news particularly for that particular department. And there is a dedicated portal for this for the Ministry of Agriculture. So these are roles and responsibilities. I will not go into the detail for this due to lack of time. So these are the types of various departments which are using the different, the different divisions of ministry which are using this portal currently. And these are the different languages which are used by Krishi 24/7 right now. And this is the, interface, how it looks like. So it is just like ChatGPT. You might have heard about ChatGPT. So you can ask any type of question related to weather, related to agriculture, mandi prices, crop insurance, and it will give you the output in your language. There are filters provided. So this is just an example I had put here just to understand the concept. So what impact we have got through these solutions? So Kisan call centers, like in India, they received almost 20,000 calls per day, but most of the queries of the farmers are not being answered properly. So what we have done, we have reduced the call time. What AI will do, by using AI, we have reduced the call timings. We will get the query from the farmer registered, and the FTA, the farmer tele advisor, will call back the farmer. And we have also created a knowledge base for this farmer tele advisor. And that will help him, because otherwise what he does Google only. And that information many times is not authentic. So we are providing a knowledge base to the FTA as well. So my question to all the audience present here, if you have data, if you have systems in place, and if you have interest, can your data be interpreted by humans to predict required output? Please reach out to us. We are ready to help you to use AI in your work anywhere. So this is our team who has contributed to all these solutions. It was not possible to accommodate all of them because we are working on various fronts. Some are looking the ministry front, some are on the field directly, some are on the solutions side. So you can visit our website and you can have a look at our team and our other works which we are doing. So thank you. Now, any questions? Yeah. Yeah. So just a moment there. Give.
Thank you very much. This is a good one. Particularly the cotton eggs solution. And my question is, any plan to extend it to other developing countries? If no, I will advise that once the solution is fully developed, has been sponsored by Google Impact, you can seek FAO assistance, to extend it at least to developing
Yeah,
countries.
sure. So we are definitely looking forward to extend solutions to other countries as well. So definitely we can catch up. You can contact me. We can discuss in detail. So thank you. Thank you so much. Yeah.
Good afternoon. That is a very interesting presentation, ma'am.
Thank you.
What I would just like to know, I'm wondering if when you picture the disease or pest, could it be identified? Is there a stage that can be identified by your app? I mean, early stage of disease or early stage of the pupal stage?
Yeah. At any stage.
At any stage.
Yeah. At any stage, you are just supposed to click the image of that symptom or that particular damage which is caused to the plant part or any plant, and then you can upload it on that application. So currently that application is there for 5 crops: paddy, cotton, maize, chili, and mango. But slowly we are expanding to other crops as well. So horticultural crops and vegetable crops will also be included in that.
Thank you so much.
Thank you. Thank you.
Okay. Can we have Dr. Hare Krishna Swami, Botanical Survey of India, Eastern Regional Center, Ceylon? Are the other speakers present over here? Dr. K. Dharmar, he's here, I know. Who else is available for the session? Divyendu Chatterjee is here? Okay.
Good afternoon to all of you, everyone. Respected chair, co-chair, and the rapporteur. And I am very much thankful to the organizer giving me the opportunity to present my work before this, present my work on this conference. Now I'm going to tell about the diversity, distribution, and indigenous uses of hypericum species, especially in the Northeast India. The genus Hypericum is worldwide known for its traditional and some modern uses. For that reason, we made an attempt to access the diversity, distribution, and what are the indigenous uses of different species of Hypericum in the Northeast India. Specifically, most of the genus, whatever present in India of the genus Hypericum is mostly found in the Northeast India. And very few species is found endemic to different regions of India, like Hypericum mysorensis is only endemic to the Western Ghat, and Hypericum gaiti is only endemic to Odisha. But other species of Hypericum is mostly found in the Northeast India. So we have made an attempt regarding that end, because Hypericum has a anti-cancer property and anti-inflammatory activity as per the reports received reported from USA and other countries. So during our survey, we covered all the 8 states of Northeast India, where we got a total number of 18 species of Hypericum, and of which 14 are economically important. Okay, all the 18 species are native to Northeast India, and 4 are endemic, and 2 species are nearly endemic. Among the species, Hypericum lobei was the most valued species, which is mostly used in the medicine, powder, fuel, dyeing, and the extraction of hypericin. And what is hypericin? Hypericin is one compound which is previously reported from the foreign countries. Okay, and that is mainly found in the Hypericum perforatum in the foreign countries. And this hypericin is mainly used for anti-cancer and anti-inflammatory activity in the foreign countries. Then, due to over-exploitation of these species in the pharmaceutical industries and the natural population of this species depleting very fast, and the species has been placed under vulnerable category according to IUCN. Similarly, due to multiple utility of Hypericum hookerianum, Hypericum tenucale, and Hypericum williamsi are also facing the major threat. It is expected that, like Hypericum lobei, other species of Hypericum also may have the higher concentration of hypericin. Then in our survey, first that is the Hypericum hookerianum. This is the photographs of Hypericum hookerianum. Then Hypericum gracilis, Hypericum japonicum, Hypericum monanthamum, then Hypericum lobei. It is only found in the Meghalaya. And in Meghalaya, there are so many forest areas, but one forest area, reserve forest, is Maflang Reserve Forest, and this species is only endemic to that region. Hypericum williamsi. During our survey, we got the Hypericum assamicum, Hypericum bengalensis, Hypericum eleodoidis, Hypericum heliquale, Hypericum gracilis, and these are mainly found in the Assam, Sikkim, Meghalaya, Manipur, and Nagaland also. Again, this is Hypericum gramineum, Hypericum griffithii, Hypericum himalicium, Hypericum hookerianum, and Hypericum japonicum. And in this species, Hypericum hookerianum is a major threat due to multiple uses of this species. Then Hypericum lobei, which previously I told you this is under the vulnerable category of IUCN, Hypericum monanthamum, Hypericum petiolatum, and Hypericum reptans. Then these are the other species: Hypericum williamsi, Hypericum wittianum, Hypericum uralum. Okay, this is the Hypericum williamsi's photo plates. Here you will see the flowering branches and other flower photographs, calyx, corolla, bundle of stems, and the TS of ovary and pistils and everything. This Hypericum williamsi, during our survey in the complete Northeast area, this is the first report to the flora of Meghalaya, Nagaland, and Assam. Previously, it is not reported from all these 3 states. Okay, this is during our survey, we have first reported this, and it is under communication with Phytotaxa. And it is so interesting that Hypericum lobei, which is contained from the India, we are going to report it is a content of high hypericin content. It is recollected after 133 years of the type collection from the Maflang Reserve Forest. Okay. And it is only endemic to that area. In anywhere India or anywhere in the Northeast, no one has reported this plant till now. After that, we collection of the plant specimens, we go for the essential oil analysis. On the hydrodistillation, the fresh leaves and the tender parts of the Hypericum lobae and other species of Hypericum yielded 0.43% of pale yellowish essential oil. Then hydrodistillation was done by using the Cleveland type apparatus. The extracted essential oil was dehydrated over Na2SO4 and stored for under the room under the freeze in an airtight dark brown glass well until further analysis. Then we proceed for the GCMS analysis of this essential oil what we'll get. And for that, we get separation performed by the TG5MS capillary column. And in the GCMS analysis, we get that the GCM revealed that in presence of 40 compounds, which represent 97% of the total oil, and monoterpene hydrocarbons is 79% predominated, followed by the sequisterpene hydrocarbons to 13%. Of these, alpha pine is 70%, beta carophyllene is 11%, sabenin 6%, myriacin 3%, and other constituents. Identification of different constituents was based on by comparing the mass spectra of each detected compound with inbuilt NIST mass spectral library. Then we go for the antioxidant activities with three different systems. We have done this. One is DPPH analysis and ABTS analysis and reducing power analysis. Essential oil exhibited moderate antioxidant activities as compared to BHT and ascorbic acid. These are the antioxidant activities of different Hypericum gracilis, Hypericum japonicum, and Hypericum hookerianum. Then conclusion is that the present research investigation of this chemical composition and antioxidant activities of essential Hypericum lobae is an endemic species to the Northeast India. Our results indicate that the oil is the rich source of sequiterpenoid, monoterpenoid, and alpha-pine, beta-caroline, and predominant constituents. And this oil showed moderate antioxidant capacity that may be due to the fact it is composed of entirely of terpene and hydrocarbons, which is not an active antioxidant. This study also reveals that other species of the genus may be used for multi-purpose, and therefore this species may also require prior attention to for the conservation. Population assessment of the rare and threatened species must be required. Mass multiplications through conventional in vitro method of rare, threatened, and multi-purpose species is also suggested. These are the works, and I am very much thankful to the Director Botanical Survey of India, Head of Office Shillong, and the South Indian Textile Research Association for doing the GCMS analysis, Dr. R. Kotaimuthu, Y. Mahesh, Riyaz Ansari for the same technician, and my supporting staffs for doing this work during the and other all the Northeast Forest Divisional officers for their cooperation during the survey. These are the Juko Valley, beauty of Juko Valley during our Nagaland tour. This is one of the photographs of Juko Valley. Thank you.
Any questions for him? No. Thank you. Now, can we have Dr. K. Dharmar, Pasupam Thiru Muthu Ramalinga Thevar Memorial College, Kamuthi.
Good afternoon, everybody. First of all, let me give my thanks to everyone dignitaries in this 6th International Conference. It's a small part of our research. Optimizing shoot cutting propagation of Arundodonax. Arundodonax is a perennial weed. It is distributed in every area. But it's a non-food tall plant. It's a grass. A particular community they can use to make a Sivali. It is a airborne instrument to use in long back. So it is very important, in particular in Tamil Nadu, because this plant distributed in everywhere. They can use various uses. But the particular group of tribes which is located in a Kaveri river bank in Tamil Nadu, Tanjore, that group to make such a wonderful instrument. It is a multi-purpose weed. They do not produce viable seed. They will cultivate it through rhizome cultivation or the axillary bud propagation. Methodology. The traditional method of rhizome propagation is somewhat difficult. The aerial shoot which produce the axillary bud, these bud easily germinate when we use the crowback method. We have selected 3 types of soil, and we will selected disease-free, healthy plant where the artisan to use for making Sivali or reed. There are 20 healthy axillary buds were cut from lower region, middle region, and upper region. The tip of the Arundodonax to leave it. Because in which part of the axillary bud which grow in a particular type of soil is very in good conditions. The main objectives of the study is to produce mass production of multiple growth of the Arundodonax and to compare the growback method cultivated plant with the naturally occurring Arundodonax for making the Sivali. Data measurement and evolution. The plants were evaluated for raw material region were Sivali. Outer diameter, inner diameter, wall thickness, and the node diameter. It's very important. And also the length of the internodes. These criteria which is very, very essential for making Sivali. This is the Kaveri belt area where the Arundodonax which is occur in freely. We will collect the Arundodonax plants and cut it into 3 parts, the lower region, middle region, and the upper region. The tip of the region is removed it. Then we cultivated in the 4 type of soil. Then the length of the selected axillary bud internode is 5 centimeter. It is a this is cultivated growback method. Then it's transplanted this material when it attained 30 days. After this, we transferred to the field. This is the field conditions. Result, Sivali, the natural raw material. The randomly selected dry raw materials approximately 3 to 4 feet of the shoot portion were collected from the artisan. The part of the OD of the diameter 1.40 plus or minus 0.14 centimeter. The internal length of the 12.90 plus or minus 1.76 centimeter. The OD of the internode is 1.4642 plus or minus 0.13 centimeter. The inner diameter of the internode is 0.88 plus or minus 1.6 centimeter with wall thickness 0.27. Before the independent, this is a Nadasram. This is a wind instrument. They can use every celebration of Hindu festivals. This is a Sivali or reed. This is the raw material of the Arundodontax. They collected and dried it. Then we make the Sivali or reed with the help of a fine needle. Then after that, they will put into this portion, they will use it. The nature of the Sivali or reed is very important because it's vibrate the tone finest and control the instrumentation of the Nadaswaram. This artisan, they will make the instrument in generation after generations. Nowadays, these artisans' activities is reduced due to the raw material shortage. Because these plants grow when the water is locking conditions. It is obtained the height in the river bank for 90 days. So after that, when the Kaveri delta regions decrease the water flow, automatically the plants are destroyed. The farmers knowingly or unknowingly, they are making fire for other purposes. So the raw material availability is very, very low. The artisans also very few in number because a group of people only, they will know to make how to make the Sivali. This is the material. We will get the natural conditions. The clay and upper part, they will plant dirt, that will get this region. This is the very important one because I already told that the outer diameter, the inner diameter of the internode and nodal regions is very important, and also the thickness of the wall. So the clay soil lower portion and the sandy loam soil lower portions, they will only produce the nearly equivalent to the natural habitat of the material. This is the indoor node region. There are four types of soils: clay soil, upper part, clay soil, middle part of the material, and clay soil, lower part of the material. More or less, the clay soil, lower part of the material, they grow equivalent to the natural habitat of the Arundodontax. Similarly, the sand loamy soil, lower portion of the materials grow equivalent to the natural habitats. So these two soil which very suitable for the growth of the material which is equivalent to the natural portions. This is the inner diameter. The same. But inner diameter, most probably the clay upper part of the plant and these two, sandy loam middle and sandy loam lower part. Even though the silt soil lower portions also grow well. and the wall thickness. The wall thickness, the clay lower part, and sandy loam soil lower part, and sandy soil, these three parts which is equivalent to the natural world, more or less similar. Certain it. Then nodal region. Nodal region also the clay soil and sandy loam soil lower portion, and the lower portion they will equivalent to the natural portion of the Arundel-Donax. Result in growth propagation. In clay soil through axillary bud from lower shoot part, it gave intermediate length of 11.98 plus or minus 1.92 centimeter. It is equivalent to naturally obtaining Arundel-Donax internode length. And with the OD of internode 1.30 plus or minus 0.10, and in the diameter of the internode is 0.86 plus or minus 0.09 centimeter with wall thickness of 0.22 plus or minus 0.03 centimeter. Similarly, in sandy loam soil, they say more or less same result we got it. The study concluded that the axillary bud from lower stem part grow well in sandy loam soil and clay soil. The propagation of the protocol can be suitable and economically viable method to establish Arundel-Donax. Conclusion. The protocol described in the study is an efficient, rapid, and simple technique for mass propagation through axillary bud. 3 to 7 days, emergence of shoot from axillary bud was observed. Axillary bud induction 67 to 83%, and number of shoot and plant height generated were significantly higher in sand loam soil. Plant propagated through this method suitable for Sivali making. This is very important because of our objective is to compare the natural habitat Arundel-Donax with the crowback method cultivations, which is suitable for the Sivali making material. Hence, this technique can be applied for mass propagation of Arundel-Donax and well suited for the conservation. Other commercial application of Arundel-Donax, biomass, biogas, and bioethanol production, bioremediation, paper pellet, and furnitures. There are so many uses for Arundel-Donax, but in India, there is no previous literature which is available for these uses. In foreign countries, they can use for, because the Arundel-Donax is a high biomass material, they can easily produce biogas, biodiesels, and everything. Thank you.
Thank you, sir, for nice presentation. If any question is there? No question? Then I think we can go to the next speaker. Okay.
Thank you.
Can we have Dr. Divyendu Chatterjee, ICAR National Rice Research Institute, Cuttack?
respected chairman of the session, co-chair, rapporteur, and all other dignitaries. The topic I'm going to present is enhancing phosphorus availability through nanoclay polymer composites impregnated with phosphorus and low molecular weight organic acids. And I'm Dibyendu Chatterjee, Senior Scientist, Soil Science Division. Actually, phosphorus is such an element that it is having complex reactions in the soil, and because of that, it is many times inaccessible to the plant. Now, the low molecular weight organic acid, we have our previous work that we have seen that it can increase the concentration of the phosphorus in the soil solution. And because of that, we thought we can start a work where we can synthesize a polymer, a nanoclay polymer composites, in which we can put both the phosphorus and the organic acid to have a composite which can help to giving phosphorus in a better way than the conventional way. So nanoclay polymer composites is the material consisting of nanoclay particles, which is dispersed within the polymer matrix. Mostly we are utilizing polyacrylate polymer. So in the polyacrylate polymers, it is dispersed. And the purpose of using this clay is to give mechanical strength, thermal stability, and barrier properties. Now, this is a diagram where we try to show the NCPC. We have synthesized the acrylic acid and acrylamide are co-polymerized in presence of nanoclay. We are using kaolin here, and with ammonium persulfate as the radical initiator and N-methylene bisacrylamide as the cross-linker. And after that, we dry it, and after drying, we grind it and sieve it. So this is the conventional way. And our hypothesis, already I told, we want that it should improve the availability of both fertilizer phosphorus and the native phosphorus in the acid soil. And we observed that there is a research gap because very limited work is done on this topic. Now we have briefly two objectives to characterize these composites for water absorbency and feed release, and second, to identify these smart delivery systems, specifically for the upland rice. And while doing it, we have three sources of low molecular organic acid: citric, malic, and tartaric acid, and three phosphorus sources, that is diammonium phosphate, DAP, SSP, and rock phosphate. We are making 9 combinations, and the 9 combinations we have a challenge because the solubility of DAP is very high, whereas SSP solubility is moderately soluble. And the rock phosphate is water insoluble. So we have a different kind of solubility. So we thought of make one uniform method of synthesis, because mostly the low molecular acids and the phosphorus we used to dissolve in the water, and then we allow it to go inside the polymer matrix. But here, since we have 3 different type of solubility, so we thought of making a new process. So here it is a synthesis of the NCPC, where acrylic acid and acrylamide was, dissolved in water and then neutralized. Then after that, we rotated in a magnetic stirrer. Then kaolin was added, and then N-methylene bisacrylamide was added to it, and then it was mixed at the room temperature. Then we raised the temperature, and the radical initiator, I told that ammonium persulfate was added, and this polymerization starts. And here what happened in between 3 and 4 steps, we did this modification, and we add this polymer, the phosphorus source and the low molecular weight organic acid. So these are the different variants we have prepared. And first, we after synthesis, we did the characterization of the product. And here first we check it for maximum water absorbency. And we have seen that per gram of our material can absorb around 83 to 88 gram of water. So water absorbency is quite high. And we check the pattern of pre-release, because DAP was completely soluble in water. When we check that that it giving, if we make even after adding step 3, if we are making the product and we are using the conventional product, if we compare both, we have seen that both have a similar pattern and quantity of release. So we decide that let us go with the method which is direct addition of the material into the process. And what it will happen, if we do like that, then it will save time and also, energy, because if you dry again, reabsorb again dry, so some kind of heat energy is required. So we can save that energy. So it was our first observation, and we modify the steps accordingly. Then we try to make, we try to see that how much maximum amount of these fertilizer we can load into that matrix. And we keep on increasing, and we can see that DAP at max, we can give 10% weight by weight, SSP 20%, where RP is 10% weight by weight. And, when we are adding in higher quantity, we have seen there is slowing of the polymerization. Probably it hinders, that polymerization process. And then some sticky consistency of the product, we cannot take it out easily. So maybe it will create problems. So this observation lead us to confine our work to 10%, 20%, and 10% of these three fertilizer sources. Then also we studied the release pattern of this in the soil, and we found that simultaneous application of DAP with a low molecular acid give a better concentration. Same way, we also study the release pattern with other variants like with SSP, and we found the power form of release, and also we see that the low molecular acid hastens the initial release. Same way, we also study the pattern of the product with RP, and we also see that power form of release curve. And same materials are also tested against the release in soil, and we found that the variant with tartaric acid with the fertilizer give better release in the soil. Now we come to the instrumental characterization. First, we did X-ray diffractogram analysis, and you can see there is a hump in the just starting of the X-ray peak. So that hump, that suggests that there is a increase in the interlayer distance due to the nanoclay intercalation. And we have also a peak of pure kaolinite, and we can compare with this peak, and we observe that where there is an intercalation, because the polymer sometimes intrude into the interlayer of the kaolinite, and also sometimes the surface hydroxyl group of the kaolinite are making graft polymer. So in the main chain there is from side chain they are making. So that graft polymerization and this intercalation, these lead to decrease in the intensity of the kaolinite peak. So this was proved by our X-ray diffractogram analysis. Further, we did scanning electron microscopic analysis. So same photograph we take, and then we observe the different structures there, and you can see the different structures here of the different materials. And then also we did FTIR analysis, and we could see the the wave number or respected to the materials used. So same way, these are the list of the different FTIR of the different NCBC variants. Then we tested in a pot, and we found that NCBC plus DAP plus TA and NCBC plus SSP plus TA, these are giving comparably better results as compared to the other variants. Further, after the pot analysis, since this material in the lab, synthesis is a bit difficult, because we don't have that large scale synthesis methods, and because of that we use microplot to study its field effect. And then once we did, then we can see that if we use if we compare the results with no fertilizer application, no P application, then these variants with low molecular acid, particularly tartaric acid, can give 19 to 28% higher yield. While we compare with the conventional DAP, then it can go up to 8.3% yield advantage. Now these are the photographs, and also we studied the soil properties, and we can see there is a higher P where we are applying phosphorus, but it is depleted in other case. And pH is also slightly improved, because the pH of NCBC is, we have seen that it is a neutral pH. And then P fixation also, where we are using, because of the chelation, the P fixation is also decreased. Then we also studied the phosphorus fractionation by using modified Chang-Jackson method, and we can see that the fraction aluminum and iron phosphate fraction is readily replenishes the phosphorus pool where we are applying these type of materials. So this is the other pools, occluded P and then calcium P. Calcium P also, it is very high quantity, but it doesn't change because our soil's pH was near to 5. So because of that, and then I can conclude we have two products. One is NCPC plus DAP plus TA, NCPC plus SSP plus TA, which can give better yield and can reduce the fixation and can give high water absorbency. But it requires some further modification because we are using acrylic acid as an ingredient. And once we are calculating the, the cost of the material, it becomes higher than compared to the conventional. So until unless we make it cost effective, it cannot, go further. But there are some way, I have seen some literature, maybe by replacing some of the costly ingredients, if we find some alternate ingredient or alternate, polymer system, if we can get, like we are using polyacrylate, maybe we can think of some other polymer system which is comparatively, low cost. So those things can be, but our target was to establish that these things are working. So it works. So now next step is to go out for the more cost effective material. Thank you. Thank you very much. Thank you.
Any question is there? So there is no other question. Hopefully.
Okay. Thank you.
So next speaker.
Is Miss Vatsala call here? Okay. Dr. H.C. Parmar. Dr. Mohammad Shahid. No. Can we check with Dr. Nandakumar K? He is about to connect on a Zoom call. Okay, Shiva Shankari. Dr. Shiva Shankari, yeah? Yes, ma'am, please. Screen switch.
A very good evening to one and all present here. Myself, Dr. Sivashankari. I am a food processing engineer working as a scientist at ICAR-NRRI Cuttack. Basically, I'm working on the fortified food products, means new food product development, rice-based food product by incorporating the natural sources like be it fruits, vegetables to enhance the nutritional value of the product. So basically, today I'm going to present this work, which is the development of rice millet based ready to eat extruded products fortified with underutilized crop of Eastern India. So okay. So when we talk about the prevalence of malnutrition in children, it's a global health challenge that significantly impacts the physical and cognitive development and also the overall well-being of the children. So despite having advancement in healthcare and nutritional awareness, the presence of malnutrition, it remains a critical issue in many parts of the world. And there are two types of malnutrition, we can say. One could be undernutrition, and another one could be due to the deficiency of micronutrients. So the main causes, if we see for malnutrition in children, it could be due to the insufficient dietary intake, poor feeding practices, or infection diseases, or the socioeconomic factors. So to address this malnutrition, the development of the fortified food products is also a crucial, it plays a crucial role in addressing this major issue to a major extent. So rice and millet, as we all know, they are the staple food in India, known for their nutritional and also their versatility in the food production. And when we talk about ready-to-eat products, they're convenient products. So when we develop more of ready-to-eat products, like this can be made accessible to areas where there are no options for diverse range of food products. And also, since it is convenient to use, it will be preferred by children and majority of the population also. So extrusion is a technology which is used to develop these ready-to-eat convenient products. As I said, these are products which will have a versatility and also a better shelf life. So in this study, we tried to fortify the rice millet based product by incorporating the water spinach, which is one of the underutilized crop because it is rich in nutrients, but it has not been commonly or most popularly used in the normal diet. So we try to incorporate that as one of the ingredient in our product to enrich the nutrition of the snack product. So as you see the nutritional profile of this green spinach, like it is rich in like majority of the minerals like calcium, potassium, magnesium, iron, zinc, copper, and manganese, and also it is rich in proteins and carbohydrates and also fiber as well. So it includes, when we talk about the underutilized crops, this is one such crop that we try to incorporate. But other than that, there are many other crops whose knowledge, like we can say the traditional knowledge, the people from that area, they know the importance of that crop. And also like locally, they'll be using it for it's like as a medicine also. So, but when we try to, means when we incorporate that highly nutritious and also it is underutilized also, and in a way that it will even encourage the people to cultivate that type of crops in a larger scale. Like if they get to know that it has the medicinal potential and also has a demand in the market in developing these type of products. So we can retain the traditional knowledge of those people also, and it will be an encouragement for them to cultivate such crops in a larger scale. So this is what. So preserving the traditional knowledge and also the farming practices is also essential for maintaining the agrobiodiversity, ensuring the food and also promoting the sustainable agricultural systems. And also when we talk about the impact of genetics and all the importance of biodiversity, genetics, it will influence the characteristics of the crops, including the nutritional content, yield, quality, the resilience to the environmental factors, and also resistance to the pests and diseases. And biodiversity is essential for the crop improvement as it provides the diverse genetic pool for breeding programs, ensuring the development of resilient and also the high-yielding crop varieties. So, but there are basic challenges when we talk about development of the extruder product, because maintaining the uniform composition of the fortificants, and also we should make sure that there is means during the process there is a minimum of loss that is happening that in terms of nutrition, and also we should have a check on the sensory and acceptable properties of the final product as well to attract the consumers. If it is not accepted by the consumers at the end, there is no point in like developing. So all these things has to be kept in mind when we think of developing an extruder product, because it is a high temperature process. Obviously, when we process these nutrient rich, because minerals, it is means temperature stable, thermal stable. But we talk about vitamins and other nutrients like anthocyanin and all, it is temperature sensitive. So when we process it, these parameters has to be optimized to make sure that there is no loss or there is minimum loss in the final product developed. So the techniques for successful fortification will be like during the extrusion, it involves, as I said, its selection of appropriate fortificants and also optimizing the processing parameters and conducting a thorough sensory evaluation to ensure the quality of the final product. So here, along with rice, and like we for this study we have taken black rice to which we have added millet, corn, and the green leafy vegetable. And through the experimental design, like we conducted experiment where the independent variables, that is the process parameters of the extrusion technology, the barrel temperature, screw speed, and feed moisture content were kept in range, and experiments were conducted to optimize these parameters. And we made sure that in the final product, from the sensory analysis, it is acceptable. It is in the acceptable range, and also with the maximum anthocyanin content in the final product. These are some of the other ready-to-cook and products that has been formulated and standardized here at NRRI. Like we use rice vermicelli was developed by mixing millet with the black rice, and rice with millet, again rice ragi pasta with a maximum retention of vitamins and minerals has been standardized. And rice vermicelli, and this third one, it was made from the CRDAN 311, which is the high protein rice released by NRRI. And so it means like we claim that it is a high protein rice vermicelli. And similarly, rice ragi pasta, again, it is with the high zinc rice was used, and it is fortified with ragi, means like millet. And again, this is another rice pasta with millet made from high protein rice. Similarly, we tried to standardize the formulation for noodles, because as we all know, kids, they enjoy noodles. But to get the healthier version of the noodles, we tried to fortify the noodles, like, and instead of wheat or atta, we used rice, which has been fortified with vegetables. Here we used the drumstick leaves moringa, and this is all natural color. We haven't added any means artificial colors to it, and beetroot and carrot, and that has been standardized for having the better and the better cooking properties. Similarly, this kurkure type of product, again, we used rice along with maize and dal. So to enrich its protein content, and these products have also been optimized and standardized. And that last one, this is the result of this study, like which has been the rice kurkure that has been fortified with the green spinach. Similarly, like we have developed some bakery products also made from rice and millet. These are some of the ready-to-use instant mixes, idli mix and dosa premix. And also these are some of the beverages, rice milk developed from the aromatic NRRI released aromatic rice varieties. So, and also apart from this, we also give hands-on training to the people for having their own enterprise. So this picture, you can see that we gave training to a group of women to, we taught them to develop some 15 bakery products, which included the millet-based, rice-based, and also they had given means like skills, techniques, and the knowledge about the machineries, how to use, and also they will be registered as an incubator under us, and we'll be giving them hand-holding support to have their set up their own enterprise. Thank you.
Any questions for her? Yes, please.
You have developed how many products? Maybe 20 varieties. So if you prioritize, what is like in a market gives you which one is the better preference than other? Because what is my personal opinion, if you go for spreading so thin, it's very difficult to catch a market. So I don't focus on a prioritize 1, 2, 3, and then you start spreading. That I think would be better actually. And I think these all products you have evaluated the nutrient availability, right?
Sir, it is in the process. We are doing it.
Okay. And the purpose is like, since it's a millet year, where the idea came, like,
So last year, since it was a International Year of Millet also, that is also a reason. And means, and we try to use our rice varieties. And
Okay.
we try to study the suitability of our released varieties, like for what type of product it is suitable for.
No, the point is the rice variety you are looking at, does the paddy is having a pan-India presence? The variety you are talking about.
Sorry.
The variety you are talking about, the whatever you told,
Hmm.
your released variety, the paddy has the variety has the pan-India presence? Pan-India means across India people are doing that farming? No, why I'm telling you, because the product you are developing, paddy cum ragi, actually you need to look into the pan-India presence. So it will be acceptance will be much more instead of
Okay.
like localized. That's what the point.
Okay.
Thank you.
Okay. Thank you.
Thank you, ma'am. Can we have Dr. Vijay Kumar? Is he here? Dr. Vijay Kumar online? Okay. Is Dr. Rajni Kapoor available online? Can we check with that? Dr. Rajni Kapoor or Dr. Nandakumar K. Are they available online? Can we check with our Zoom link, please? Please check the Zoom link, Dr. Rajni Kumar or Dr. Nandakumar K. Rajni Kapoor, sorry. Available? Yes. So, Dr. Nandakumar, are you available online?
Yes.
Sir, you can start your
Yes, madam.
presentation. You can start your presentation now.
Okay.
Yeah.
Okay. Good afternoon to one and all who all are present national conference at Cuttack, Odisha, and Dr. Nandakumar Kray. And presently now I am working as a CEO at in the reward project at University of Agriculture Science Shivamogga, and the sub campus that is called Babbur, and the okay, and this is my part of work during my master degree, which was conducted at Zonal Agriculture and Horticulture Research Station, which is concerned the same campus that is case in Shivamogga, Karnataka. Okay, the title of the research evaluation of coriander genotype for both yield and quality and zone of Karnataka. And so the experimental studies, I will introduce my research crop. The coriander, it is one of the important seed spice, one of the horticulture crop. It can be used as a vegetable purpose also, and as well as the spice purpose. And it is botanically called as coriander, and it is belongs to the family Apiaceae, and it's the chromosome 22, and it is one of the ancient origin by the traders to the Asia, India, as the Mediterranean region is the so there are different two types of the coriander seeds, that is Coriandrum sativum, variety microcarpum, sativum, variety macrocarpum. We can also there are bold seeded coriander seed as well as the small type. Yeah, they are the two important seed spice with respect to the aroma. Yeah, the coriander type which contains highest lineal and have the high market price in the content over the variety macrocarpum, yeah, which is a bold type. So coming to the uses of the as we know that the coriander, which can be used herbs type as well as the seed spice type, and used for the especially in curry purpose as a cooking, as well as the which can be used in case of the baking, sausage and pickle, candies, and soup. Yeah, these majorly used for the extraction of essential and the which have the high value in case of aromatherapy and soap making and the food flavoring purpose. That is 60 to the linalool content was identified over the fertile. Yeah. And it also rich vitamins and minerals also the especially like and vitamin K as well as the calciums, and that's why they are used for the salad purpose also, and even the different pharmaceutical property have been identified. Okay. Some brief background of the crop. Okay. So coming to my experimental details. Okay. This experiment was conducted at the Zonal Agriculture and Horticulture Research Station, which is under the University Shimoga in Karnataka. Okay, the experiment was designed in PD design, that is randomized complete block design. The experiment, the number of treatment have been used as a 20 genotypes, and the number of replication is 3. And the spacing is usually followed by 30 by 22.5 centimeter. The plot size is 2 meter by 1.5 meter. Yeah, it is 3, and the number of plants per plot was 44. So the main aim of this experimental, the experiment was conducted. The Hiru region is so the some of the small as well as they are usually go for the cultivating special crops, and as well as the another important thing is the cultivated only for the leaf purpose, but the through leaf purpose they cannot get much more price in the market by comparing the seed production purpose. We have evaluating some of the important seed purpose, some of the important of the different research stations. Yeah. There are the different genotypes have been used for this research, and which was collected by the different states like Rajasthan Agriculture University have been collected, and then Tamil Nadu Agriculture University, and that they Surrender 1, 2, 3, 4 collected, and then Gujarat Agriculture University GCU1, and then from National Rice, National for Seed Spice Ajmer. From that we have collected one ACR1. Yeah. And then we have locally collection like Devi Hossur Coriander collection 1, 2, 3. They are locally which is near to the location we have also checked. Yeah. So these are the genotypes we have been identified and collected these seeds, which was sown during the Rabi season, during the 2016 and 17. At that time, we have conducted this experiment. And coriander is biennial, the other seeds, especially in case of seed production, sown during the Rabi season crops. Okay, for the leaf purpose and the entire year we can grow, but for seed production, the Rabi season, because it requires cool and dry weather for the production of the flower. Okay, so that's why we have sown during the month of November 40th by using these the 20 genotype package of practice which have been given by the university. Okay. This is the objective of these the main important thing is to evaluate the for both yield quality parameters and to the variability for both yield quality parameter in the core-end genotype to study the correlation path analysis for yield and yield. Okay. So the observation was recorded 30, 60 and at the time of harvest were parameters like plant height, number of primary branches, and then plant spread. Yeah. And the days for first flowering, days to 50% flowering, umbels and umbellate per umbel, number of seeds per umbellate, days to maturity, seed yield per plant and per plot, and per hectare, and dry weight of 1,000 seeds. Okay, and then finally harvest. These are some of the yield parameters and quality parameters, which is the essential oil content was extracted by using hydro method by the Clevenger apparatus. Okay. And then genetic parameters. Okay. Analysis of variance and genotypic variance and phenotypic variance, and PCV and heritability and correlation and path analysis. So these are the parameters while going for the evaluating the different genotype. Phenotypic nature of the genotype, we cannot go for the selection individual genotype based on the GCV, PCV. We can reduce the effect and whether is the heritable nature of the individual genotype based on their additive as well as non-additive gene effect. Finally, correlation path analysis based on their direct as well as the indirect effect over the seed yield per calculated, and then go for the final the suitability of the genotype in the particular location we can calculate it by these parameters. So so this is the ANOVA table. The analysis of variance growth as well as the yield parameter of these selected genotype calculated by the this ANOVA table, and in the find that there is significant difference parameters like number of plant height, primary plant, secondary. Okay. As well as the yield parameter. For all those things, we have find that there is at 5% as well as the 1% level we have identified. Based on that, we have concluded that there is significant study. Based on these variation, we can go further select. Based on these one season data, we have get the phenotypic character of the 20 genotypes, and we have difference for all the 17 from starting from plant height, number of primary plants to essential eye. Okay. So these data was recorded during of the plant, that is 30 days after sowing, 60 days after sowing, and in all those parameters, the genotype 475 has been found that the maximum plant height during 30 days, 60 days, as well as the time of harvesting. Number of primary branch also, this genotype has been other genotypes. Okay. With respect to days to first flowering, and Devi Hosur collection 4, that is it requires very less days for the first flower, that is 39.33, as genotypes. With respect to days to 50% for that also the DCC4 recorded duration. With respect to days to maturity, DCC3 found that early mature type compared to the other genotype. With respect to the yield parameters like umbels per millet and seed per umbellate, seed yield, plot, seed yield.
Dr.
Which was,
you have another 1 minute left only.
Sir?
You have another 1 minute left, so please conclude.
Okay. So for all the major growth as well as the RCR 475 recorded the superior over the other genotype with respect essential oil content, quality parameter, the RCR as well as the ACR1 recorded the best over the other genotype. With respect to the in almost all the for this today the 17 the traits in these the GCV as higher than the GCV. So the GCV and PCV have narrow genetic base, which indicates that there is less influence of the effect, and some of the heritable nature. In that also it has the highest heritable as well as the parameter essential. And based on these, some of the important criteria I will go for the selection of the traits for the particular genotype. We have to go based on these traits for the selection of the genotypes. And other thing is the phenotypic correlation. The phenotypic correlation is to identify the association between the and these 17. Sir? Hello.
Your time is over. Please conclude.
So based on the different 20 generated under the central region of Karnataka, we have the early maturing genotypes like disease 1, 2, 3, 4, as well as the yielding genotype 20 and 446 will be best for the earliness for the seed production, whereas seed content 475, 446, 41, and 41 will be suitable for the. So I will conclude by evaluating these genotype, which will the in and around the hilly region, the small and can be go for the seed production, which will be boost for the economy of the farmers of the hill. So superior genotype while conducting that is RCR 446 and RCR 475. This is the RCR 41. The seeds of these are the two small seeded variety. It has high aroma content than the bold seeded type. Okay, these are the bold seeded one. The essential oil.
Thank you, Dr. Nandakumar.
Okay.
Thank you, sir, for
Okay,
a talk. Okay. The last two speaker for the session, Dr. Vijay Kumar. Is he there on Zoom call? Can we check with the participants, please? Dr. Rajni Kapoor. Change the screen. Okay, so for now, we'll take a break and we have another closing session in a while that will start shortly around 4:30. We take a coffee break now. Everyone is requested to please collect their certificates. I hope you people have done that. And also do not forget that in case of any pictures that you have clicked, please post it by tagging SARM Odisha, SARM ODISH. Thank you. Check. Okay, Dr. Vijay Kumar, I guess you're there online. Okay. Dr. Vijay Kumar and Dr. Rajni Kapoor, ma'am.
Yes. Yes, I'm here.
Okay.
Am I audible?
Yes, ma'am, you're audible. And you can start your presentation too.
Yes. Is the screen visible?
Yes, ma'am.
Okay. Am I able to move the slides? No.
Currently, that window screen was open. Can you please try? Yeah. Please close this, ma'am. Yes. Yeah, ma'am. Your slides are moving. Ma'am, you can start.
Okay. Is the screen visible? Okay.
Yes, ma'am.
Fine.
It is visible.
Hello.
Yes, ma'am. Ma'am, are you there? There's some glitch with the Zoom call. Can we try to establish the connection again? Ma'am, if you're there, please start your presentation. Your screen is visible too. Dr. Kapoor? Dr. Kapoor, please unmute your mic. Dr. Kapoor, please unmute your mic first. You're not audible, ma'am. Dr. Kapoor, if you can just reconnect. Yeah, you're audible now.
Okay.
Yes, ma'am, please start.
Yes, I'm going to present my paper on the topic analyzing the impact of economic freedom on farm efficiency. It's an empirical study. It is based on my PhD work. So, regarding economic freedom, my paper has an empirical approach to investigate the impact of economic freedom on farm efficiency. Economic freedom in agriculture refers to a set of multidimensional initiatives including policy regimes, property rights, supportive programs, technical updates, marketing reforms started by the government to enhance the productivity and efficiency of the farming sector. These options and opportunities are implemented with a legal and democratic framework. So there is the institutional role, and in my study, I studied its impact on the farm level efficiency. So the research problem is there, as there is inefficiency existed at the farm level. Okay, it means higher inputs lower the farm output and vice versa, and there are examples. And the studies, I could say, in the literature review that have introduced institutional reforms, rural development programs, higher wages or farm job opportunities to enhance the efficiency of the agriculture sector. And there is significance of economic freedom in the field of growth and development that was specified in several studies. And economic freedom is empirically estimated by Eastern Walker, and there is a literature that actually underline the capabilities that can be enhanced through freedom options and opportunity that are given to the farmers. And in the agriculture sector, the significance of economic freedom was explored by Gulatis and other studies. In this regard, my study has contributed and explored an empirical approach to determine economic freedom index. First of all, what are the determinants, how to measure economic freedom index. Then there are specific objectives of my study, and supporting research methodology is there. The paper aims to assess economic freedom index, okay, which is the composite average of 4 dimensions of economic freedom index. All indexes are measured using principal component analysis to examine its impact on economic efficiency. So economic efficiency is calculated through stochastic frontier analysis to measure farm level economic efficiency of cereal cultivation. So basically rice, wheat farming. Then Tobit regression is used to examine its impact. So input output information was collected through a primary survey. As per the theoretical framework is considered, as I said, so there are 4 determinants found from the literature of agricultural efficiency that are supporting the growth and efficiency of the farming sector, and there are sub-components or indicators too. These are labor freedom, financial, technical, and business freedom. So there is the area of research, that is the Haryana state. So the fertile state, okay. So regarding there, a total of 336 farms were selected from the different villages of Haryana, and there was the questionnaire, okay. So to get the responses for different freedom indexes and farm efficiency. So scheduled questionnaire was designed to obtain responses for each measurable indicator. So that is used to assess individual indicator on a 4-point assessment scale. There are several other indicators like agricultural property rights, wage contracts, rural development policies are the indicators of labor freedom. Then stable input prices, stable output price policies are the indicators of financial freedom. Further, we have technical freedom, research and extension services, intellectual property rights, information and communication technology are coming under technical freedom, trade liberalization and protection policies for food security and sustainability coming under the business freedom. Then principal component analysis is used to reduce the data to estimate the weight for each indicator of the determinant of economic freedom as a latent variable. So the result states, yes, economic freedom index is calculated. Weighted mean of economic freedom index for the medium farms is the highest one, that is 2.77, which is greater than the small size farms, that is 2.58, marginal and landless farmers. So there are 4 categories of farm size taken in the study, hence the estimates truly reflected the average score of the determinants. So there is a positive association between the farm size and the economic freedom deliberated by the farmers. Farm-wise intensity of economic freedom states that 80% of the medium farmers have the moderate score, which is greater than the smaller, that is nearly 35%. Then to marginal and small farmers, marginal and landless farmers to 4%. So this is farm-wise intensity of economic freedom index. Shortage of time, I have not actually presented all the labor freedom, technical and business freedom indexes. So this is the average one. Then there is the farm-wise weighted, yes, average of all freedom indexes and positive association between the farm size and intensity of freedom indexes found. Still, the extent of determinants of economic freedoms differs across the farm size. Freedom scores varied across farm size and results are generic and expected. There is a summary statistics of input-output data used in the cereal production. So it is found that marginal and small farmers are better utilizing the resources. So input used in the cereal production are land, labor, seeds, fertilizers, pesticides, electricity, machinery. Input use varies farm size, and it is found that average farm-wise income of medium farmers is higher in comparison to marginal, and again it is higher to small farmers. So there is the paradox. So while the rational use of input is made by the small and marginal farmers, but the revenue of the medium farmers is greater than marginal and small farmers. Labor requirement of marginal and medium farmers were low as compared to small farmers. So that is the one reason I could be able to deduct the labor cost. Labor cost is higher for the medium farmers. Then there is farm-wise stochastic economic efficiency calculated, and the size specific variation is also found. There is, I could say, the estimated mean of economic efficiency of overall farms indicated that there is economic inefficiency, that the overall sample results are displaying here, that 85% of their potential output is foregone. And 15% of the farm produce is foregone due to cost inefficiency. So it is found that the production capacity existed to increase the farm, means there is actually a space, there is a existed capacity that can be reused. So 15% inefficiency is found. It means more opportunities and support can enhance the efficiency. So inefficiency reduction is also very important. So production capacity existed to increase farm output by 10% from the given input. So these are the stochastic economic efficiency result. I could show this graph, and there are the results related to economic inefficiency, that when the values of gamma and sigma square deducted. So the results state that 99% of the variation in the output is due to inefficiency component, and 1% variation is due to random shocks in the agriculture sector. And for the marginal size farms, inputs such as seeds, pesticide, estimated positive relationship. All the inputs are almost having positive relationship, means larger the use of the inputs, the efficiency increases and significantly enhance the civil economic efficiency by 21 and 20% respectively. So the most one is the fertilizer is positive relationship, but found insignificant to production. Okay, so size specific results are there. So this is for the marginal size farms. For the small size farms, inputs like fertilizers, labor, seed and machinery increase the efficiency by 35, 32, 29 and 25% respectively. So it is found that medium-sized farms have consumed fertilizers the most. The extent of elasticity is 80% approximately. So the one side means this is actually the first stage of econometric analysis, at where I did calculate the economic efficiency of the farmers. Okay, then the second stage of empirical analysis, I have used Tobit regression. Tobit regression is used to study the impact of economic freedom index on the economic efficiency. So the impact of economic freedom index on economic efficiency. So it is found that almost all the freedom indexes have positive relationship, but there is labor freedom index that is actually keeping negative relationship with the medium sized farmers. So there is a labor loss effect. Okay, so the literature is actually valid here, and the results were consistent, as 1% increase in the freedom options has improved the economic efficiency respectively for the marginal farmers. And technical freedom is found supportive but insignificant to the efficiency results. Then I use the statistics too, like R squared value states that 60%, 72%, 53 and 42% of the total variation is found by the freedom indexes, and other socioeconomic variables were taken, like age, education and income also, to small, marginal, medium and overall farms respectively. So there are I could show there are 4 models, 1, 2, 3, 4 for the marginal farmers, for the small farmers, for the medium farmers, and this is the common pool, the overall results. Okay. There are implications and suggestions to a lot of things. Actually, I have skipped due to the shortage of time. Thesis has provided several suggestions and the policy implications and recommendations to the government. So it is actually the study actually has created the awareness within the farming community to access the options and opportunity to enhance the productivity and efficiency of the farming sector. And it actually has suggested the authorities to set the provisions for increasing opportunities to the farmers. And there are actually there are steps and policy suggestions given, like through promoting and developing the agriculture cooperatives in the farming sector. So it may enhance the economic freedom in agriculture because cooperative model is designed to improve agri-business efficiency under specific conditions by collective bargaining, economies of scale, membership benefits are there. So directly, the awareness can be spread within the farming community, and the prevalence of economic freedom in agriculture throughout, this is actually required because an investor can fascinated by the rural economies that offer freedom, options, and opportunities better than the other economies. So in this way, the study investigated the implications of rural development policies. There are several sub-components or indicator I could find out in my study. So that are required: farm jobs and non-farm job opportunity for improving the labor freedom, rural wages, employment rate will affect the mobility of the rural landless and migrant families. So it will have a positive effect on the efficiency and growth of the farming sector. That's all from my side. Thank you so much. Thanks the organizing team, SARM. Thanks for giving me the opportunity. Thanks for listening my presentation. Thank you, sir.
Thank you so much, ma'am, for joining and presenting a wonderful presentation.
Thank
Do
you.
we have Dr. Vijay?
Any question?
I guess, ma'am, there's no question because.
Okay.
Is there any?
Yeah, I am here. I am Vijay Kumar. I am here.
Okay. Okay. So, Dr. Vijay Kumar, you can start your presentation after this.
Okay.
Yes. Thank you, ma'am. Thank you for joining. You can take a leave now.
Good evening, good afternoon, everyone.
Can we change the screen, please?
Shall I share the screen? Yes.
Sir, just a second.
Okay.
Yes, sir. Can you please try and share your screen now?
Just a minute. Okay.
Your slides are visible, sir.
Okay. So I can put full screen, this one, slideshow.
Yes, sir.
Okay. So welcome and good afternoon to everyone here. So my presentation is on identification of potential physiological and biochemical stress indicators during thermal stress and its recovery in synthetic prolific and native non-prolific ramps. It is in our semi-arid region in Rajasthan. So prolific ones, I am from ICR Central Sheep and Bull Research Institute, presently working as principal scientist at Arid Region Campus, Bikaner. So that is the Arid region. So briefly, I will give you just a background of what a prolific ram is and what a non-prolific ram is. So the sheep rams which are having the introgression of this fecundity Brula gene, which is responsible for causing more than one fetus or more than one lamb during lambing. So that gene is responsible to cause more than one lamb lambing, multiple lambings in the sheep. So when it is in homozygous condition, it is called as FACB, superscript as BB it is written for Brula, and FACB++, that is homozygous non-carrier of Brula gene. That is our the native adapted sheep, which is Malpura. So we developed this breed, Avishan, we call it, and it is now having a fecundity or say prolificacy of around 1.7, 1.8. So that means if it is 2, it is 100%. That is more than 2 we are getting out of 100. Say it is there, all are giving. So accordingly, we can see 1.7 means at least 80, 90 they are giving more than 2 lambs. So in this, the composition is the gene of Garol, which is actually the homozygous for fecundity Brula gene carrier. Patanwadi and Malpura, these are actually non-carriers. Patanwadi from Gujarat, Malpura from Rajasthan. But to give a higher body weight, it has a very low stature, low height and low body weight, but these are having a high body weight and this has a more of milk. So to give more of milk, and this one, here you can see this is the Malpura ram and this is the Avishan ram with more than some 3 or 4 twins lambs are there. And this is the Avishan ram, Patanwadi ram, and this you can see the Garol, native of West Bengal, very short stature. So to make these lambs have more milk and more higher body weight, we have crossed with these two, Patanwadi and this Malpura with this one. So after this introgression, there is always a chance that there is a adaptability issue to the climate of the arid region. And so how to see that? So we compared both this native lamb and this one, Avishan rams, and we try to expose them to heat stress, extreme heat stress for 4 hours from 10 o'clock in the climate chamber to 2 o'clock, to 2 p.m. Followed by a recovery period because it was in the winter. So we shifted the animals after 4 hours back to the semi-open sheds where these animals were exposed to thermoneutral climate it was, except during early morning or 2 a.m. in the morning, the temperature was low. And we can see here, all these parameters we have recorded. And these were the times where we took all the observations, even we had taken blood and some work is also going on. So here we can see how the temperature was. Say Malpura and Avishan, 2 different days we have done. Because 8 animals here, we cannot take so much rigorous samplings every 4 hours and 2 hours, 1 hour. So 2 different days, but just nearby, 1 day after. So you can see this is the dry bulb, this is the wet bulb temperature, and this was the RH. But from 11 p.m., because 10 a.m., before putting the rams in the climate chamber, we took the recordings. And then after that, every 1 hour, we took the recordings up to 2 p.m. So that now here the temperature was 49, wet bulb was 30 degrees centigrade, humidity was 25. And here, this is the THI, thermal temperature humidity index, which is for sheep calculated by this equation. So it categorizes whether the rams were actually exposed to very high stress or they were to a normal stress or what it was. The condition was extremely stressful. But here these are green shaded, no? These THIs show that these animals were under comfortable zone. Here you can see very well if it is less than 22, it is absence of stress. But 25.6 and above, there is extreme stress. But these are the indices developed by all the European nations or in say temperate regions. And so this world over, we are having only these indices. And we have conducted this one exercise, and there are several exercises conducted because these are not applicable to our animals. So it was one exercise in this field also, but primarily we wanted also to know whether how these ramps will perform. So if you see the respiration rate, this yellow is the Avishan and this is the Malbora. Here we see the rise in the respiration rate is very high in Avishan ramps, the synthetic lamps. It is almost 181, so mean. And here the star shows that with the respective values here, it is significantly higher. But okay, of course, when they were shifted back to the thermoneutral climate, it has declined back. So this declining back means, okay, it has come back to normal means, yes, the plasticity is there and the animal is able to thermoregulate plus also recover. So not much issue that, yes, they were performing, they were able to adjust, but within their limits. Okay. So similarly, we could see the pulse rate also. This also increased during the stressful period. But pulse rate is not a very good indicator of stress. In very extreme stress also, we are not observing much difference even through the normal, except a few. So thermal, this rectal temperature when we saw that was also higher in case, significantly higher in case of the Avishan. Okay, of course, the recovery was also not as good as seen in Malboro. So there is some kind of issues there. So we wanted to then see what was the difference in the wool and the skin temperature. Because when the animals are in cold outside, animals are in chamber heat outside, what the wool is playing a role. The woolly fleece, it is about 1 inch or so, the thickness. Then how much protection or the thermoregulatory mechanism it helps, it contributes. So here we have seen the temperature of the wool surface temperature, and this is the skin surface temperature. But what we want to show here is that was in Malpura, this is in Vishan. Here what we have seen is what was the difference, that is from the wool to skin, the difference was how much. If it is in the morning when the temperature is very low, then we find that this difference is very high, about 14 degrees centigrade. But 49 means the skin temperature is higher. Here we see in the cold, wool is able to maintain a higher skin temperature. Then when the heat stress is there, slowly we find that yes, the wool temperature remains, but the skin temperature declines. So this is one mechanism that wool forms a barrier, some kind of insulation a barrier it forms, and that helps in actually regulating the body core temperature through regulating the skin temperature. In both the breeds, there was not much any significant difference between the breeds, but yes, of course, when the time period was taken into consideration, there were significant differences. So when we saw the hormones, what was the difference in the hormones? So as we all know, cortisol is a very prominent and also a good reliable marker of stress. Everywhere we know this is the generalized marker of stress. We wanted to study this, and there was a difference in Avishan and in Malpura. And this difference was huge because here around 150 and above nanomoles, and here starting around more than 30. But here it did not show any this one. So there was some difference when they were exposed to this one. Some kind of difference is there. When we saw here how the difference is actually, we see here in Malpura, yes, increase is there, decline is also there. But if you see in Avishan, there is a constant increase. But this basal level is, the amplitude itself is very low. Now we are now trying to do this again through more precise systems, so that we can confirm whether it is like this only. Then the other thing is the triiodothyronine T3 hormone. So that is also a stress indicator, many times used for identifying whether the animal is in stress or not. It is a metabolic hormone and plays several roles. So here we see that in Malpura it was very stable until last when the 4 hour of stress was there, then only it increased. And this increase was fluctuating thereafter until next day morning. This was a different behavior. And when we see in Avishan ramps, the increase was very abrupt. It was not here very low in Malpura native. But in the synthetic ones, the increase is there gradually, and higher increase is there. and then decline also. So it is utilized, and then once utilized for stress combat, then again we see that yes, there is no more T3 hormone available. So this is a difference. Similarly, the precursor of T3, the T4, that we see here in case of the Malbora, it is declining because here it should get converted to T3 and utilized for the stress combat. Here what we see here, after 1 to 2 hours and then 3 hour and 4 hour, it is having a higher, lower values because it gets converted to T3. Afterwards, it is in recovery phase, up and down is there. But here in case of the Avishan, we are not able to see any such trend. So there are some issues with this. We have to confirm again. Once breed is stabilized, that comes. Now another hormone, creatine kinase. These are also metabolic enzymes. This is a metabolic enzyme found in liver, kidney, and various other organs, muscles. Primarily, it is in muscles, then heart also. So when so much of stress is there, so much of respiration is increased, so much of exertion is there on the animal, panting is also there. Yes, this hormone, some muscular injury activity, some trauma type of effect is also there. Is that bringing any change? Yes, it is bringing a change. Yes, the increase is there. And in Malpura, as it is adapted one, it comes down. Here we see in case of Avishan that different picture is there. This is higher from here itself. That means there was more stress in this one. The byproduct, the creatinine of that action of creatine kinase, what we see here, there is an increase here. But afterwards, some decline is there when the increase is there during the stress. But in case of Avishan REMS, we could not see this kind of result. But afterwards, we saw, yes, there is a steady level. Then there was triglyceride. These are also mobilized during stress to be utilized for our energy purposes. But here we could see within the first 2 hours itself, this has increased. Mobilization has occurred in the first 2 hours itself. But when we see in Avishan, it is a very gradual and keeps on increasing. That is, it keeps mobilizing even up to night. After about 7 to 8 hours of recovery period, also it has been higher here to provide energy to them. Then there are oxidative stress indices. That is a total oxidant status, a ratio of total oxidant status and total antioxidant status. So in this, we see that in case of here during the stressful period, there is an increase, but it keeps those slight down is there, but it remains until next day morning. But Avishan, when we see, there is a very different picture. This is very higher also, and at 4 hours after that also it is higher only. It has not declined. So this OSI is a ratio of this TOS, but we see here TOS. The end in Malpura and Avishan, see here TOS oxidant status is too high after the stress is over. Land at the last hour of stress, very high oxidant stress, and then remains higher in case of Avishan. But if we see total antioxidant system, when we see the antioxidant system, it is declining. Yes, it will decline because antioxidant system, it is getting utilized, keeps on utilized. But here in case of Avishan, it starts getting depletion only after 2 hours of stress. Until then, it was maintained. Afterwards, it has depleted very low. Then other enzyme, that is lactate dehydrogenase enzyme. So this enzyme also plays a very important role. It is also in muscles, in heart also, and in liver also, and various other organs also. So if there is an injury, there is some kind of stress, these will leak into the blood system. What we could find yet, yes, there was a low, but there was not such leak in case of Malpura. And here in case of Avishan also, there was a decrease. Okay, there was not much leak, but why this decrease has come, we don't know. Afterwards, here it has increased. So it is here also it has increased after the stress one. This we will see this. Other enzymes were the glutamic oxaloacetate transaminase, the serum GOT. But here we could not find any trend. Generally we see, yes, there is if there is so much of stress, liver function, but it is primarily for the liver function. So we could not find any major impact on this one. But in case of Avishan also, there was not much impact. The other liver enzyme, this is specifically for liver. Here we find only at post-stress some change is there. But otherwise also in both Avishan and Malbra, there is a small, no trend is seen. So these enzymes are not much affected. So what we could conclude that, what markers we can, potential markers, which can be the potential markers out of these. We have seen a lot of literature, so many markers, these markers are being used and reported. Why should we spend when such a stress is there? We should only include those markers which are reliable. So we found that cortisol significantly changing, it is reliable, but there is a breed wise basal change which needs to be seen again. T3 and T4, of which T3 can be used as a marker. This is a moderate marker. Then another is creatine kinase. Yes, this can be used as a reliable marker, but no other marker is so reliable as was this one, except this OSI and total antioxidant system. These can be utilized as a marker, moderately reliable. And these are the hormones, except this creatine kinase, none is reliable. So what we have seen is the woolly fleece, physiological indices, of course, they are reliable. Woolly fleece, it facilitates excellent thermoregulation. There is a negative temperature gradient of around 4 degree on the skin during extreme heat stress and a positive gradient of 14 degrees centigrade during extreme cold stress at the skin surface. And these I have already told, these can be used. But the most important finding that yes, this breed needs to further stabilize to the climate for which it has been developed. So this is all from my side, and thank you all very much. Were you able to hear me, or should I stop?
Yes, sir.
Yeah,
Thank you
okay,
so
okay.
much. Yeah.
Okay.
So this was the last talk of the entire session of Agri Vision 2024. So we take a break from here now, and we will soon meet you back with the presentation ceremony. Thank you.
Thank you. Then,
Hello. Hello.
Hello. Hello. Hello. Hello. Hello. Hello. Hello. Hello. Hello. Hello. Hello.
আসিনি তো। মাইক অফ করো। আর মাইক বলো।
Check. 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 Check.
Namaskar, Jai Jagannath. A very good evening to one and all, each and everyone present over here. All the honorable dignitaries, luminaries, all the esteemed scientists, researchers, exhibitors, students, farmers, print and electronic media friends, ladies and gentlemen. A very warm welcome to the valedictory session, the closing ceremony of Agri Vision 2024, the 6th International Conference on Agriculture for Sustainable Development, organized by Society for Agricultural Research and Management in collaboration with ICAR National Rice Research Institute, Cuttack. On the behalf of the core organizing team, I would like to take this opportunity to thank each one of you for your sheer dedication and involvement and active participation to make this conference a successful one. The theme of AgriVision 2024 was economic development through sustainable agricultural practices. Together, we make it possible. Last 3 days were full of knowledge, discoveries, skills, and informations, making the platform of very fruitful one. We have witnessed the intelligence through the insightful plenary sessions, and thus it was a great opportunity. And here we are to celebrate its outcome. To add the cherry on its top, today it is our utmost pleasure to welcome our honorable dignitaries on the dais. I request each one of you to welcome all our honorable dignitaries with a huge round of applause. It is our absolute honor to welcome all our honorable dignitaries on the dais. Let's have a huge round of applause to welcome our honorable chief guest, Dr. Pramod Kumar Sahu, Honorable Director, ICAR Central Institute of Freshwater Aquaculture, Bhubaneswar, Odisha. Our guest of honor, Professor Dr. Prasanajit Mishra, Honorable Dean, Extension Education, Odisha University of Agriculture and Technology, Bhubaneswar. Let us welcome on the stage the Chairman, Organizing Committee, Dr. Amresh Kumar Nayak, Honorable Director, ICAR, National Rice Research Institute, Cuttack. Let us welcome on the stage with a huge round of applause, Honorable Convener, Dr. M.J. Beg, esteemed Principal Scientist and Head of Division, Crop Physiology and Biochemistry, ICAR, National Rice Research Institute, Cuttack. Let us welcome on the stage with a huge round of applause our guide and always he is the man behind AgriVision 2024. And throughout the years, he was with us constantly, the President, Dr. Arup Kumar Mukherjee, Principal Scientist, ICAR, National Rice Research Institute, Cuttack, and the President for SARM. And let us welcome with a huge round of applause. Let's congratulate and please give it off for Program Director, AgriVision 2024, Engineer Dibyanshu Prasad Das. With a louder round of applause, let's welcome the panel of dignitaries on the dais. A very warm welcome, gentlemen. Let us take the name of the Almighty to begin with the valedictory session. I would request all our dignitaries to kindly proceed for the lighting of the lamp. with the very auspicious lighting of the lamp, the valedictory session is officially started. Thank you very much, all the honorable dignitaries, for doing the honor. I request all of you to kindly be seated. Now, may I have the privilege to request Dr. M.J. Baig, esteemed Principal Scientist and Head of Division, Crop Physiology and Biochemistry, ICAR, National Rice Research Institute, Cuttack, to kindly present the welcome address. Please, sir.
Thank you very much. Good evening to all. Honorable Chief Guests, Dr. Pramod Kumar Sahu, Director, ICR SIFA, Bhubaneswar, Professor Dr. Prasanjit Mishra, Dean Extension Education, Odisha University of Agriculture Technology, Bhubaneswar, Chairman Organizing Committee, Dr. Amresh Kumar Naik, our Director, National Rice Institute, Cuttack, President SARM, Arup Kumar Mukherjee, Principal Scientist, ICAR-NRRI, Cuttack, Program Director, Engineer Dibyanshu Prasad Das, esteemed dignitaries, respected guests, scholars, farmers, industry experts, a very warm and gracious good evening to each one of you. It is with great pleasure and immense pride that I stand before you today to welcome each and every one of you to the closing ceremony of the 3-day International Conference of Agriculture for Sustainable Future. Over the past few days, we have witnessed a gathering of brilliant minds, innovative ideas, and collaborative spirits, all covering with the shared commitment to address the pressing challenges facing in agriculture and pave the way to a more sustainable future. Our conference has become a knowledge, fostering a global dialogue that transcends borders, culture, and disciplines. We have been privileged to host distinguished speakers, experts, researchers, policymakers, and industry leaders who have shared their insights, experiences, and cutting-edge research. Together, we have explored the intricate web of challenges and opportunities in the realm of agriculture with a steadfast focus of building a resilient and sustainable future for generations to come. Throughout these 3 days, we have engaged through thought-provoking discussions, collaborative workshops, and networking sessions that have not only expanded our knowledge, but have also forged lasting connections and partnership. The exchange of ideas and the cross-pollinations of expertise are the consents upon that we build in better tomorrow. As we gather here for the closing ceremony, let us reflect on the collective wisdom that has been shared and the seeds of changes that have been planted. The impact of the conference extends far beyond these walls. It resonates in the field, laboratories, and boardrooms across the globe. Our shared commitment to sustainable agriculture is a beacon that will guide us as we navigate the challenges of rapidly changing in the world. I extend my deepest gratitude to our esteemed speakers, sponsors, organizing committee, and each and every participant who has contributed to the success of the conference. Your dedication and passion for creating a sustainable future through agriculture, a true spirit. As you bring the curtains down on the memorable event, let us carry forward the momentum and the spirit of collaborative that has defined these past few days. May the knowledge gained here be the catalyst for transformative action, and may the friendship forged be the thread that wave a global network dedicated to the cause of sustainable agriculture. Thank you for being a part of this remarkable journey. Safe travel to those returning home, and may the seeds of change we have sown together blossom in future abundance and sustainability. Thank you. Jai Hind.
Thank you very much, sir. That was absolutely a sum of these 3 days. Thank you for presenting a warm welcome address. Now, may I have the privilege to request our guest of honor, Professor Dr. Prasanajit Mishra, Honorable Dean Extension Education, Odisha University of Agriculture and Technology, to kindly address the August gathering. Please, sir.
Good afternoon, everybody. Esteemed Director Sifa, Professor Sahu, Director NRRI Cuttack, Dr. Naik, the Secretary, Organizing Committee, Dr. Beg, Dr. Mukherjee, Chairman of the SAM, and Mr. Dibanshu Das, who is the key person behind the organization of this international conference. I congratulate the organizers and the entire team of ICAR-NRRI Cuttack with the able leadership of A.K. Naik, sir. This conference has been organized very successfully. Just we heard about the 3 days program from Dr. Beg. The organizing committee has rightly chosen the topic sustainable agriculture development. Since long, we have been discussing about the development only. Grow more, grow more. This was the slogan earlier. To feed the ever-increasing population, there is demand for growing more and more from the limited resources. The land is limited, the water is limited. However, the population is increasing. There is an estimate from 1.42 billion. By 2050, we'll be reaching 1.6 billion. And globally, our population will be 9.8 billion. And the present trend shows that it will increase and increase till the end of this century. There will be some relaxation at the end of this century when the population growth rate will be 0. But till then, we have to produce more and more from the limited resources. So coming to our country, where 60% of population depends on rice, there is more pressure on my friend Dr. Naik and his team to develop new varieties, new production technology, new post-harvest management technology, besides horticultural crops, besides other pulses, oil seeds, and many more. So sustainable development is the need of the hour, and we are all, the scientists, the students, and the policymakers are concerned for that. Coming to first, seed system. Sustainable, we are now talking about sustainable seed system. Previously, farmers used to grow his own seed. Subsequently, many agencies came for Odisha. Few years back, the farmers were totally dependent on the state government for supply of quality seeds. If you talk about the paddy, nowadays about 4 lakh ton of seeds is being sold in the market. 4-5 years back, the entire 4 lakh quintals of seed has been sold by the Odisha Seed Corporation. But you see last year figure, it reduced to 2 lakh only, less than 2 lakh. The private players now come up in a big way. They are selling 2 lakh quintals of seed in Odisha nowadays. And very rapidly, you will not astonished if it will, the government system of supplying the paddy seeds will diminish and the entire market will be captured by the private agencies. What is happening in case of hybrid seeds of vegetables? So there is need for sharing responsibility among the government, the researchers like university, ICAR institutes. They have to involve themselves more and more with the production of developing high quality seeds, and government role will be restricted to its enforcement, how the quality seeds is produced, how it is supplied. There is no spurious seed in the system. And private players, private organizations are welcome to take the varieties to the farmers' field in any way with the government system, within the government system. Similarly, in case of nutrition, we talk about the sustainable soil health management. It was not previously a talking point. After Green Revolution, use of high quantity of pesticides and fertilizer, now we have deteriorated our soil. Even in our student time, we have been hearing about the compost, different type of compost. But till now, the success rate is very negligible. That's why the use of organics in the field, particularly in rice field, is very less. Though some quantity of FYM and compost is available with the farmers, they will prefer it to apply in vegetables or high value crops rather than applying to the rice field. Nowadays, this stubble burning is a problem. Now it came to Odisha, it was not heard in our state. Now stubble burning is an issue in major rice growing areas of our state. How to manage that? Use of decomposers. NRRI has developed a decomposer. ICAR-NRRI has also developed decomposers. So many companies should come forward how to take it to a commercial scale, how to manage the rice straw in the field. Similarly, weed management. We have been using the herbicides in a large scale. Fortunately, Odisha is not a state where herbicide is used in large quantities. The percentage is very low. But due to use of pesticides in wheat and rice wheat system, the weed flora has also changed and herbicide resistance has developed. Similarly, in case of pesticides, also pesticide resistance is an issue to be discussed by scientists and students like this forum, how to overcome those issues and make the environment a better environment for our future generations. There is a greater role of extension scientists because I am leading the 31 KVKs of our state. Definitely, I will express concern the way it is being transferred to the farmers. There is a greater role of this extension machineries, particularly the KVK scientist as well as extension officer of state government, to take this new technology to the farmers' field. Now, it is not a single farmer. Many self-help groups, many farmers producer organizations are come up in this field. They are involved in production, post-production marketing. So it is easier now to transfer a technology to the masses. We have been giving different technology to the farmers through different media. Particularly KBK is involved through training, research, training, demonstration, awareness program. But our government of Odisha has very innovatively developed a system in which every farmer, 60 lakh farmers are given messages on their crops, advisories twice a week. Twice a week, a message is going to their farmers' mobile. 60 lakh farmers. It is a huge achievement from the government of Odisha. Similarly, WhatsApp groups, every KVK scientist maintain in their district WhatsApp group in which they transmit good technology, not only the information through leaflet, booklet, also success stories through farmers' success story videos also they share among their farmers group. And also government of Odisha has a very robust system of technology transfer in which every day morning by 9 a.m. a message is given, video and leaflets is given by the department to the chief district agriculture officers, chief district horticulture officers. District heads are given every day a WhatsApp message with this by 9 a.m. And there is such a mechanism, it transmits to the farmers within 1 hour. The district level officers transmit it to the block level officers to their village level workers, and each village level worker of horticulture, agriculture and soil conservation department, they are maintaining a group of 200, 300 farmers in their WhatsApp, and immediately by 1 hour from the state government to the farmers, the message reaches. So very robust mechanism there. So my concern is, we know what was happening. We know this agriculture situation pre-independence era. For giving food to our population, increasing population, we ran for producing more and more from the limited resources. We know there is a day our rainfall is so high in India, particularly in Odisha. We never felt earlier water stress during the Kharif season. But you see, in the last 5 years, either the rainfall is reached Odisha, monsoon reached Odisha 15 days, 20 days, 1 month later, disappears sometimes 15 days, 20 days earlier. Sometimes in between the season, Kharif season also, there is dry spell for more than 15, 20 days. So the rice crop is a resilient crop. However, in many cases, we have been observing the productivity is decreasing due to this type of climate variations. So weed population, irrigation, water is a stress, climate change, so many things these are all challenges for us. So in coming days, how we will face the challenges and make our farmers aware with their alternative, how to meet it. This is the real challenge for all of us sitting here. So I hope with all collaboration, cooperation among all the stakeholders, the benefit of this sustainable development activities reaches the farmers in a faster way. Thank you all.
Thank you very much, sir, for the insightful speech. Thank you very much for sharing the real-time data and for talking on the current need to bring a sustainable change. Ladies and gentlemen, each day on this platform, we are getting to have the opportunity to felicitate some of the very renowned and recognized personalities, organizations. Likewise, today also, we are very much thankful to some of the personalities for being a part of this international conference. Now it is the time to give away the awards. May I request all our honorable dignitaries to kindly do the honor of giving the awards. Ladies and gentlemen, this category of award is an utmost honor. The award is Padma Shri Dr. Ajay Parida Memorial Lifetime Achievement Award. I would request each one of you to have a huge round of applause, and with a standing ovation, let us welcome on the stage Dr. Prasoon Mukherjee, Honorable former Head of Nuclear Agriculture and Biotechnology Division, BRC, Mumbai. I would request her to kindly come up on the stage. I would request Dr. Mukherjee to receive the award on his behalf. A huge round of applause. Let's congratulate Dr. Prasoon Mukherjee on getting the Padma Shri Dr. Ajay Parida Memorial Lifetime Achievement Award. Now it is our honor to give away the award of Distinguished Scientist Award, and the award goes to Dr. Pramod Kumar Sahu, Honorable Director, ICAR Central Institute of Freshwater and Aquaculture, Bhubaneswar, Odisha. We congratulate you, sir. Could we have a huge round of applause? We are very much thankful to you, sir, for your valuable time, and we are looking forward for his talk as well. Moving ahead, now it is the time to give away few academic awards. I would congratulate all the winners in these categories with a huge round of applause. Let's congratulate all the winners, and let's welcome for the academic awards Dr. Vidyashri Bharati from ICAR CIFE Mumbai for the Women Researcher Award. In the category of Women Researcher Award, let's welcome Dr. Vidyashri Bharati. Next up in this category, we have Dr. Agri from North State University. I request in the category of Women Researcher Award, let's welcome Dr. Arlene Agri on the stage with a huge round of applause. Let's welcome her on the stage from the University of Camarines, North State University. We congratulate you, ma'am. Thank you very much for your active participation in 6th International Agri Vision 2024. Let us have a huge round of applause to congratulate her. Being awarded in the category of Women Researcher. Give it up for Dr. Arlene Agri, everyone. In the category of Women Researcher Award, we are having 3 ladies and the women researchers. Congratulations. Moving on, let's welcome on the stage Dr. Tanuja Ace from ICAR CIWA Bhubaneswar. I request ma'am to kindly join us on the stage to receive the award. Let's give it up for Dr. Tanuja Ace, ladies and gentlemen. In the category of Women Researcher, the award goes to Dr. Tanuja Ace. Congratulations, ma'am. Thank you very much. The next category is Research Wizard Award. And in this category, let's welcome on the stage Motisi Chakrabarti. She is not here. Griffith University, Nathan campus, Australia. She was there with us for the plenary session. We have in this category Ramya KR, ICAR from ICAR IARI New Delhi, and Ankala Sai Kishore. We congratulate 3 of them. Next category is Young Scientist Award, and the award goes to Dr. Sirigredi Sivajoti from College of Veterinary Science. Let's welcome her. Let's welcome Madam on the stage with a huge round of applause. We have a huge round of applause, ladies and gentlemen. Dr. Sirigi Reddy Sivajoti. We congratulate you, ma'am. Can I see some hands up in the air to congratulate her? In the category of young scientist, she is getting an award. Congratulations, ma'am. With a huge round of applause, let's welcome on the stage Dr. Christelle Ledrate from Bio Reassociation, India. I request sir to kindly join us on the stage. Next up, in this category, we have Dr. Surjay Kumar Saikia. Dr. Surjay Kumar Saikia on the stage, please. From Viswa Bharati University, Santaniketan. We congratulate you, sir. Thank you very much for being a part of Agri Vision 2024. The award of Young Scientist goes to Dr. Surya Kumar Saikia. Can I see some hands up in there? Come on, ladies and gentlemen. Congratulations. The next category is Shining Star. And the award goes to Dr. Ashish Kumar Awasthi from Maharshi University of Information and Technology, Lucknow. In this category, we have Professor Johnson Edinson. I request sir to kindly join us on the stage. A huge round of applause for him. The most active participant in these 3 days. Let's give it up for him, everyone. A huge round of applause to congratulate for the category of Shining Star. We congratulate you, sir. In this category, let's welcome Dr. Hare Krishna Swami. Dr. Hare Krishna Swami on the stage, please. From Botanical Survey of India, ERC, Shillong. Congratulations. We had excellent speakers from the Young Research Forum, and the category of Best Oral Presentation will be presented now. And in this category, let's welcome Rupalin Jena from ICAR-NRRI, Cuttack. With a huge round of applause, let's congratulate all the best performers in the oral presentation. Rupalin Jena from ICAR-NRRI, Cuttack. A huge round of applause. Followed by Ms. Swagatika Das, also from NRRI, Cuttack. Congratulations. The budding talents of ICAR-NRRI in the Young Researcher Forum. Ms. Swagatika Das on the stage, please. Followed by Motasi Chakrabarti, Sakshi Gumbar, and Pujashri Mishra for the award of Best Oral Presentation. Rupalin Jena, Swagatika Das, Motasi Chakrabarti, Sakshi Gumbar, and Pujashri Mishra. Next is for Best Poster Award. And let's welcome Ms. Anupama Kar on the stage from State Bank Institute of Rural Banking, Hyderabad. Let's give it up for her. Ms. Anupama Kar on the stage, please. Yesterday, bestest of posters on the theme was presented by the participants. And here we present some of the best awards to the best participants in this category for Best Poster Award. Ms. Anupama Kar, followed by Ms. Soumya Sefalika D, Ms. Soumya Shri Purohit, Soumya Surabhita Mishra, Aishwarya Priyadarshini, and Subhra Parija. I would request the winners to kindly come up on the stage to receive the award. Ms. Soumya Sefalika D, Ms. Soumya Shri Purohit, Soumya Surabhita Mishra, Aishwarya Priyadarshini, Subhra Parija. Could we have a huge round of applause to congratulate all the winners in the poster presentation? Congratulations to all of you. Moving on with a huge round of applause, we would welcome our exhibitors on the stage for the active participation in AgriVision 2024. Let us felicitate all the exhibitors. I would request the team choir board to kindly be on the stage to receive the award. Let's have a huge round of applause to welcome them and felicitate them. Team choir board on the stage, please. We are very much thankful to ICAR National Rice Research Institution for the support, and also I request the team to kindly be on stage to receive the award. ICAR-NRRI Cuttack. Next up, Botanical Survey of India, Kolkata. Let's give it up for them. Let's give it up for all the exhibitors. Choir board, ICAR-NRRI Cuttack, Botanical Survey of India. Next up, India Post. Followed by Fisheries Survey of India, Vizag. Can I see some hands up in the air? I hope you all have visited the stall areas and have seen the wonderful presentation over there. Give it up for India Post, everyone. Next up, Team Fisheries Survey of India, Vizag. All these 3 days, they made the conference an interactive one, and the stall area was absolutely wonderful with a kind of unique presentation over there of the samples of the products. A round of applause for Fisheries Survey of India. Could we have on the stage CCRS Ministry of Ayush, Government of India. A huge round of applause to congratulate CCRS Ministry of Ayush, Government of India. Now I would welcome on the stage Spices Board of India. Let us give it off for Spices Board of India. Followed by Odisha University of Agriculture and Technology. We congratulate the entire team of Spices Board of India for the active participation. Odisha University of Agriculture and Technology, I would request the team to kindly be on the stage. A big round of applause for the entire team. Not only this year, every year, they are actively participating in AgriVision and the kind of uniqueness they have. And OUAT stands for something different and absolutely an international platform as well. Next up, let's welcome team Kirfco on the stage. Team Kirfco on the stage, please. Followed by ICAR CIFE Mumbai. Team Kirfco on the stage, please. With a round of applause, let's welcome the team on the stage. Followed by ICAR CIFE Mumbai. Let's have continuous round of applause to congratulate and thank all the exhibitors for the kind of participation and relentless effort towards AgriVision 2024. ICAR CIFE Mumbai on the stage, please. Moving on, the team of Central Warehousing Corporation, CWC, on the stage, please. Team CWC on the stage, please. A round of applause for them, followed by Centurion University of Technology and Management. Please welcome our next exhibitor, ICAR CRI Bhubaneswar, with a round of applause. Team ICAR CIFA Bhubaneswar on the stage, please. Could you have a round of applause for ICAR CIFA? Team CIFA, let's give it up for them. A huge round of applause. Absolutely commendable work in the field of fishery and aquaculture. Next up, let's welcome ICAR IIHR CHES Bhubaneswar. Give it up for CHES Bhubaneswar. Now let's welcome the team ICAR CIWA Bhubaneswar on the stage, please, followed by Coconut Development Board, CDB on the stage, followed by National Medicinal Plant Board, NMPB on the stage, please. Team National Medicinal Plant Board, NMPB, on the stage, please. Next up, let's welcome Team OMFED on the stage. A round of applause for Coconut Development Board, CDB. Thank you very much for the active participation. Next up, please, Team OMFED be on stage to receive the award. Next up, please welcome on the stage CCBF Chipilima Sambalpur, followed by IMD Regional Center Bhubaneswar. We talk for IMD Regional Center Bhubaneswar. Once again, I would like to thank and congratulate our exhibitors, COIR Board, ICAR-NRRI Cuttack, Botanical Survey of India, Kolkata, India Post, Fisheries Survey of India, Vizag, CCRS, Ministry of Ayush, Government of India, Spices Board of India, Odisha University of Agriculture and Technology, KIRFCO, ICAR, CIFE Mumbai, Central Water Housing Corporation, Central University of Technology and Management, ICAR, CTCRA Bhubaneswar, ICAR, CIFA Bhubaneswar, ICAR, CIW Bhubaneswar, Coconut Development Board, National Medicinal Plant Board, OMFED, CCBF Chipilima, IMD Regional Center Bhubaneswar. A round of applause for the exhibitors, everyone. Now we will take this opportunity to felicitate Dr. A.K. Panda from CCBF Chipilima, Honorable Director, for chairing the session. Dr. A.K. Panda from Kyari, Bhubaneswar. I request her to kindly come up on the stage to receive the award. Thank you very much, sir, for participation and your guidance for Agri Vision 2024. Give it up for Dr. A.K. Panda from Kyari, Bhubaneswar. Now I would request all our honorable dignitaries to felicitate Honorable Director, CCBF Chipilima. Let's welcome Dr. Sarada Sahu, Honorable Director, CCBF Chipilima. A round of applause for Dr. Sahu, everyone. Now we will take this opportunity to felicitate our collaborator, ICAR National Rice Research Institute. I would request Engineer Dibyansu Prasad Das to kindly give away the award. I request the Honorable Director, NRRI, to receive the award. Thank you very much for the constant support during these 3 days. A huge round of applause for ICAR-NRRI, everyone. Thank you very much, all the honorable dignitaries, for doing the honor. Now, I would request Engineer Dibyanshu Prasad Das and our President, SARM, Dr. Arup Kumar Mukherjee, to felicitate our honorable dignitaries with a token of gratitude. I request both of them to felicitate our chief guest, Dr. Pramod Kumar Sahu, Honorable Director, ICAR Central Institute of Freshwater and Aquaculture, Bhubaneswar. We are very much thankful to him for his valuable time. And also we are waiting for the awaited moment to hear him out. It is an honor to felicitate Dr. Pramod Kumar Sahu, Honorable Director, ICAR SIFA, Bhubaneswar, Odisha. The gentlemen, please give it up for Dr. Sahu, everyone. Now I would request Engineer Dibanshu Prasad Das and Dr. Mukherjee to felicitate our guest of honor, Professor Dr. Prasanjit Mishra. Honorable Dean, Extension Education, Odisha University of Agriculture and Technology, Bhubaneswar. We are very much thankful to you, sir. Thank you very much, all the honorable dignitaries, for doing the honor of giving away the awards. Now I request all of you to please be seated. Now is the most awaited moment. May I have the privilege to request our chief guest, Dr. Pramod Kumar Sahu, Honorable Director, ICAR Central Institute of Freshwater and Aquaculture, Bhubaneswar, Odisha, to kindly address the August gathering. Please, sir.
Good afternoon, everybody. All our esteemed guests on the dais, Professor Dr. Prasanjit Mishra, Dean Extension Education OUAT, my colleague director and chairman of the organizing committee, Dr. A.K. Naik from NRI, Cuttack, esteemed convener of this international conference, Dr. M.J. Beg Sahab, Program Director, Mr. Dibanshu Prasad Das, and President of the Society, Dr. Arup Mukherjee ji, all our esteemed delegates from different parts of the world and India, press and electronic media, students, farmer friends, ladies and gentlemen. Once again, good afternoon and welcome to this particular validatory session. First of all, let me congratulate ICAR-NRRI Cuttack and SARM for making this event a very successful one at the outset. Once again, I thank the whole team of ICAR-NRRI and SARM for making this event for everybody a successful one. I think most of us have received it. Mr. Das has awarded all of us, those who are presented in this hall. So congratulations to all the awardees. I am thankful to the society as well as NRI for giving this opportunity to me to be with us in this particular afternoon or evening to share a little whatever I know about the agricultural science and particularly with regards to this particular international conference. First of all, I congratulate one and all once again those who have joined for the cause of making the agriculture sustainable and this particular noble profession, to fulfill the cause of this noble profession, whether you are a scientist, whether you are a farmer, whether you are any kind of stakeholder, those who have contributing to make this particular system a sustainable one. And all you know, the country is providing or achieving the food security, which is ultimately leading to economic security and also leading to societal security in terms of health, in terms of education, in terms of employment opportunity. And we have achieved a lot in terms of contributing to the societal growth, providing employment opportunity through agriculture. But as Dr. Mishra mentioned, we have several challenges ahead. Perhaps the last point what he mentioned, the climate, which is one of the theme area of this particular conference, which have been debated a lot, might be debated a lot in this particular conference, is one of the biggest challenges for the coming years. So as a scientist, as a researchers, we have a lot of painstaking path ahead to address this particular challenge of climate change. Whether in terms of addressing newer or novel technologies, what he mentioned about resilient system of seed production system, seed management system, till full value chain reaching out to the stakeholders. So throughout the value chain of the, whether it is crop, whether it is animal, whether it is fish, all the food production system suffers from the same challenge, and the challenge need to be addressed in all the segments, in terms of bringing out novel technologies, newer technologies to the system so that we all together can address this particular mega challenge. And the theme, the part of the theme of this particular event, together we make it possible. Perhaps it is well thought theme which has been given, and it might have been debated a lot in this particular international event. Just, I want to share my experience. Just a day before yesterday, I had been to one of our farmers' field, which is doing aquaculture. Along with aquaculture, he is doing a lot of integrated farming in one of the remotest place of Nabarangpur districts near Umarkot. I was surprised to see he has done 1 acre of tomato farming. As he mentioned, each one plant of tomato, he has already harvested within 3 months around 18 to 20 kg average. He is not that much educated farmer. He is doing integrated farming, but it is really worth seeing. That is also in a remotest corner where you cannot reach by vehicle to his farm. And he is not, maybe his earlier generations, they have been migrated from different parts of Bangladesh. They have been settled in Koraput, earlier Koraput district. But in the nearby area of that farm, there are many tribal farmers, huge amount of land holdings, and absolutely you can see in the same ecological condition, same environment, nothing is being happening. So why I mean to say here exactly, we have the technology, we have the resources, but the wide variety or divergence of the scenario in the same ecological niche, same environment, which needs to be addressed by all the stakeholders, those who are sitting here. For example, I should not say, it's not my subject, but still then, this segment in the horticulture segment in India, which is almost contributing more than 30% of the production of agriculture production from the 10% of the area. There was a theme on sustainable horticulture. Hopefully it might have been debated well, but we have several challenges in the segment also. So it is high time, within very short time, we need to achieve more and more what Dr. Mishra mentioned, probably through bringing or importing newer or novel technologies, and also building up skills, which is very crucial for any of the technology. Just I'm citing the case of horticulture, but it is applicable to all the agricultural technologies, which is very crucial. And nowadays, slowly, we are more moving towards artificial intelligence, use of drones and other technologies, and probably our course curriculum should be, from the beginning, it should be strengthened with this newer technology to address. And many of these technologies, which are age-old technologies, you can say, which has not been explored. So a lot of efforts needs to be given, research efforts needs to be given, like perennial lesser explored crops or the plants, which has not been thoroughly studied. So we need to utilize our recent technologies, any kind of omics technology, to genome editing, to develop resistant varieties, whether it is plant, whether it is horticulture crops, whether it is animal or fish. Slowly, we have to utilize the latest generation technologies to address the challenges, to make the system more resilient, to bring the productivity, to increase the productivity, so that we can achieve self-sufficiency. You might have heard the term blue food, the food which we are getting from sea or ocean or lakes or rivers. And there is a huge demand on this blue food in the future because it is one of the most traded food commodity. By 2050, maybe we have to double the production from blue food. But if you see the scenario in case of India or any other country throughout the globe, the capture production from sea or marine environment, it's not growing that much. It is almost stable. But to address the nutritional security, now we are not talking of not only nutritional security, we are talking not only food security, nutritional security, we are talking about the quality of nutrition that we are going to get in the future. That is a big challenge. Everybody is thinking that how I will get organic product, less pesticide residue. It should be applied with minimum fertilizer or no fertilizer. So that's what we are, the next generation, they are thinking about the quality. So the quality will remain a bigger challenge in the coming future. Again, one of the important challenges the whole globe is facing, that is antimicrobial resistance and One Health. One Health means the resistance pattern which the microbes are developing in our food chain, it is going to the human, whether it is animal, whether it is plant or the environment. So it is totally, the full circle is involved, which is creating the resistant microbes, which is very difficult to handle. Millions of deaths, which is happening every year throughout the globe because of this antimicrobial resistance. Now it is being widely debated, and perhaps this One Health, the management approach, we all should be concerned how to address this particular AMR through this One Health approach. Like this, we have several challenges. The way we grow, we have to face several challenges. So what we should do? Now, our approach perhaps should be much more integrated instead of working in isolation. Earlier, we are thinking of, for example, agronomy will work on agronomy, plant pathology will work on plant pathology aspect. Now we have joined within the plant sector. Now we are thinking of how plant and animal as well as aquaculture sector should be joined, or human sector will join together. So it will be a comprehensive approach is required to address the future. So I hope this particular international conference will provide a good number of ideas, solutions, which will be ultimately reflected in terms of proceedings. But I request the organizing committees to take up these proceedings and major action points to the policy level so that it should be addressed. It should not end up with the proceedings. Further, I thank Mr. Das and Dr. Naik for organizing such an international event in this particular campus. And I hope also in future, all the organizations in and around Bhubaneswar and Cuttack or in Odisha, they should join hands together in future to bring into all the heterogeneous group together to discuss on different aspects and challenges and to address the challenges of the future. With these words, I thank once again Naik Sahab and Das Sahab and the whole team for giving me this opportunity to be here with you, and particularly Dr. Das for felicitating me on this occasion. Thank you once again.
Thank you very much, sir, for putting up a resounding note on the very reliable topic and absolutely relatable. Moving on, now I have the privilege to request Dr. Amresh Kumar Nayak, Honorable Director, ICAR-NRRI Cuttack, to kindly share few insightful words, please, sir.
A very good evening to all of you. Distinguished chief guest of the valedictory function of the international seminar, Dr. Pramod Kumar Sahu, Director, ICAR Central Institute of Freshwater Aquaculture. Honorable guest of honor, Dr. Prasanjit Mesra, my good friend, Dean Extension, Odisha University of Agriculture Technology. My colleague, Dr. MJ Beg, Head Physiology and Biochemistry and convener of the international seminar. Dr. Arup Mukherjee, President of the Society. Engineer Divanshu Prasad Das, Program Director, Agri Vision. Distinguished delegates, ladies and gentlemen, we have come to end of this 3 days international seminar. Theme was sustainable agriculture, and the topic is so important. So last year and this year, you will be finding dozens of seminars having words that we have coined in this seminar, title of the seminar. The topic is so important. That means this particular seminar is not end in itself. Rather, this is the means to achieve the objective that is enshrined in the title of the international seminar. This seminar provided a platform to young researchers, students to come together and share their findings and thoughts that can help finding the most important solution the mankind is facing today. Earlier, when we are having a seminar on the sectoral aspects, the scientists of a particular discipline or related disciplines, they were gathering and discussing and deliberating on the issues that is relevant to that particular subject. But the title of this seminar is so vast that required the multiple disciplines to gather and share their findings, thoughts, and challenge the established knowledge so that the new knowledge can come out, new insight can come out, so that pave the way for solving some of the problem. And as I rightly pointed out my previous speaker, this require multidisciplinary research, multidisciplinary interaction, sometimes transdisciplinary research. Earlier, some of the research institute, universities, they are not tuned to work with the transdisciplinary approach. Of course, within the university, we were working in a multidisciplinary fashion, but solving a problem of the global magnitude, that is a sustainable agriculture, that is associated with the 17 Sustainable Development Goal, that requires a multidisciplinary approach. And this seminar has provided a platform or agriculture, horticulture, dairy, animal husbandry, poultry, other discipline, people they gathered, they share their findings, their thoughts, establish contacts, good friends that will help in future making further collaborations. So that is the, I think, in terms we told that this is a successful event. I see the success in these terms. But every event, everything we organize, there are some flip side also. So what are the failure side that we want to tell you? Today's validatory function should have the rapporteur's report. That is glaringly missing. Of course, we are being the organizer, we are owning the responsibility, and I hope the rapporteurs, they will quickly submit their report, and the organizers, they will compile the report, and the report need to be circulated among the participants and other stakeholders. So that is one glaring lacking we are having, and we own up that responsibility. And other glaring thing which I need to point out, that part of our failure, that is the kind of participation in the validatory function. I do not know. I think the participants, including me and all the participants in front of me, we are sitting because we cannot escape our responsibility, because we have been given this responsibility to finish. Besides the people, those who have received awards, and the dignitaries on the dais, those who are there to perform a particular function assigned to them by name and designation, I am not seeing any other person here. I don't know, we will have to again introspect why these participants, it should provide a euphoria, enthusiasm among the young researchers and the scientists to be here, because sometimes it may happen the scientist may not learn something new from the discourse and deliberation because he is knowing the things, but it is his honest responsibility. So whatever the knowledge he is having, he needs to pass on to the next generation or other scientists. So that responsibility he has to accomplish. For that reason, he has to be, or she has to be here. So that is also part of lacking, which I am observing here. And that needs a very thorough introspection among us, why the interest of young researchers and researchers are declining for this kind of activity. We know whenever we do research, get some observations, and find something new, we want to tell the whole world, Eureka, Eureka. The Archimedes discovered the buoyancy, and he came out of the bathtub naked and told, I got this excellent thing. That is the way of expressing the euphoria and the scientific discovery. And the structured way through which we share our knowledge to the rest of the world and subject ourselves to the greatest criticism in seminar and conference, because other discipline people, how they are seeing our work, what kind of lacking was there in our planning, in our methodology, in our interpretations. So we improve upon that. So this seminar provides a platform for this kind of things. But if we are losing interest on that, that means something seriously lacking in our discourse and deliberations and the way we are seeing the seminar and conferences. So that needs a great introspection. But as I pointed out, the similar kind of, with the same title, the conference will go on, because the problem which is the theme of this international seminar is so dynamic. The thing which is sustainable today, it will be unsustainable tomorrow. So this kind of seminar or conference is valid now, and times to come in future also, the same title will be there. And we need to work, introspect, learn from each other, add some to the knowledge which is there, and challenge the existing knowledge which our predecessor and we have developed also. So if you do not have ability to challenge our own knowledge which you developed and discovered yesterday, then we stop our growth. So with this, I will again welcome and congratulate all of you, those who have got an award. And that's a great inspiration for a scientist, a worker who is working in the field. That's some sort of recognition. And when you will go back to your institute, then you will have to share the things which you have learned here, the idea which you learn here, and that will fertilize the minds of the researchers, those who are working in your organization. And in future, many new things, new idea that will come out for solving the problem. And I thank the SARM. They are continuously trying to provide this kind of platform to the researchers working with different institutes, and slowly they will evolve in the future year, and this platform and this society will be having greater collaboration with the institute, research institute here and the other states. And in future, we will be seeing similar kind of activities, and we can get benefit out of it. With this, I thank you, thank all of you, and thank our chief guest of the today's function. Despite his busy schedule, he could come here and address us. And my friend Dr. Prasenjit Misra, Sunday time, because of his busy schedule, he came here and spared a lot of time. We are very much thankful, sir. And with this, I thank all of you. Jai Hind.
Thank you very much, sir. After the resounding speech by Dr. Nayak, now may I request our Honorable President, SARM, Dr. Arup Kumar Mukherjee, to kindly share his closing remarks. Please, sir.
Respected chief guest of today's ceremony, and our guest of honor, and our honorable director, Dr. A.K. Nayak, convener of the seminar, Dr. M.J. Beg, and Dipanshu Babu, and all the dignitaries of the dais, that any good thing has to come to an end. And now it is the end of the seminar, 3 days international seminar. But this is not the end. Why I'm telling? Because I am sure that from tomorrow onwards, Dipanshu Babu will tell, sir, what will be the next topic and where to organize it. So our new journey will start from tomorrow. So this topic is agriculture for sustainable development. Earlier it was sustainable agriculture, but now it has been converted to agriculture to sustainable development. So how agriculture will be used for sustainable development. And last 3 days deliberation, what I noticed that the works are going on in right track, because different topics are discussed, and we heard the lecture from horticulture, then crop production technologies, crop protection technologies, crop improvement technologies, and what are the latest improvement or in climate resilient agriculture, what research is going on, and how biotechnological tools can be of help. But I'm sorry what our directors have told. I really agree with that, because when I reached in the morning, then I saw almost nobody was there, except few of the organizers. But I expected that at least students should be present here in large number. Why I'm telling? Because you know that students are there who are the budding scientists. They have to learn lots of things. And today morning, we got 2 excellent lectures. One is from Bark, that how a basic research can be converted to applied, and then how biotechnological tools can be used for crop improvement, which is climate resilient. How to get the climate resilient products. So I don't want to linger on, and I don't want to take your time. So thank you all, and thank you all the sponsors. Thank you. And another thing I should tell, that symposium was highly successful because of the 2 persons. One is our honorable beloved director, because I know when NRRI has taken the responsibility, there will be a stamp of NRRI, which will be successful, there's no doubt. And another person is Dr. M.J. Beg, whom I know from my PhD time, because we are together. and whatever responsibility he takes, he makes it successful. And of course, Dipanshu Babu is behind everything. And so thank you all for your cooperation. And I hope that you have enjoyed your stay here. And again, I am telling that please participate in the same manner as you participated this year, next year, and in large number, and try to be a member of the Society of Agriculture Research Management so that we can come out soon with good publication in future. And welcome you again for the next seminar, and thank you all.
Thank you very much, Dr. Mukherjee, for your constant contribution and relentless effort towards AgriVision 2024. Each year, you are there for us. Before we draw a conclusion to the event, I would request team SBI to be present on the stage. Our diamond sponsors, give it up for SBI, everyone. A huge round of applause for the entire team. Let's hear it out for State Bank of India. Let's have a huge round of applause. We are very much thankful to SBI for being the diamond sponsors in the AgriVision 2024. Could you have a huge round of applause? Thank you very much. Thank you, dignitaries, for doing the honor. Now, I would request Engineer Dibanshu Prasad Das, esteemed Program Director, AgriVision 2024, to kindly propose the vote of thanks.
Jai Jagannath. So finally, we reached the validity session of AgriVision 2024, the 6th International Conference on Agriculture for Sustainable Development. I am really indebtful to our collaborator, ICAR National Rice Research Institute. I'm really thankful to Director Sir and Beg Sir, who really helped me in many ways, because there are many things which were lacking in this. Like we were organizing the conferences, but there are certain things which we can say a craftsman or a guru can only suggest you. So I felt that when I was advised by them. So there were regular meetings, and Director Sir and Beg Sir, they consistently looked into each and every topic diligently and asked me that Dibyanshu, that you should not have a loophole in that. So I'm really thankful to both of them who have really helped me a lot in organizing this conference. I'm thankful to our organizing committee members and our society members who have really helped us in organizing this successful event. I'm thankful to our chief guest for the valedictory session, Dr. Pramod Kumar Sahu sir, Honorable Director, ICAR Central Institute of Freshwater Aquaculture, who made himself available within his busy schedule. I'm really thankful to our guest of honor for the valedictory session, Professor Dr. Prashanjit Misra, Sir, Dean Extension Education, OUIT Bhubaneswar. I'm really thankful because he helped me in many ways. Because in a very late moment, I requested, sir, please help me with few of the farmers. At least they should come and visit. And immediately, sir, arranged that. And today morning, we had a beautiful farmers interaction session. And all our exhibitors, they interacted very nicely with the farmers. Really thankful to you, sir. I'm thankful to all our delegates from different universities of India and abroad who joined us physically and virtually and shared their research works, their findings, and enlightened this August gathering. I'm really thankful because of you people, the different students and researchers of Odisha, India, and abroad, they got enlightened. I'm really thankful to all our delegates, and it's my humble and sincere request to do participate with us in the next upcoming versions of AgriVision. I'm thankful to our sponsors, our diamond sponsor, State Bank of India, our silver sponsor, Tata Agrico, not in order, IREDA, Coir Board, National Rice Research Institute, Botanical Survey of India, India Post, Fisheries Survey of India, Ministry of Ayush, National Medicinal Plant Board, National Fishery Development Board, Spices Board of India, NALCO, Annapurna Finance, OUAT, CRIPCO, CIFE, Central Institute of Fisheries Education, Central Warehousing Corporation, Centurion University, Central Marine Fisheries Research Institute, Central Tuber Crops Research Institute, Central Institute of Freshwater Aquaculture, Indian Institute of Horticulture Research, Odisha Hydropower Corporation, Odisha Language, Literature and Culture Department, Central Institute of Women in Agriculture, Coconut Development Board, OMFED, Directorate of Animal Husbandry and Veterinary, Paradeep Phosphate Limited, Central Cattle Breeding Farm, Chiplima, Sambalpur, and last but not the least, Indian Meteorological Department, Government of India. I'm also thankful to all our coordinators and volunteers who have worked tirelessly in organizing this event, starting from our camera crew, our MSc, our moderator, my entire volunteer team. They've really helped me a lot in organizing this conference. I believe, like as directors have pointed out, there has been few lacuna and we still left out with few of the loopholes. And I assure you that in the upcoming versions, we'll certainly try to make it up. So if there is any lacuna or if there is any fault, I beg all your sincere apologies. So I thank all our print and publicity media for publicizing this event widely. And I expect this participation and this same spirit in our upcoming versions. Thank you so much, all the present esteemed delegates over here. Thank you so much. Jai Jagannath.
Thank you very much, sir. There is nothing great than analyzing your own fault, and definitely in future years, we are going to serve better. Now, I would request all our honorable dignitaries to join us off the dais, and we will move forward for the cultural session. We are very much thankful to all our honorable dignitaries for joining us and for sparing their valuable time with the insightful words. Thank you very much, all of you gentlemen. Please take your seats off the dais. A round of applause for all the honorable dignitaries. After a stage clearance, we would move forward for the cultural session. I request all of you to kindly be present. I would request team for a stage clearance.
Check.
Ladies and gentlemen, now is the time to indulge in some musical performance. Let's welcome our performers on the stage. Now we are going to witness another major dance form of Odisha, which is a Paika Nritya, Paika dance form, which is a form of battle dance, which is usually performed by the group of warriors, Odia army warriors. And let's welcome Khurda Paika Akhada, the performers of Khurda Paika Akhada for the performance. Let's welcome all of them. This dance form is usually performed with shield and sword, and this dance form usually shows the kind of strength the Odia army has its own blood, and they have a huge contribution towards the independence war as well. Let's welcome once again Khurda Paika Akhada, the performers of this group, with a round of applause. Could you please have the performers on the stage? अनुरोध करिबी समस्त कलाकार माननकु मंच उपरकु आसीबे एवं जो नृत्य आम मानक पई प्रेजेंट करबा पई समाने आज प्लान करिसेंती आपण मानक सम्मुखरे रखिबे एवं It's my kind request to all the performers. अनुरोध करिबी कलाकार माननकु मंच उपरकु आसीबे जोरदार करताली सहित स्वागत जणैबा. Let's have a round of applause to welcome Khurda Paika Akhada on the stage. Once again, introducing the performers of Khurda Paika Akhada. Paika dance or Paika Nritya is a form of battle dance. This dance is usually performed and practiced by the warriors of Odia army. Today we are having the amazing performers of Khurda Paika Akhada. The main target of the dance is to enhance the physical strength. Let us watch out this amazing performance. Could you have a round of applause? बहुत कम लोक अछंती किंतु मैं अनुरोध करिबी गोटे जोरदार तालीर सहित आमे स्वागत जणैबा हमर कलाकार माननकु मंच उपरकु. As you all can see, the performers are getting ready with the properties. Those who are meant to be used. These properties are meant to be used in the dance. Paika dance usually showcases the ability of performers to handle a weapon. Let's watch out this amazing skill. Could you have a huge round of applause?
হ্যালো হ্যালো এই যে দিଶୁଛି ଲାଲ ମାଟିର ଖୋର୍ଦ୍ଧା ଗାଡ଼ ଏଇ ଯେ ଦିଶୁଛି ଲାଲ ମାଟିର ଖୋର୍ଦ୍ଧା ଗାଡ଼ ଏ ବୀର ମାଟିର ଉଷ୍ମର ଗତି ଯୁଗ ଯୁଗ ଧରି ଉଷ୍ମତର ଆସ ହିଁ କଳ୍ପନା ସୁଦୂର ଅତୀତ ଆସ ହିଁ ଆସ କାନେ କାନେ ମୋର କହିଦିଅ ତାର ଗୌରବ ଭରା ସେଇ ଇତିହାସ ଦେଖାଇ ଦିଅ ହେ କଳ୍ପନା ରାଣୀ ଏ ଗଡ଼ ବୁକୁରେ ଶୁଭିଲା ଯେ ବାଣୀ ବାଜିଲା ଯେ ଦିନେ ସମର ବାଦ୍ୟ ବୋହିଲା ଯେ ଦିନେ ଶୋଷଣ ଆଦ୍ୟ କିଭଳି ଏ ଜାତି ସ୍ୱାଧୀନତା ପାଇଁ କିଭଳି ଏ ଜାତି ସ୍ୱାଧୀନତା ପାଇଁ ଯୁଦ୍ଧ କରିଛି ଭୀଷଣତର ଏଇ ଯେ ଦିଶୁଛି ଲାଲ ମାଟିର ଖୋର୍ଦ୍ଧା ଗଡ଼ ଯୋଉମାନଙ୍କର ତ୍ୟାଗ ଆଉ ବଳିଦାନ ଦିନେ ମାଟି ମାର ସମ୍ମାନ ଆଉ ସ୍ୱାଭିମାନକୁ ଫେରାଇ ଆଣିଥିଲା ଇଏ ସେଇ ପାଇକ ପୁଅ ଏଠି ଏକଲବ୍ୟ ପରି ସାଧନ ସାଜିଲେ ରଣହୁଙ୍କାର ଦେଇ ବଳିଷ୍ଠ ବହୁରେ ମନ୍ଥନ କଲେ ଅମୃତ ଗଲେ ପିଇ ଏଠି ସାଧନାରେ ମଜି ଶହୀଦ ହୋଇଲେ ଦେଶ କରି ଦେଇ ଧନ୍ୟ ଯଦି ଚିହ୍ନିବାର ଅଛି ଚିହ୍ନ ଇଏ ସବୁ ମାଟି ଠାରୁ ଭିନ୍ନ ତ ଜୋରଦାର କରତାଳି ହେଇଯିବ ପାଇକ ପୁଅ ମାନଙ୍କ ପାଇଁ
ଏହି ଥିଲା ମାଟିର କୋଟ ଝାଗଡ଼ ଏହି ଯଦି ଥିଲା ମାଟିର କୋଟ ଝାଗଡ଼ ବୋଧିର ମାଟିର ଅପମାନ ଆଗେ ଜୀବନ ଆମ ଦୁହେଁରେ ଗଡ଼ ଏହି ଯଦି ଥିଲା ମାଟିର କୋଟ ଝାଗଡ଼ ଏଇଟା କଲବ୍ୟ ପରି ସାଧନ ସାଜିଲେ ରଣହୁଙ୍କାର ଦେ ଏଇଟା କଲବ୍ୟ ପରି ସାଧନ ସାଜିଲେ ରଣହୁଙ୍କାର ଦେ ବଳିଷ୍ଠ ବାହୁରେ ମନ୍ଥନ କଲେ ଅମୃତ କଲେ ବି ଏ ସାଧନାର ମାଦି ସହିତ ଓଇଲା ଦେଶ କରିତେ ଏଇ ଧନ୍ୟ ଯଦି ଚିହ୍ନିବାର ଅଛି ଚିହ୍ନ ଏ ସବୁ ମାଟିରେ ଭିନ୍ନ ଦୁର୍ଗମଗିରି ଦୁର୍ଗ ପ୍ରାଚୀର ଛିନ୍ନ ଦୁଆରେ ବସି ଏଠି ଆକେ ତାନ୍ତ୍ରିକ ମନ୍ତ୍ରେ ସାଧନେ ଜାଗ୍ରତ ପୁରବାସୀ ଓ ପାଇକେ ଓ ପାଇକେ ଜୟ ମା ଭବାନୀ ଆରେ ଜୟ ଜଗନ୍ନାଥ ବନ୍ଧୁ ଆପଣଙ୍କ କଥା ହିଁ ଆମେ ଶକ୍ତି ଲଗେଇବୁ ଜୟ ଜଗନ୍ନାଥ
क्या बात है, जोरदार धमाकेदार तालियां सभी के हाथों से। Come on everyone, give it up for Khodha Paika Akhada. That is absolutely an amazing act. जोरदार ताली सबके हाथों में। Come on everyone. Hear it louder. Your magnificent act by Khodha Paika Akhada. Can I see everyone both hands up in the air? Come on everyone, जोरदार धमाकेदार तालियां और एक बार for this amazing act by Khodha Paika Akhada. सभी के हाथों से जोरदार तालियां। Come on everyone. Please hear it louder for Khodha Paika Akhada. Give them a huge round of applause. Absolutely brilliant. Could you please hear it louder, everyone? सभी का हाथ ऊपर.
जोरदार कंसल्ट सर्टिफिकेट पाई को ଯେଉଁମାନଙ୍କର ତ୍ୟାଗ ଆଉ ବଳିଦାନ ଥିଲେ ମାଟି ମାର ସମ୍ମାନ ଆଉ ସ୍ୱାଭିମାନ ଫେରାଇ ଆଣିଥିଲା। ନାହିଁ ସିନା ଜଗବନ୍ଧୁ, ନାହିଁ ସିନା ରାଜଗୁରୁ, ରାତି କାଳେ ମାଟି ମାର ସମ୍ମାନ ପାଇଁ ଲଢ, ସ୍ୱାଭିମାନ ପାଇଁ ଲଢ। ଆଉ ଦେଖାଇ ଦିଅରେ ପାଇକ ପୁଅ, ଏ ମାଟି ଦେ କେତେ ବଳିଆନ। ବର୍ଷାରେ କରି ବୈରୀ ଚର୍ଚ୍ଚା, ଦେଖାଇ ଦେଲାରେ ପାଇକ ବସା। ବର୍ଷାରେ କରି ବୈରୀ ଚର୍ଚ୍ଚା, ଦେଖାଇ ଦେଲାରେ ପାଇକ ବସା। ଜନ୍ମ ମାଟିର ଅପମାନ ଆଗେ ଜୀବନ ଆମର ନୁହେଁରେ ବଡ। ଏଇ ଯେ ଦିଶୁଛି ଲାଲ ମାଟିର ଖୋର୍ଦ୍ଧାଗଡ।
we can have this जोरदार समापन तालियां बजती रहनी चाहिए हमारे जवान ऐसे What a brilliant afternoon. दोनों हाथ ऊपर 15 is here, it's louder. जोरदार समापन ताली समापन
अभिमान को भरे जिला सिंह परी करती पाटी की गुलाई काकिले कामुड़ी बहा उठे रे उठे मोपाई को भाईनी हम राज्य को होई भाषा हम धर्म भूमि ना बड़े विदेशी देखा की विरोधी हम गाड़ी पर जिला जिले गंगा धार गोदावरी
What a brilliant act it was. A huge round of applause for all the performing artists of Khordha Paiko Akhada, everyone. Samastanga hataru jordar karatali. Could we please have a huge round of applause? Can I see everyone's both the hands up in there? Such an amazing act. Absolutely heart-throbbing. Ladies and gentlemen, let's give them a huge round of applause, a standing ovation. I request each one of you to please stand and clap for them. Let's have another round of applause for this amazing team. They have already represented Odisha in India's Got Talent. A huge round of applause for them. Jordar tali, dhamakedar taliyan sabhi ke haathon se. That is a masterpiece of art that deserve a huge round of applause. Once again, can I say everyone's both the hands up in there to congratulate the entire team of Khordha Paiko Akhada. अनेक अनेक अभिनंदन अनेक अनेक शुभेच्छा आपण माननकु निज जातिर स्वाभिमान सम्मान को बंचे रखिबा पई ए जो प्रयास ताहा आगु को मध्य जारी रहिबा आवश्यक वर्तमान मु अनुरोध करिबी एग्रीविजन प्रोग्राम डायरेक्टर इंजीनियर दिव्यांशु प्रसाद दास मंच को आसीबे एवं हम कलाकार माननकु सम्मानित करिबे टाइम टू फेलिसिटेट द एंटायर टीम for the kind of hard work, the effort they're putting into their art. That is one of its kind, everyone. Once again, let's hear it out for Khordha Paiko Akhada. That was a performance full of courage and strength. The entire team deserve a pat in the back. And we are very much fortunate to have all of them in AgriVision 2024. I have personally not seen such a performance before. There was like never before. बहुत ही सुंदर performance से थिला आंख और वो खुले के खुले ही रह गए but these performances are absolutely commendable. We are very much honored to felicitate the entire team of Khordha Paiko Akhada. ସମସ୍ତଙ୍କର ଜୋରଦାର କରତାଳି ସହିତ ଆମେ ସେମାନଙ୍କୁ ଶୁଭେଚ୍ଛା ଜଣାଇବା ଅଭିନନ୍ଦନ ଜଣାଇବା
ଓଡ଼ିଆ ପୁଅ ଓଡ଼ିଆ ମାଟି ପାଇଁ ଏଇଭଳି ଆଗକୁ ଗୌରବ ଆଣୁଥାନ୍ତୁ. We are very much proud of Kota Paika. Thank you so much for commendable performance. Let's hear it for entire team. Come on, everyone. ଅନେକ ଅନେକ ଧନ୍ୟବାଦ ଆମର ସମସ୍ତ କଳାକାର ମାନଙ୍କୁ ସୁନ୍ଦର ଭାବରେ ସମସ୍ତଙ୍କର ମନ କିଣିଛନ୍ତି. Once again, could you please have a huge round of applause? ତାଳି ଯେତେ ବାଜିଲେ ବି କମ ପଡିବ ଏଇଭଳି ଆକ୍ଟ ପାଇଁ. That was an amazing ending to the Agri Vision 2024. Now we would take this opportunity to felicitate the members of our organizing committee with a huge round of applause. I would request Dr. Soren Das from Indian Statistical Institute, Kolkata, West Bengal, to kindly come up on the stage to receive the award. I would request each one of you to have a huge round of applause for Dr. Soren Das. Thank you very much, Dr. Das. Each year, you are gracing the occasion. We are very much thankful to you. Now, I would request Mr. Sarada Prasanna Das to kindly come up on the stage to receive the award of Krusaka Sathi. He is having a YouTube channel as well. Sarada Prasanna Das on the stage, please, for the award of Krusaka Sathi, having a very popular YouTube channel. We congratulate you, sir. Let's have a huge round of applause for this young talent. Could you please have a round of applause? Congratulations. Now I would request Team Buna Vision to kindly come up on the stage. The people behind the IT support, the web management, I request them to kindly be on the stage to receive the award. Team Buna Vision on the stage, please. I would request the entire team to be on stage. Bunna bhainku mu request karibi on stage asibe. Give it up for Team Buna Vision.
Trading in futures and options today.
Team, please be on the stage. Entire team. Are you round for all the young talents behind the IT and web? Give it up for Team Buna Vision, everyone. Entire team will be on stage to receive the award. We are very much thankful to the entire team for the continuous effort to make Agri Vision a successful one. जोरदार ताली समस्त खातर everyone, come on. Thank you for carrying out the session, for connecting to the people over web. Effortlessly, you have done your job. Thank you so much. Our next organizing member, Debasis Nair. Thank you very much, Team Buna Vision. क्या बात है
కళ్ళు మూసుకుని పాలు పిల్లలాగా నేను నిన్ను చూడలేదని అనుకుంటే పిల్ల పుట్టుకెళ్ళ బాణమని కొమ్మ ముడిసి పోతే ఎల్లా నిన్ను పొదిలిన విల్లు మరి నేను మళ్ళా నా కంటి చూపును
Now I would request Debasis Nair to come up on the stage to receive the award. I would request Dibansu sir to kindly felicitate Debasis. Now please welcome Mr. Jeet Singh on the stage to receive the award. I would request Dibanshu sir to felicitate Biswajit. Give it up for the young minds behind Agri Vision 2024. Now, please welcome Jogendra Pradhan on the stage. Jogendra Pradhan.
अब दुल्हा दुल्हन के बारे में
These are the people behind
अरे
scene.
इनको छोड़ो शीता जी है 54 लेकिन स्टाइल ऑफ मिस्टर 2024 फ्लिपकार्ट है स्वीटी और शादी का गिफ्ट हाँ जी झुमका दिखा दिया जी क्या झुमका मतलब
Now, please welcome SK Kaidin. Round of applause for SK Kaidin. Please welcome Vasundhara Pradhan. Vasundhara, please be on the stage. Thank you so much, Vasundhara. Let's hear it for her. Give it up for our very lovely and talented host, Pinky Jha. She's not here. So I would be taking the opportunity to receive the award on her behalf. Give it up for Pinky Jha, everyone. Now, Biswajit Malik. Please be on the stage. Biswajit Malik.
ସବୁଠୁ ଶେଷରେ ଆଉ ସବୁଠୁ ମୂଲ୍ୟବାନ ଏବଂ ମହତ୍ୱପୂର୍ଣ୍ଣ ଆୱାର୍ଡ ମିସ୍ ଆଦ୍ୟାଶ୍ରୀ ଦିବ୍ୟଦର୍ଶିନୀଙ୍କୁ ଆମେ ଦଉଛୁ ଆଗ୍ରି ଭିଜନକୁ ସୁଚାରୁ ରୂପେ ଚଳେଇ ନେବା ପାଇଁ ଏବଂ ତା ସହିତ ସମ୍ପାଦନ କରିବା ସହିତ ଲୋକଙ୍କ ପାଖରେ ଇଂରାଜୀରେ ଯୋଉ ଆମର ଡେଲିଗେଟ୍ସ ଆସିଥିଲେ ତାଙ୍କ ମାନଙ୍କ ପାଖରେ ଇନଫରମେସନ ପହଞ୍ଚିବା ସହିତ ଓଡ଼ିଆରେ ମଧ୍ୟ ସମ୍ପାଦନା କରିଥିବାରୁ ବହୁତ ବହୁତ ଧନ୍ୟବାଦ ମିସ୍ ଆଦ୍ୟାଶ୍ରୀ ଦିବ୍ୟଦର୍ଶିନୀ ବହୁତ ବହୁତ ଧନ୍ୟବାଦ बहुत बहुत धन्यवाद मैडम को पाई ताली ही जाओ ताली 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 मिस ओके ओके मिस पूजा जेना सेठी प्रेजेंट ना आंती तांको बिहाफ रे अवार्ड को एक्सेप्ट करिवे मैडम
पूजा जेना on the stage, please. Nahanti, तांको behalf रे more team members को request करिबी award नेबे. Now I would request engineer Satyabrata Mohanty sir from ICAR-NRRI to kindly come up on the stage to receive the award. I would request you, sir, to please come down and receive your award. It's a kind request, please, sir. Without your support, it would not have been possible. So I would request you to kindly come downstairs to receive the award. And also engineer Abhinash Parida. Please give it up for both our young engineers, Engineer Abhinash Parida and Engineer Satyabrata Mohanty. Give it up for both the young engineers, Engineer Abhinash Parida and Engineer Satyabrat Mohanty. Thank you very much for your support. ଉପରୁ ଥାଇକି ଆମ ଇଶାରା ବୁଝିଛନ୍ତି and ବହୁତ ସୁଚାରୁ ରୂପେ ପରିଚାଳନା ପାଇଁ ସାହାଯ୍ୟ ମଧ୍ୟ କରିଛନ୍ତି ଜୋରଦାର କରତାଳି ସେମାନଙ୍କ ପାଇଁ ଇଞ୍ଜିନିୟର ଅବିନାଶ ପରିଡ଼ା. Let's hear it for both the young minds. Last but not the least, we are very much honored to welcome Dr. Biswajit Mandal from ICAR-NRRI, Cuttack. I would request sir to kindly come up on the stage. Thank you very much, sir, for your constant guidance and also for chairing the session. And he is the only person I'm seeing till last. So please give it up for Dr. Biswajit Mandal. A huge round of applause. Samasthanga hatare jordar karotali. We are very much honored to felicitate you, sir. Could you have a huge round of applause? Esteemed Principal Scientist, ICAR-NRRI Cuttack, Dr. Biswajit Mandal, ladies and gentlemen. ମୁଁ ଚାହିଁବି ଟିକେ ଜୋରଦାର କରତାଳି ସହିତ ସରଙ୍କୁ ଆମେ ଶୁଭେଚ୍ଛା ଜଣାଇବା. ବହୁତ ବହୁତ ଧନ୍ୟବାଦ sir. Now we would take a group photograph and now is the time to wrap up the show. Thank you very much once again, everyone present over here for making the international conference a very successful one. Thank you very much for your presence and for your patience to hear all our speakers. All our sessions also were very much interactive with all your presence. Once again, thank you very much, everyone. I would request each one of you to please be on stage for a group photograph. Thank you very much. Have a good time, everyone.